all the delegates. Chairman of the session is Dr. Anit K.N., Director of Research, Kerala Agricultural University, and the co-chairman is Dr. Sainamol Kurian P., Professor, Department of Plant Pathology, College of Agriculture, Padnakad. And the rapporteurs of the session are Dr. Nidish P., Dr. Manigandan N., Dr. Naishant N., and Mrs. Anjali A.S. We humbly request the chairman and the co-chairman to conduct the session. We request the rapporteurs also to join the session.
Very good morning to all of you. I am happy to be with you here and chairing a session on artificial intelligence and smart technologies for plant health management. Here we have today with us Dr. Hemilata N., Dean IT Academics, St. Aloysius College, Mangalore. She'll be talking on artificial intelligence and smart technologies for plant health management. It's a wide topic which will encompass so many aspects, starting from the detection, diagnosis, and then recommendations of various diseases, pests, etc. And it's an emerging field, and there are many people who are working, starting from small startups to multinational companies, and we are also going to be part of it. And I'm happy that we have with us Dr. Hemilata. He is an expert in IT, which could be connected to agriculture. Welcome, Dr. Hemilata. Thank you.
Good morning, all of you. A brief career profile of Dr. Hemilata N. She's a retired academician from the St. Aloysius deemed to be University Mangalore, where she has served as the Dean of School of Information Science and Technology. She has over 22 years of teaching experience, 4 years of industry experience. She holds a master's degree in computer applications and a master's in management with a specialization in information systems from the All India Management Association, New Delhi. Dr. Hemilata has published over 25 papers in reputed Scopus indexed journals and other conferences. She has been invited as resource person and keynote speaker in national and international conferences and faculty development programs also. She has also served as a reviewer for several international journals and conferences. She has co-authored 4 books related to machine learning in agriculture and has contributed several book chapters, edited volumes published in Springer, and she is a member of the Research Advisory Committee for Scholars at several national and international universities, including Kannur University, St. Aloysius deemed to be University. And Dr. Hemilata has received 2 copyrights and has applied for a patent also. She is a senior member of IEEE. At present, she is guiding more than 5 students in the various divisions of engineering. I cordially welcome Dr. Hemilata to deliver the first lead talk in session 2 and day 2 of the national seminar.
Thank you, ma'am, for the introduction. I really thank the organizers for having me here for this session. So thank you to the organizers once again. So a very good morning, one and all. So my topic today is AI and smart technologies for plant health management. So as Dr. Anit was saying, it's a very vast topic. So I think I'm given 20 minutes. Is it, ma'am? What's my time?
Ma'am, it is 15 plus 5 minutes discussion.
Fine. So 15 minutes I am given, and I don't know whether I'll be able to keep up that 15 minutes because it's very short. So I'll try to give you a brief intro, and I know many of you already have an idea of doing something in machine learning and deep learning. So I'll start my Can you just show me? So as I told you, I will just give you a brief intro of everything, and also like how actually in AI in agriculture, like when you introduce AI in agriculture, how actually it works. So this is just a brief intro. Everyone may be knowing this particular thing, like what is basically an AI. So it is nothing but a computer system which will help you to learn from data and support decision making. So when you talk about artificial intelligence, so we are trying to give some intelligence, and this intelligence is definitely given by data. So data is very, very important. So I'll be talking about the data in later stages. So in agriculture, you know how actually AI will be helping the agricultural part. So if you look at, you have in agriculture, you have crops, so definitely you may have pest diseases in crops, you may have to detect pests, you have to have yield prediction, so everything. See, these kind of things can be done with using AI in agriculture. So now when we make AI tools, so as I said, definitely we need data to process that, and what kind of data we can give it to AI. So definitely the data which can be given to AIs are crop images. So if suppose you are dealing with pests and diseases, it will be definitely images of crops. And then another data which can be given is sensor data. Okay, so if suppose you are working with sensors, so you will be giving sensor data, then it could be weather information, drone images. So drone, because if you are trying to implement AI in vast area, so definitely it could be drone, it could be satellite images, and at small I'll be touching upon the genomic data also. So as I told you, I won't be able to go deep into anything. I'll just give you a small idea, and in case you later need any help, definitely I'll be helping you. So supports. So basically AI in agriculture supports, another important aspect is precision agriculture. So supports precision agriculture by helping farmers to take the right action in right time. So precision farming is another important thing which definitely AI is helping. So that is another which machine learning and deep learning can be applied, and we can go for precision farming. Okay, now, so AI in agriculture as a whole, so now our topic is basically plant health management. So it is only, we are more concerned in today's session, we are more concerned with the plant health management. So when we talk about plant health management, definitely it could be different pests, diseases, it would be yield prediction, because definitely when you have this outbreak of pests and diseases, so it will be major loss for the yield. Then another one is, so now when we talk about AI, so why we are introducing AI in today's world. So one thing is like manual scouting is time consuming and depends on expert availability. So when you go with manual, for example, a farmer who have problem in his farm, and he has to go to an expert to consult it, and then he has to give the remedial measure, which takes a lot of time. So those can be overridden with this AI. Then early detection helps reduce crop damage and also pesticide misuse. That is another advantage which can be done using AI. Now AI supports faster database and location, and another important thing is location specific. So when we are collecting the data, so where we are trying to apply the AI, we have to collect the data from that particular location, and then we cannot generalize anything in particular in agriculture. That's what I understand. Now this is a general workflow. I'll be going little deeper in my next slide. So as I said, the first and foremost is nothing but the collection of data. So which I feel in agriculture is very, very difficult, because 6 of my students are working in this particular area, and usually in the case of health and all, when we work, we have vast amount of data in Kaggle and so many other repositories, but when we try to work with agriculture, nothing is available. Very less amount of data is available, and the students have to go to the field to collect the data. So first and foremost is collecting the data. Now again depends upon what kind of data they want. So some of my students are working in pest and diseases, so obviously they may need images through camera. Then if suppose you are working with something with respect to the sensors, it should be sensor data. And if suppose it's a vast amount of area, so then definitely drone. So in one of my students is working in coffee, so she actually is working with drone. So she has to collect, because she cannot go to each and every place to collect the data. So she is using drone, and drone images, drone data are taken. Then even she is working also with satellite data, because some of the images are also available through satellites. So ISRO, the data are available. And it could be weather data, and it could be genomic data also. Now once you collect the data, there are so many further steps which I'll go in the next slide, but basically what we have to do is analysis of the data, and this analysis could be like finding out like what are you trying to do, what is your problem. So based on that problem you will be doing the analysis. That comes the second part. Decision support, alerts, diagnosis and management recommendations, and field action. I'll be going deeper into the next slide. Yeah, so this is, I know it will be too much, I'm not able to, I cannot walk. Okay, fine. So this is basically the entire workflow, which is the most important part. So because I found this particular slide and I thought this tells the entire story. Okay, so the thing is, as I told you, I'll just put some more time into this slide. Yeah, I'll put some time into this slide because this tells the entire story of AI. So if suppose you are working in machine learning, so in AI when you work, either it could be machine learning or it could be deep learning. So I'm not going deep into that particular area, but how do you do that? So first one definitely is going to be the problem definition. So you should know what you are trying to work on. So if you are trying to work on pest diseases, or if you are trying to work on yield prediction, or you are trying to predict on some nutritional, whatever it is, so you have to define your problem. And that problem, so I'll read out in case back people can't read it. So next thing is, so in the problem definition, you should be selecting the crop, okay, selecting the crop which you are going to deal with, and also the environment, okay, because it could be the environment is very important in that case. And so these, once you define your problem, so you are through. Okay, what you want to do is in your hand. Next comes the data acquisition. That is second most important part. So now you have to, based on your problem, you will think what kind of data you are going to collect. Is it sensor data, it is drone data, satellite data, what kind of data? So data collection is very important. And this data can be collected, and then it could be the lab, and so you are collecting the data. That is the second part, data acquisition. Then comes a third part. Okay, so once you collect the data, so if suppose for example you are taking the images, if suppose you have dealing with the pests, so you are just going to take the image of pests, and that will be entire image. You don't require the entire image actually, so you need to annotate that. So that is called data annotation, and data annotation basically cannot be done by computer scientists. We cannot do that. We don't know anything about biology, so we have to go to the expert. We have to sit with the expert, and with the help of experts, we will be able to annotate, mark that. So that can be done by different things. So bounding box method can be done. You can do the segmentation task. So whichever you are trying to do, you have to label it. Okay, labeling, that is another most important part. So I may, for example, I should have positive data and negative data. Positive data will be my pest, for example, it will be pest, and the other one will be without pest. So I should have both the data, because computer should be able to identify which is the one which is having the pest and which is not having the pest. So I should have both the data, positive data and negative data. So that is the data annotation. And in the data annotation, we are also labeling. Remember that we are labeling, we are putting the classes. Okay, we are putting the classes, and these data, once we are through with our data labeling part or data annotation part, which is the most important part, I would say, because if this goes wrong, your entire thing goes wrong. So now, once you do the data annotation part, next thing comes your data management. So as I said, nowadays, whenever you try to find some agricultural data in any of the repositories, it is very, very less. So we don't have any data. So next comes the data storage. So you can have the metadata, and then you organize by crop, which crop you are collecting, which pest you are collecting, diseases. So you organize the data, and then it could be again organized based on location. As I said, the plant data, whenever you are taking, it is with respect to location. So location data, so you have to segregate that accordingly. Also ensure data quality, because data quality is very, very important. So that anyway we will be doing working out with the data quality in our next step. So first thing is only you are collecting the data, you are just whatever data you are collected, you are just annotating. Now comes your another most important part, data pre-processing. Because whenever you collect the data may not be very clean. It may have noises. It may have unnecessary things. So you have to remove that. Because this unnecessary information in the data may lead you to unreliabilities, non-reliability. So you have to remove those kind of parts. So cleaning and deduplication. Sometimes the data may be duplicated. So duplicate data also we don't want. So in the data pre-processing, you have lots of steps. So if you're doing with machine learning, there are lots of steps in machine learning. If you're going with deep learning, there are again other steps. So here, whichever learning you are applying, machine learning or deep learning, you have to go with the data pre-processing. Steps are a little different. Now this here, basically more the steps are written in with respect to deep learning. So once you do that, another one is resizing, because you would have collected the data in different places in different locations. So the data sizes may be different. So you have to resize it. That is another important part. When you go with deep learning, resizing is another important part. Then you have to normalize it. That again depends upon your problem. Then you have another important part is augmentation. You would have heard the word augmentation. Augmenting is nothing but increasing the numbers. Because in deep learning, whenever you are applying deep learning, 100, 200 data may not be enough. You have to augment the data. You have to increase the number of data. And in images, for example, the augmentation is quite easy, because suppose you have an image in this shape, it can be rotated in different. That rotation could be one of the methods to augment the image. Similarly, there are many other methods. So you have to augment the image, and then handling imbalance. That is another important part. These are some of the important technical terms which will come in deep learning or machine learning if you are going with artificial intelligence. Imbalance means I was telling you about positive data and negative data. That is, for example, if you are working with the pest, you are having image with pest, without pest. So the balance of this with pest and without pest should be maintained. If that imbalance is there, definitely that will reflect. Because you know that, for example, when a teacher is teaching you and the teacher is teaching only the positive side, negative is not taught. So obviously when a question is asked, the kid will always have a tendency to answer the positive side, because he knows positive more. Negative was not taught. So similarly in the case of machine also, if images without pest is not given, it may not understand. So you have to balance that. That is very important. So class imbalance has to be taken care of. Then missing values. So these kind of things are common. And then fuse multimodal data, because you are having multiple kind of features. So all this has to be put together. So that is data pre-processing. So I hope till now it's clear, because you have to do lot of things before you come to the model. Now comes another most important one, feature extraction and representation. Feature extraction means, for example, usually we use biometric. Why? Because that is a unique thing. Okay. So similarly, you have to find the unique quality in your data. Okay. So unique quality, those are called features. Features or attributes, we can say in another term. So you have to find out what are the features which can be used for recognition. Now in machine learning, we have to identify the features. In deep learning, we don't have to identify. Deep learning automatically identifies, because in deep learning, we are giving in the form of pixels. So but then you have to apply the techniques to extract the features. Even you can use generative AI to extract the features. So that the feature extraction is very, very important, because without features, it may not be able to identify which is with pest and without pest. So feature extraction and representation. So visual features from leaves, lesions, color, texture. So it could be any kind of, these kind of features can be used to identify one from another. Then it could be sometimes it could be time series data, but in our case time series will be less. Then environmental features has to be taken care. Multimodal feature extraction, feature integration. That is, you are having different features, so you are integrating the features, because more number of features, it will be better for the computer to understand the difference between A and B. So that is feature extraction. Once you are through with this, then comes your model. So now this 1 to 6 step is basically you are playing with the data. You are taking the data, cleaning the data, pre-processing the data, identifying the features from it, and then an annotation is in between there, and then finally extracting the features. So once you are through with this 1 to 6 steps, you are through with the features. Now comes the important part, model development. So now this is the place where you are trying to find out what are you trying to do. Are you trying to do classification? Are you trying to do detection or segmentation or forecasting? What is your problem? So you would have defined the problem in the very beginning. What is your problem? If I am saying that pest identification or pest detection is my problem, so you can go with classification and detection. If it is segmentation, you are trying to segment the area where actually the problem is, that is another problem. So you have to define your problem, and based on the problem, you have to take out whichever the algorithm you are going to apply. Are you going to apply the transformer, or are you going to apply the CNNs? That depends upon ensemble models, machine learning, deep learning. What are you trying to do? So that actually will be clearly told over here, model development and training. So once you do the model development and training, what happened? Okay, so once the model is ready, so next thing is you have to, so you don't know how good is your model, so you have to validate the model. So for validation of the model, you have so many performance metrics which you have to calculate. So if suppose you are going with machine learning, you have to find what is the accuracy of your model. Is it having 90% or 80%, 60%, etc. So similarly, there are so many metrics. There is something called F1 score, precision, recall. These are machine learning, basically machine learning performance metrics. If it is deep learning, there are other metrics. Similarly, you can have something called IOU, that is intersection over union. This is basically this IOU, actually I found out yesterday when I was taking out this. So this is basically AI is telling you that this is a problem. So you are just confirming that with the experts, and then you are calculating the IOU. If you are having an IOU score of 1, that means what AI told and what expert told is correct. So it is matching. So that means your model is very, very good. So you can take out the IOU once you do your deep learning model. So similarly, you can calculate confusion matrix is another performance matrix. So you can have so many robustness checks. So even external validation or new field data also can be done to evaluate how good is your model. Then comes, so till here 8, the model is ready, it can be used. Now further is actually, this 9th is basically once this AI and generative AI has come, this, there is something called explainability AI. Okay, so this is nothing but once you build your model, it is just checking whether the one, actually the place where it has told that there is a problem, that is the actual problem only. So that is the saliency measure or Gradsium measure or feature importance. So similarly, you can find out the bias, uncertainty of your model, and actionability for users. Finally, another one is deployment, that is you have to deploy. So deployment can be either through IoT device, it could be in your mobile app, you can have an Android mobile app, or you can have an edge device. Edge device is something where you are taking the picture and instantly you are getting the output, something called Raspberry. There are Raspberry device, Raspberry Pi, so you can actually have, you can take the pic and then find, it can tell you whether the problem is existing or not instantly. So that is called edge devices. So you can either have in the edge devices or you can have in the cloud. So cloud will take some time because you take the pic, it will go to the cloud, calculate, it will go through the model and give you the answer. That will take time. So depends on, if you want real time, it has to be in edge devices. Then integration with farm advisory systems. Similarly, decisions, those are the regular things, decision support and intervention, and then finally monitoring, feedback and control. So it is, so this, if you see the down, so this 12 is linked to 7, that is the data, the new data which comes, because when you make a model, it cannot be for next 20 years. So many new data are coming in, so it has to be updated. So when you get the new data, it has to go through the development, model development. So this goes again and again and again. So the model which you build, you have to update all the time with new data, because always the data has to change, because it cannot be for long term. Yes. So this was the basic principle of AI. If suppose those who are interested for building an AI application tool, definitely they have to go with this machine learning or deep learning, the method which I have just told. Now, okay, so this is use case, yes, use case. So this use case I'm not going explaining further. So I was just finding out like what are the tools currently available. So it seems there are plenty of tools available in India and abroad also, something like Plantic Speed. Those are mobile app which can be used for identifying pest diseases and nutritional disorders. Similarly, you have something called the Dihat Kisan app, Agrio. So these are some of the apps which basically works in this plants, diseases and nutrient disorders. So this is a case, so this is how actually it should work. The farmer will take the pic, and the pic will be identified, and it will be giving the result whether that is actually a disease or not. So this is the Plantix, etc., those kind of apps, and even this is what we are also trying to do. Next one. Now another one is sensor data. Now in this case, the sensor data could be, now for example, we are trying to measure the humidity. So you will be using the humidity sensor. So from there, the data will be extracted, and that data will be definitely, as I told you, we can take the sensor data. Rest all are same. Rest you have to go with the machine learning or deep learning model, build the model, deploy the model, and get the result. So there are some app which is already available, Fasal and GeoKrishi. These are some of the apps which are working with the sensor data. Okay, so this is how, for example, sensor can be kept wherever you want to measure the information, and then from that sensor data can be collected, and that can be converted and pre-processed, and we can go with our machine learning or deep learning model. Now another one, as I said, another deployment can be in robotics or in AI, IoT devices. So here I think I was, so this is Garuda, so here basically it is with respect to IoT devices. So for example, some of the applications which are already existing is Garuda Aerospace AgriKisan drone. So this is basically working with the drone data. Then this is a foreign product. So here all they are using drone applications, drone data. So here, so this is something like a precision farming. Precision farming, they are using the drone data, and they are trying to identify the places which is having more diseases, which is having less diseases, and based on that they are trying to give the remedial measures. Now another one, as I said, this is again something with the drones. So here they are using the drone data, and to show the picture, next one. Here again something similar to precision farming which I shown you. We are collecting the data. So from that data itself, it is identifying and giving the how severity is that particular area. So based on that severity, it is trying to give you the remedial measure. So this is again all how to collect the data. So here the data is collected through UAV devices. Now the last one is bioinformatics. So I don't know here how many people are working in bioinformatics. So that was another area in my PhD also. I was working a little in bioinformatics. So that is another area. So here the data is not the one which we were talking about. It is not sensor data or what do you say, like photos. It is something like the genetic data. It could be genes, it could be proteins, it could be microarrays, it could be anything of that sort. So you have to take the data, it could be genetic, so those kind of data are definitely available. You have that in NIH and in all the repositories, the data are available. So based again, you have to define your problem, what is your problem, and if suppose you are able to get the data from that repository, then you can further work on that. So AI helps identify pathogen races, mutations, virulence, resistant genes. So all this again, as I told you, depends on your problem. What is your problem, and then define your problem, and according to that take the data and then define the features. So again, to take the features in genetic data is little different from the one which I was telling you. It is not like that. You have to identify how to extract features from genetic data. That is little different. Anyway, I'm not going to explain that right now. So it is, there are many companies which are working in this genetic data also. Now, for example, in India, IIT Indore is working on that. They have an IIT Indore Agri Hub, it seems, which is basically AI-driven genomics. So that is a wonderful area again which we can work on. Since you have an idea on biology, bioinformatics is another beautiful area where you can actually implement the machine learning or deep learning part. So it has been shown. See, for example, genomic surveillance of wheat rust pathogens. So as I said, the first one, they are taking the data. That data has to be in genetic data or the protein data. Then you have to sequence it, because if suppose it is not sequenced, you have to go with sequence. Again, it's not so easy way, because if suppose the data is available in what do you say, like NIH and all data is available, no problem. If the data is not available, you have to sequence the data. So that again is costly. You have to sequence the data and then do the bioinformatic analysis. You can go with bioinformatic analysis and then you can go with the machine learning part. So this, you can integrate bioinformatics and AI, which is again another beautiful way of bringing AI tools. So this is another area where we can work on with respect to AI using bioinformatics. So true, AI has really helped the farmers, or I would say that the normal people with actually the whole scenario is now fastened up. It's not so, now it's not like a farmer is identifying the disease and is going to an expert. So all this can be integrated and can be done fast, but again, always challenges are there. So you always have to have the, as I said, the data should be very much specific, clean. Suppose your data is not correct, entire thing goes wrong. Limited internet, and yes, for rural areas it's not so easy. High cost of sensors, drones and advanced tools, if it is drone data or satellite data. Farmers need training and local language advisory support. So all those things has to be done. Only then we can have this tool in our, in the local with farmers. Next one. So to conclude, as I said, this has definitely, AI has definitely improved early detection and responses. It has reduced crop loss. It may definitely, if suppose we have a tool like that, it will reduce crop loss, pesticide misuse and production cost, integration of AI sensors, drones, genomic supports, the resilient agriculture, and definitely farmer-friendly tools and local language advisory system essential for adoption. So it is easy to say, I know it's easy to say, but it's not very easy to implement. Okay, to give it to the farmers, I don't know how much is happening in the current scenario, but if it happens, if we have precision farming, etc. in India, it will be really beautiful, and that's what we are trying to do. I don't know how far we can reach. Even Pitamaran sir was saying that he is also working in that. So yes, everyone has to look into it, because I know in agriculture lots and lots of data are available, but we have to get it and we have to put that into a database so that every person can use it, and they can do also something like health data which is currently available. So that's all I have to say. So thank you one and all. So open to questions.
Thank you very much, Dr. Hemilata. Of course, I'm proud to say that Kerala Agriculture University is collaborating with their institute to have an application developed to identify a small thing, but it has got so much of application just to identify the black pepper varieties which we have developed from 1 to 9, just based on the digital imagery. Okay, we have already given consent for that. Hope that their institute will be coming up with a solution for that, so that there is a lot of confusion among us and among the farmers as well. So far as the identification of these varieties based on the morphometric parameters are concerned. Thank you very much. Of course, she has been given 15 minutes time. Even if we give her 15 days time, and it is difficult for her to sensitize all of us in the ongoing research areas which are connected with information technology as well as artificial intelligence. Of course, we have two different areas as agriculturists with which we will be able to work on these aspects. One is research-based application. They do the research and then we develop the application. Artificial intelligence and CNN networks and other stuff, they do work and give us some information so that we do the application based on that. Another one is application-based research. That is the last one which we have seen, especially in the case of genomics and other thing. Our research would be based on the application which they develop. And in two aspects, we could contribute very much. Of course, there are some challenges which are involved. One is the mechanization that is needed for carrying out such precision farming system, as well as the carrying out the applications developed by AI tools. That is one thing. In small farm holds, it is a bit difficult. Of course, when it comes to urban agriculture, you have not dealt with that. Sometimes that is one area that is getting emerged. Urban agriculture, where remotely controlled equipments and then sprays and such things are used. One sitting in Dubai can control, of course, everything that is happening in the terrace of his building. There also a lot of research is being done, and investment in urban agriculture is going to pay something big in Kerala, because getting farmlands for cultivation in other parts is going to be very difficult, and this is something which AI could help very much. Okay, that's all. And I'm happy that Dr. Sainwal and her team is into the business, and then they have been developing some tools for early detection of plant pathogens based on the imageries, and of course that I think the second part based on the genomics also you may have to develop, so that a single drop of the sap may give you some big idea regarding the pathogen attack and lead you to better decision making in controlling the disease. Thank you very much. Thank you once again, Dr. Hemilata.
Can wind up, right?
Dr. Hemilata, I have a small doubt.
Yeah.
See, you are talking about image data. So I'm also working on that, using that.
Yes.
So if image data, whether it should be completely clean or perfect images, or is it okay with some blurred images?
The blur images, what happens, when we are giving the blur images, if you're going with deep learning, of the feature extraction will be very difficult. You identify if the picture is not very clean. So if suppose that area wherever that attack is there, if the annotation, it may not be able to properly annotate. The annotation is not proper, the entire thing goes wrong. So
because we cannot expect the farmers to
I know.
it's given to farmers.
We are having this problem. So what we have to do is those kind of images we have to discard. We have to discard. If you go through that, we will have n number of images, but that may not help the deep learning to identify.
Thank
so
you.
That is very important.
Thank you.
Yeah. Yes, sir.
See, nowadays I'm getting a lot of photos from the farmers
Yes.
to identify disease. But the problem I am facing is they send that leaf with a spot.
Yes.
That's only that. But for diagnosing, we require a field data, plant data, as well as the actual symptoms. So they said, I have posted in the AA, and the AI is saying this is a disease. Many a times these are not diseases.
Yes.
Maybe a deficiency symptom or something like that. So what I'm saying is, we should educate the farmers for diagnosis of the pan, how to take the data.
True, sir. That should happen. So once we have a proper tool, as I said over the last, that was one of the line. We have to educate the farmers how they have to take the pictures or how they have to actually use the system. It has to be educated.
That is one problem that we are facing nowadays, because many of the farmers are sending only the actual symptoms. That doesn't give any.
That may not give. That may not.
With that, many of them are recommending you do this thing, you do that.
Yes. Which may not.
That is one problem which
Which may not be
another
correct.
thing is that in the plantation diagnosis, many cases we are directly thinking that is a pest or disease, but maybe due to many other factors.
Many other factors. Nutritional problem also.
That is not usually seen in many of the pictures that you have.
Sir, that is what. So I have a doubt. For example, Plantix software is there. So Plantix, it says it is for all the crops. Sir, is it possible to make one general software? It's not possible, right? So because I have seen. That is what. So we cannot have one software for all the crops. It should be individually for that particular crop. So I don't know, because many area based. Yes, exactly. So that is why that. Yes. So even I think IRI also has come up with tools which are general for all the pesticides. I don't know. I got to know one tool, but that may not be correct, no sir, because it may not lead to the correct answer, isn't it? For one crop.
And also, ma'am, in this Plantix and other sites, no, we get the disease in the last stage or the typical symptom. So we want to detect the disease at an earlier stage. So that type of pictures, images are not available. Many of the websites.
Yes.
So we
have to take
it
entire picture
our
from
own,
the starting to the end. Yes.
Ma'am.
Yes. Yes.
So you said that drone-based identification. So how does the topography of India will influence the drone-based identification and plant health management?
No, the thing is, for example, if you're taking a large area, you cannot go to each and every place and take the picture. So drone picture has to be taken. So that again, drone also, there are different levels. From what level you have to take? You have to I think you can program that, because one of my students is doing that. So she actually said we can program that from what level we are taking. So based on that level, your clarity of the picture differs.
So is it plain areas are there and hilly areas?
Hilly areas.
So is that any of that if we take the hill stations, the area will be slightly like 90 degree or 60 degree. So is there any problem with
problem may be there definitely. Yeah, you have to do so many adjustments in drone. You have to. That problem will be definitely there. Yes. Yes, sir. It is possible. It is possible. You have to program it. Yes.
Congratulations.
Yes, sir. Thank you.
I'm really appreciative. Asking to your says the land means good land means properties than making in robotic. I mean robotic like that. They explain this how to make and kind of robotic agriculture's details.
See, robotics actually, robotics, that's what, and the last one I think I've shown you something like, see, in robotics also you have to use sensor data. You can take the information and then you are trying to apply through robots, that's all. Thing is, you are making a machine learning or deep learning tool, and through drone you are trying, through robots you are trying to apply, instead of humans, it is robots which is doing that. But model is safe, model building is safe. Yeah, it will be safe, yes. Yeah, robots take over. It is a, deployment. It is a deployment tool. You are deploying through robots, that's all.
Ma'am.
Yeah.
Ma'am, in deep learning models, what are the criteria for choosing the models?
Deep learning, again, if it is images, it is better to go with CNN. If it is a text kind of thing, it is better to go with RNN. And so that again, so what is your data?
Ma'am, in my case, deep learning model for anthracnose disease of cowpea.
Sorry?
Disease,
Images only. CNN is okay. And so many inbuilt models are available. So ResNet, VGGNet, so many that again you have to check out which model is best for your application, mobile net. So you have to check out which algorithm will be best. CNN is best. It will work out. I think I can stop. So thank you for patient listening. Thank you once again.
Now we request the chair and co-chair to present a memento to Dr. Hemalata N. on behalf of Malabar Plant Protection Society.
The next lead talk will be by Dr. Selvarajan, and he'll be joining us a bit late. Therefore, we are going to start the oral presentations. Thank you.
The first oral presentation,
Hello.
T2OP1, Bharat AR. Are you ready, Bharat?
Hello.
Hello, Bharat? Hello, Bharat?
Hello, is it audible?
Yeah, you can present now.
Yeah, okay. So I'll be just talking about the topic compact and explainable AI explainable approach for banana leaf disease detection. So in this paper, I just gone through about how we can implement CNN model or deep learning model for agriculture data. So in this, I have just focused on three different diseases and one healthy images. Basically, as I mentioned in the lead talks, we want the positive as well as negative images. So negative images we here informs diseases, and positive images we inform healthy images. So as we can see here, banana is economically important crop in tropical agriculture. So actually we lack the early detections. So we having the agriculture or important this one, but we lack the early detections. So I have taken three disease and one healthy images. In this case, traditional disease identification. So basically it will be depending on the experts. So as soon as we figure out any disease or we have come up with any diseases, we'll be not sure which disease is that, what we need to do for overcome that or to avoid that or detect that. So it will be detected on expert inspection. So it will be time consuming and it will be we need to consult an expert or etc. It's a bit more complicated. So deep learning and CNN provide the automated disease detection from leaf images. So we have multiple things when you get the data, we can build a CNN model, and if you want for further steps, we can even deploy. So as ma'am was mentioning that we can just take a photo and we can just detect which is a disease or not. So we lack lightweighted model explainable predictions for real farming applications. As I just come up with that, just researching on that, I have figured out we lack the lightweighted models. We have the complex models only, as of now. So what will be disadvantage in that case is whenever you want to convert into an application. So it will be like in some cases accuracy drop will happen. Sometimes the prediction will be not that accurate because of you converted the original uh, parameters into the conversion thing. So it will be like a kind of drawback, I can say. So the main objective is like develop a lightweight CNN model and classify the disease as well as a healthy one. And I have just tried to compare with the gap and without gap. So basically that is like when you're building a model, it will be without gap. I mean the flattened model, but with gap it will be like it will be reduced params. So it will be like good for this thing. And to improve, to evaluate model generation using unseen real data. So unseen real data, actually what happens here, like if you train and validate the model using the augment data basically is like original data which you collected, but the thing is like original data you will be having the proper one, and augmented thing like the one image you will be augmenting using different the thing like zoom or flip horizontally everything. So it will be like no, we can't call it as directly the real one. Like when you're after building a model, if you want to host or deploy anything, you can't really call it as a real one because when you're taking the image, it will be like in different shape or like in different angle. So augmenting. So I have tried like trying augment for training and validation, and the real image for testing purpose only. To improve interpretability, I have used GrandCam visualization. So basically, GrandCam visualization is like we can use explainable AI, and once once the model is detecting the proper area, we can with confidence we can say our model is predicting good, and in future the model will be helpful for the real world scenarios. So design models suitable for agriculture field deployment. As I already mentioned, like lightweight and what I have targeted, and when you come to material and methods, so as I mentioned, data is taken from Kaggle and it has 4 classes. And data split is like augmented set for training and validation, and version for only for testing. So as you can see, just the data set sizes, we have multiple images for different types, and pre-processing, resizing, pixel normalization, and data augmentation. That's like normal pre-processing steps. And the main architecture, when you can see, it's a lightweight CNN architecture, conversion blocks of filter, 120 filter, and filters. And when you if you come for result and discussion, you can see there the accuracies of different models. I mean the with gap, without gap, and we have tried one more fine-tune gap. Just tried the fine-tuning of that model. So you can see 90% for training and 87.50 for validation. But when you are coming for test accuracy, it is like it increased. So you should be keep in mind it is like we have given augment for training and validation, and the real image for testing. So when it comes for key finding, I can just mention model without gap achieved highest test accuracy, and gap improved stability, training stabilities. Flattening preserved special disease feature better distinction between visually similar diseases. You can just see the graph as well there. And when you come for performance observation, a high recall for cordon and sigatoka, like we got high recall percentage, and that is a different types of validating the recall, precision, everything, and good precision as well for this disease. Healthy leaves occasionally misclassified because that for early stage and healthy is like almost like kind of similar, so it will be sometimes misclassified. You can see the confusion matrix here. The diagnosis is like how much predicted properly, and like the other thing is we can see healthy which misclassified, and confusion matrix insights, codon and sigatoka classified reliable, and detect symptoms cause some overlap. So important contribution here is like clean data set separation, prevented data leakage. So basically if you want to say the model is like good model or something, you should be always focus on the original image, so prevent it. So you can just try in the augment set and all. If it is performing better in the real image, you can be going for the deployment of that model. Testing performed only on unseen images. That is what I just mentioned. And when you come for training and behavior, without gap, fasten canvas and slight overfitting observed without gap. And when it comes for gap models, more stable learning curves and better generalization behavior. And in explainable AI, GANCAM, what I have just mentioned is like GANCAM highlights disease affected regions. And the model focused on lesions, dark spots, or infected tissues. Importance like improved trust and interpretability, useful for practical agriculture deployments. So that silence findings and conclusions like banana sequence achieved 87% for text accuracy, lightweight architecture suitable for field application, so which can be deployed in the future purpose as well. And model without gap performed based on unseen data. Grand can confirmed meaningful feature learnings, strong performance with reduced computational complexity. So proposed model provides, the conclusion is like proposed model provides accurate, lightweight, and explained disease detection suitable for model and smart agriculture system, can support agriculture diagnosis. So just I want to acknowledge Interashes and partner card and my research guide and collaborators. Thank you.
Thank you, Mr. Bharat. Am I audible?
Yeah, you're audible.
Yeah, one question that comes to my mind is that, for training your models, you have used 1,281 imageries. Don't you think that this is very less in number when it comes to the use of imageries?
Yeah, it's less images. Like as I mentioned, I have taken from Kaggle, banana elastic dataset. So that is what even when ma'am was talking, she was telling the images or like the data set, we are lacking. Like we have some images, but not that images. But for building some models, it is enough. Not that compatible, but when you're taking when deployment scenarios, the images is not sufficient. So in this case, we need to collect the data. So this is like a study I have done from existing images. So the total images only it has around 2000. So in that I have just split that to training, validation, and test images.
Okay, have you come across any mixed infection when it comes to the foliar diseases? And if so, what is the accuracy of getting 2 of the diseases or 2 to 3 of the diseases getting diagnosed simultaneously?
You mean misclassification? You mean misclassification?
Pardon me?
Yeah, you mean misclassification you are asking?
Mixed infection, Sigatoka with Bacillopsis or something of that as a scenario that would come, the leaves would have different diseases occurring simultaneously. You have trained your model with specific images for specific diseases. My question is, what is the possibility of getting 2 diseases, mixed infection of 2 diseases detected accurately?
Yeah, so when the classes will be classifying, right? So it will be already classified from the data set. So when we classifying the disease, if 2 diseases were there, highest percentage, so that will be classified as 1 disease. And when prediction also, it will be seeing how much confident he has. So depending on that, it will be classified.
Okay, thank you.
Try to keep in the time and
Audience?
all.
Any questions?
For this oral presentation, we have 7 minutes time, 7 minutes and 1 minute for discussion. And the second one is T2OP2, early detection of Ralstonia wilt in tomato through reflectance spectroscopy. Anusha, are you ready?
Yes, sir, I'm ready.
Yes.
Can I start?
Wait. Yes, you can. No, one minute. Yeah, you can.
Okay, thank you. Good evening to everybody. I'm Risha.
It's not audible.
As the video will talk about.
No, it's not
It's
clear.
audible.
Anusha,
Okay.
it's not clear.
What about now, sir?
Yeah, please.
Okay, I'm going to present my work on early stage detection of Ralstonia wilt in tomato using reflectance spectroscopy. As we know, the tomato crop is seriously affected by the bacterial wilt disease caused by the Ralstonia solanacearum species complex, which poses a serious threat to tomato production in India. Unlike other diseases, the bacterial wilt does not produce any characteristic symptoms, and also it is of no use to take up any management practice after we observe the symptoms, because the plant would have been damaged beyond any scope for recovery. Hence, it is very important for us to explore early tools that can enable early or pre-visual disease detection. Hence, we need to look for rapid non-invasive disease detection techniques. The objectives of my research are hence to spectrally phenotype the Ralstonia infected tomato plant and then to identify the key spectral markers that would enable pre-visual disease detection of bacterial wilt on tomato. The workflow is as follows. Next slide, please. Three treatments are considered: healthy plants which served as negative control, plants subjected to pathological wilt which was the actual treatment, and plants subjected to moisture stress which served as the positive control. Different sensors were used to capture different data from the electromagnetic spectrum, which included RGB imaging, then data from the VNIR region, which was obtained as hyperspectral data, as well as the quantum efficiency of photosystem 2 was measured by calculating the FE/F ratio. The data obtained was initially processed for noise and by data analysis, which involved identifying the discriminatory bandwidths that would enable early detection and identifying suitable reflectance indices that would enable pre-visual disease detection of bacterial wilt of tomato. Next slide. RGB image analysis revealed that the control plants showed no signs of stress, and there was also increase in biomass over time, whereas the inoculated plants began to show visual disease symptoms from the 8th day post-inoculation, whereas the moisture stress plants showed symptoms from the 12th day onwards. So using the mean reflectance data obtained by the hyperspectral camera, we plotted the spectral reflectance curves over the days of observation. Observations were taken at every 4 days intervals for all the 3 treatments. Here we can observe that on the first day of observation, the curves of all the treatments tend to overlap. But as the days progress, we can see that the stress treatments, that is both the inoculated as well as the moisture stress treatments, show significantly reduced reflectance compared to the control. The inoculated plants show reduction in reflectance on the 4th day, whereas the moisture stress plants begin to show reduced reflectance, that is from the 12th day onwards. Next slide. To further study, to further understand the difference in reflectance between inoculated and controlled plants, mean reflectance values inoculated and the different spectra plotted. We can observe in the different spectra shown in figure 4 that on the 0th day, there is not much difference between inoculated and control, but as the days progress, there is consistently reduced reflectance beyond 700 nanometers, with a degree of difference increasing with each successive observation. Hence, to identify the discriminatory bandwidths, we used a supervised feature selection method termed the relief algorithm. This algorithm assigns weights to the wavelengths based on their discriminatory potential. Higher the weight, higher the discriminatory power. So based on the peaks obtained in the curve, we identified the bandwidths 700 to 780 nanometers, 750 to 780 nanometers, and 930 to 950 nanometers. These bandwidths were further validated using students' t-test. Can you go to the next slide? Yeah, this one right here. No, the t-test table. Here we can see that it was only in the 750 to 780 nanometer and the 930 to 950 nanometer bandwidth that we got statistically significant difference between inoculated and control plants right from the 4th day of treatment imposition, that is around 4 days before visual symptoms appeared. Hence, these two bandwidths have pre-symptomatic diagnostic potential. Right? Several spectral indices were also evaluated for their early disease detection potential, but out of 7 moisture stress related indices evaluated, there were 3 indices that showed good correlation to moisture stress and also had capability of early detection. They were the NDVI red edge index, NDWI, and the red by NVR index. In all 3 indices, we can see that the inoculated plant shows significantly different value compared to control 4th day, whereas the drought stress begin to show difference from the 12th day on. That is actually after visual symptoms have been appearing, the visual symptoms of drought stress. We also calculated the impact on photosynthesis by measuring the Fe by Fm value using the handheld fluorometer. Here we can see that in the inoculated plants, a significant reduction in Fe by Fm value was obtained on the 8th day, coinciding with the appearance of visual symptoms. Hence, this tool is not useful for early detection. Next slide. The salient findings of my study are: the effective bandwidths identified for pre-symptomatic bacterial detection are the bandwidths 750 to 780 nanometers and 9 to 950 nanometers. Both of them lie in the system. Also, all of those will turn into and it can also be used for the development of AI-based disease detection models to enable pre-visual, early, non-invasive, large-scale disease detection of bacterial wilt, particularly in endemic regions. Next slide. I would like to acknowledge my chairperson, my lab members, Division of Plant Pathology and Nanaji Deshmukh Plant Phenomics Facility, ICIRI New Delhi. And I would also like to thank MPPS for providing this opportunity. Thank you.
Thank you, Anusha. Am I audible?
Yes, sir.
Okay, just one question. What was the differences in spectral variability between the inoculated wilted plants and the moisture stressed plants? You have compared in the penultimate slide that there were some spectral differences between the control and the wilted plants, bacterial wilted plants.
Yes, sir.
Previous slide, please. Okay. Validation using students t-test revealed that only 750, 85 nanometer and 950 showed significant difference between inoculated and healthy plants. Now our idea was to have detecting the differences between the wilted, bacterial wilted plants and moisture stressed ones. Did you do any analysis on that aspect?
No, sir. The moisture stress treatment was only taken as a positive control to see if the spectral the trends are similar to that are similar in both cases or not. And so I did not validate for the moisture stress plant, sir.
Did you get it validated in the field?
No, sir, that I should, field level trials should be carried out. And maybe I should try to validate, the bandwidths that I have got and the results that I have got, I have to use larger data sets to see if these results still hold true or are still valid or not.
Okay, thank you.
questions from the audience? Students?
Moving to the next presentation. T2OP3.
What about the lead talk 2?
No, I think you told 11:30, no?
Okay, after this.
Next is T2OP3 by Lekshida. Are you ready?
Yes, sir. Are you audible?
Yeah, you are audible.
Yes, sir.
Is it visible now?
Yeah, yes, sir.
Yeah, please.
My topic is automated classification of the black pepper leaf diseases using VGG 19, SNES 50, and the YOLO models. So, myself, Lekshita, research scholar, I am at the research center under Mangalore University. I'm doing my research under the guidance of the Hemalata ma'am, Hemalatayan, Dr. Hemalatayan. So, can you please go to the next slide? So my work is based on pest and disease in a black pepper. Plant diseases reduce the crop growth and agriculture productivity, as we know that. So my here the main topic is I'm focusing on the disease only here in this work. So black pepper plants are affected by the diseases. So many diseases have. I choose only 6 classes. So the manual disease detection may cause wrong identification and the improper pesticides used. So this study uses deep learning to identify or classify the black pepper leaf diseases for early and accurate detection. So my objective of research is to develop a based system for early detection and the classification of the black pepper leaf diseases, and to implement and compare the VGG, SNES 50 and the YOLO models to get the accurate result. So these are materials and methods which I used in this work. In black pepper, we have to collect the data first. So the data is limited here. I collected the data from the Kegel data set. There will be having 6 categories or 6 classes. The images were pre-processed to improve the quality and consistency. So we can't use raw data as it is. So we have to pre-process the data for improving the quality. So after that, I'm going to use that data to develop the deep learning models. So VGG 19 and the SNES 50 and the YOLO for disease classification. So and also I'm used transfer learning and hyperparameter tuning applied to improve the model accuracy. So okay, well, come to the result and discussion. So I got the highest accuracy rate while using the YOLO model. So then use VGG 19, then SNES 50. So comparatively, we have I built 3 models. In that, YOLO got 100% accuracy. So here, the models were tested using the different evaluation methods, using having the accuracy, precision, like that only. So go to the next slide. Okay, so this is a result I got this is a result for the VGG 19. This predicted the polio disease, and it will be predicted 100% correct. So here we used performance evaluation by using the accuracy. So that means that overall correctness of the model, and also I used precision. So that indicates how many predicted positive samples are actually correct. So that is also shown. So then accuracy graph also shown. Then what is the loss? So that also shown. So here we have a F1 score also. So that is a numeric mean of the precision and the recall. So then we have a confusion matrix. So this is a result for this one, SNES 50. I got 99.91 accurate result. So these are the result I got. The next one. Okay. So this is a result for the YOLO model. Here, the confusion matrix will have the label used for the evaluation, performance evaluation. So there is a classification mode. So that is to compare the actual class labels and the predicted class labels. So these are the results I got from these models, these three models. Go to next slide. So in silent findings, the classified black pepper leaf diseases using the VGG19, SNF50 and the YOLO models. Here, I also find VGGS also, but there I got lesser accuracy. So I want to compare with the higher accuracy models. So data set contains 6 classes. There I have food roots, leaf blight, bollo disease, slow decline, yellow and healthy leaves also. So YOLO achieved 100% accuracy. So then it will be the next highest value will be VGG19. So that is a 99.57 and the ResNet 50 with a 99.13%. So as all of you seeing that the result shown that deep learning model can accurately support early plant disease detection. So I conclude that the study demonstrates the transfer learning based deep learning models are effective for the black pepper leaf disease classification. Among the models can have a YOLO. We are going to prefer the YOLO for the best performance. There we have the versions also. So proposed approach can help farmer to identify the disease early stage disease also identify and reduce the crop loss also and support the smart agriculture system. In futurely, we can deploy in a mobile application also. So I acknowledge. I would like to thank my guide, Dr. Hemaltha for valuable support and guidance and encouragement provided throughout the study, and also acknowledge Kagil for providing the publicly available data set used in this research. Finally, I extend my thanks for MPPS and Kerala Agriculture University. Thank you.
Thank you very much, Lakshita. Just would like to ask you one thing. When you validated your results, could you get it done in different varieties or a single variety?
No, sir, not a single variety, different variety also. But I'm using the 6 classes. No, so in that, which is the disease that will be provided the answer or the result?
I just asked this question because there is wide variability with respect to the laminar architecture of black pepper. When you train with the single variety, sometimes what happens is that it may not be able to detect what is there in another variety.
Yes, sir. Yes.
Did you experience something like that?
No, sir. So now I'm using if I use the raw data, no, so then I can go to that. I think so. But I'm using now it's predefined data is available in a Kaggle. So I'm using that. After that, I'm going to pre-process that particular data.
Okay, thank you.
Hello, Lakshita. Can you hear me?
Ma'am. Yeah, ma'am.
Can you take the picture of that healthy? Yes. Do you think it is healthy?
Yes, ma'am. Yes. So the image was blurred right now. So because I took up print screen.
I can see one brown spot over there, and it shows intervenal chlorosis also. So if you classify this leaf as healthy, I think the application will get confused, no? If this is your sample photograph,
I think so, ma'am. You are seeing like that only, but it is healthy leaves only. So that is there in a healthy class only. So it's not infected anything. It's clean and clear. But so now I place the picture, no? So that is blur right now.
Where did you collect the photographs for this experiment?
I collected the data set from the Kaggle.
It's not clear. Is it clear? Okay, Lakshita, thank you.
Yeah, thank you.
I think next presentation, that is T2OP4. Mr. Heyram. Heyram.
Yes, sir.
It's audible?
Yes, sir. Audible.
Yeah.
Am I audible?
Yeah. Yeah, it's visible now?
Yes.
Yes.
Shall I start?
Yeah.
Good morning to all delegates. I am Heyram from Tamil Nadu Agriculture University. I am going to present my paper entitled Rice BSP got a deep learning assisted system for yield reduction of black and brown spot in rice. As you all know, rice is one of the staple fundamental food crop which is attacked by major polyfungal diseases like blast and brown spot, among which blast accounts for about 30% yield loss annually and brown spot accounts for about 22.5% yield loss annually. So yield reduction of these two major polyfungal diseases in rice is very much essential. So objective of my research is to design a mobile application for yield reduction of blast and brown spot in rice. Coming to materials and methods, these are the basic steps involved in development of a disease reduction system. First, a survey was conducted all over rice fields of Kerala in order to identify the brown spot and blast infected rice fields. Based on the survey, the early and late stage symptom images of brown spot were acquired from the brown spot infected field, which was located at Kartuvel, Kanjangad of Kasaragod district, and early stage symptoms of blast, which were acquired from blast infected nursery located at Regional Agriculture Research Station Patambi, and late stage symptoms of images of blast, which were acquired from the blast infected field located at Keshavikan Kendra Patambi. After image acquisition, it was grouped into several stages, several scales based on the percentage of infected area. Then once grouping is done, the number of images in each stage were augmented, multiplied using methods like rotating at different angles and flipping horizontally and vertically and adjusting the brightness. After augmentation, the images are pre-processed in order to meet the requirements of the deep learning model what we have used, Sim transformer based deep learning model, 224 pixel values and normalization. Then the images were used for training the model and testing the model. This is the score chart which is used for image grouping based on the severity of blast and brown spot. In case of both the diseases, the images are segregated into 4 scales based on the percentage of infected area. In first scale, it is less than 5% of infected area. In second scale, 5 to 25%. In third scale, 26 to 50. And fourth scale, 51 to 100. And one class, 8th class, I have kept as healthy images. Out of total images acquired, that were 6,000, which were segregated into 3 types of data set based on the purpose of use in deep learning model. Out of total images, 70% of the images which were used for training and 20% for validation purpose and 10% for test data are number of images which are tabulated here. Next slide. Coming to resultant discussion, this is a confusion matrix which were derived from the Sim transformer based deep learning model. This confusion matrix clearly depicts that the developed deep learning model have capability to clearly differentiate and classify the early stage symptoms of brown spot as well as blast with excellent accuracy. In case of blast first stage, it exhibits 96.97%, and in case of brown spot first stage, it exhibits 85.71%. And the model overall exhibited training accuracy of 92% and test accuracy of 90%. Then the developed deep learning model was deployed into a mobile application, converted into a mobile application named Rice BSP GOT. In this is the general working flow of the mobile application. In the mobile application, we have 2 options. That is, you can capture image lively from the deceased field or you can upload from gallery. Once the image gets uploaded, the application will detect whether it is healthy or infected. Once it's confirmed it is healthy, it will recommend a preventive measure. Once it gets infected, it confirms whether it is brown spot or blast. Once disease is confirmed, it next it will predict what severity stage of the particular disease the image belongs to. Then according to that, it will describe symptom of the particular stage and recommend a remedy measure. Next slide. These two videos depicts how the mobile application works in case of detection of first stage symptom of brown spot and blast. First video will shows you how the application works in detecting the first stage symptom of brown spot. Please play first video. Here, once you can upload the image from the data set, the application will detect what it is. Once it confirms infection status and disease, it will predict the state and symptom details of the particular state and remedial measure. Please play next video. This video will depict how mobile application works in case of detection of first stage symptom of blast. Once it confirms the disease and stage, based on that, it will recommend a remedial measure. Please go to next slide. Next step, I have validated the mobile application by using chi-square test by evenly feeding of images in all severe stages of both brown spot and blast disease, among which how much the application correctly detecting the images. So based on the chi-square tester, it is clearly visible that there is no much significant difference between the observed and expected values. So the application exhibiting uniform classification performance over all severe stages of brown spot and blast, and particularly in case of early stages of blast and brown spot. In case of blast first stage, it exhibits 90% accuracy, and brown spot first stage 90% of accuracy. Coming to salient findings of the research, the developed mobile application BSP GOT gives excellent differentiation between healthy and infected as well as all severe stages of brown spot and blast, and it can correctly identify and describe the symptom with the percentage of infected area. Based on that, it will recommend a remedial measure. So I can conclude my presentation by saying that early detection of to major foliar fungal diseases like brown spot and blast of rice is possible with AI-based technique like deep learning. It is helping in detecting the disease at early stage, which is helping preventing further spread of the disease through timely management. Next slide. This tabulation will give how much a farmer can save, how much it saves the economy of the farmer. So if the farmer detects disease at the early stage, he can control the disease with only one time spray of a contact fungicide. If he fails to detect at early stage, he have to give at least minimum 2 sprays of contact and systemic fungicide. So adding all the cost employed, the farmer can save up to 24,670. It is calculated based on 1 hectare of paddy. So up to 25,000 if farmer detect at early stage. Next slide. I duly acknowledge all faculties of following departments and colleges for their consistent help and support and guidance throughout my research, particularly to chairman and advisory committee members. Thank you.
Thank you, Hera. Thank you. One thing I just wanted to know about is that there was some discrepancy regarding the accuracy when it comes to the validation at different stages of the diseases. You know, there was 100% accuracy in the healthy one, and of course, when it comes to the stage 3, there was a little bit of confusion. It came down to around 60% or so. Not this slide, previous one, I suppose.
Next slide. So
Yeah,
actually here,
this one.
in case it varies due to number of images which were so much low in case of severe stages, sir. As it disease progressed to severe stage, the plant got completely damaged and it was not able to occur more number of images, sir. Actually here I have used somewhat less number. In yearly study, you can see in case of training how many number of images I have used and in case of test purpose. So it is due to some
low number of images in case of brown spot severe stages, fourth stage. It is very challenging to collect more number of in that stage, sir. And in case of blast, first and second, there is some confusion in the model, so it gives some less accuracy, I guess.
Another question is that you have shown an economic evaluation. Did you test it, or is it simple theoretical in nature?
It is theoretically based. I just collected to just to show how much economic impact of LED reduction. Sir, it is not any experiment. That's theoretically I have collected based on market cost.
Thank you.
Okay. Yeah. It's not there. Mic.
Just curious to know what would be the output if you are to feed a brown spot fourth stage.
I couldn't be able to get.
My question is, out of 60% of the time it predicts correct. So out of 40% it is wrong. So what would be the output in those 40%? Does it say?
Sir, actually, I suggest if you train with more number of images. In case of all other stages, I have trained the model with 500 number of images, sir. While coming to fourth stage, I have just trained the model with 350 images, sir. Actually, it was much difference in number of images. I suggest if we able to collect more number of images in that stage, model could have performed more better as like all other stages, sir. I purely able to unless like due to low number of images only, model couldn't be able to learn more features from the images, so it can't perform well in that particular stage, sir.
Thank you.
Rajesh, shall I clarify that? See, you asked about 3 and stage 2 and 3, no?
Okay. This is next presentation, T2OP5. Swadhi, are you in line?
Yes, sir.
Yes. Is it visible?
Yes, sir.
Yes, please.
Good morning all. I am Swadhi GS from College of Agriculture, Velaini. The topic of my presentation is eco-engineered piper-derived silver nanoparticles for inducing resistance and suppressing Phytophthora foot rot in black pepper. We all know foot rot is a devastating disease in black pepper caused by the oomycete fungus Phytophthora capsicii, and on a global scale, an annual crop loss of about $4.5 to $7.5 million has been reported. So next slide. Nowadays, green nanotechnology is gaining so much attention because these green synthesized nanoparticles can effectively manage these plant diseases. And the process of their synthesis is easy and simple, and they are eco-friendly also. Next slide. So in the present study, leaf extract of foot-rot-resistant genotype Piper colubrinum and that from susceptible genotype Piper nigrum were used for green synthesis of silver nanoparticles, and their relative efficacy in suppressing the disease in a susceptible pepper cultivar was evaluated. This is the procedure for green synthesis of silver nanoparticle. First, we have to collect leaves, wash it and boil it with distilled water. Then we have to filter it and collect the leaf extract using a water number 1 filter paper. Then add 1 millimolar concentration of silver nitrate solution into the leaf extract in the ratio 9:1. That is 9 part of silver nitrate solution is added to 1 part of leaf extract. Then we have to incubate this mixture in dark for an hour. And after incubation, the solution color changes to a dark brown color. And then we can separate the nanoparticles using centrifugation technique. Next slide. This is the color change observed during the green synthesis of silver nanoparticles. On the left side, you can see the color change observed with synthesis of Piper nigrum silver nanoparticles, and on the right, you can see the color change observed with that of Piper colubrinum silver nanoparticles. Then the nanoparticles are characterized using different characterization methods. This is the result of UV visible spectroscopy. A peak of 440 nanometer was obtained for Piper nigrum silver nanoparticles, and for Piper colloidinum, peak was obtained at for 60 nanometer. This indicates the presence of metallic silver. Next. The sharp peaks obtained with the X-ray diffraction analysis indicate the crystalline nature of the nanoparticles. Next. Using FTIR analysis, we found out the functional group associated with the nanoparticles. Next slide. Using scanning electron microscopy and transmission electron microscopy, we identified the shape and size of the nanoparticles. The average size of the nanoparticles was about 56 nanometer. Next slide. These are the TEM images. Next slide. Next is the in vitro bioassay of green synthesized silver nanoparticles against the pathogen. For this, we have used the poison food technique. The plate A is the control plate, and on the top you can see different concentration of Piper nigrum silver nanoparticle, and on the bottom you can see different concentration of Piper colloidinum silver nanoparticle. We have tried 100 to 1250 ppm. Among this, 550 ppm came out as this treatment. Next slide. Here you can see 550 ppm selected as the best treatment, and another trial of poison food technique was done to compare the effect of same concentration of commercial silver nanoparticles and chitosan nanoparticles and leaf extract on pathogen suppression. Next slide. Here plate L is the chemical control, A is the pathogen control, and here also the green synthesized nanoparticle came out as the best treatment. Next slide. Here you can see maximum mycelial growth inhibition was obtained when green synthesized nanoparticles was used at 750 ppm. Next slide. Same set of treatments are tested in detached leaf assay also. For detached leaf assay, we used leaves of Panyu 1 variety, and this is the observation 3 days after inoculation with the pathogen. Next slide. And this is the observation 5 days after inoculation with the pathogen. In detached leaf assay also, treatment with green synthesized nanoparticles at 750 ppm concentration came out as the best treatment. Next slide. Next slide. So the best treatment from in vitro study is taken for in vivo evaluation. For in vivo evaluation, we have 3-month-old root cuttings of Panyu 1 variety, and the treatments were given prophylactically. And after 1 hour after spraying, we challenge inoculate pathogen on the top leaves of the plant. And this is the observation on the 3rd day after inoculation. Next slide. And this is the observation on the 6th day after inoculation with the pathogen. Here also, next slide. Particle came out as the best treatment with minimum percentage disease index. And from the best treatment from monoculture study is taken for estimation of defense enzymes. We have estimated 3 different defense enzymes on the 2nd, 4th, and 6th day after inoculation. And in peroxidase activity, the maximum peroxidase activity was observed on the 4th day after inoculation in plants treated with Piper colloidinum silver nanoparticles at 750 ppm with pathogen inoculation. Next. Next slide. And coming to the polyphenol oxidase activity, maximum activity was observed on the 6th day after inoculation. Next slide. Maximum activity is observed on plants treated with Piper nigrum silver nanoparticles at 750 ppm. And coming to phenylalanine ammonialase activity, maximum activity was observed on the 6th day after inoculation in plants treated with Piper colloidinum silver nanoparticles with pathogen inoculation. And the maximum activity was next slide. Here you can coming to the silver nanoparticles from the leaf extract of pepper genotype exhibited remarkable anti-oomycete activity, and it helped to reduce the incidence and severity of food rot disease in black pepper when the foliage is sprayed with the nanoparticles at 750 ppm concentration, and the treatment was given prophylactically. And green synthesized silver nanoparticles can also act as elicitors and can trigger plant defense response. This could be an effective and useful antifungal substitute for crop protection system, and multi-location field trials must be conducted to confirm the effectiveness of these nanoparticles. I acknowledge KU for funding and CSIRNS for characterization of nanoparticles. Thank you.
Thank you, Swati. I just have a question. Did you have a check with the chemically synthesized silver nanoparticle in this?
Yes, sir.
How did it behave?
Sir,
I mean chemically synthesized, not the green synthesized.
Yes, sir. Commercially available silver nanoparticles were also used for comparison, sir.
Did it use that commercially available one?
Yes, sir.
What was the concentration used?
Same concentration, sir. 500 and 750 ppm.
But that is not shown in this graph, is it?
Yes, sir. It is there on slide number 15. So plate F and G are treated with commercially available silver nanoparticles.
Yeah, but you didn't test it in against the pathogen under in-vivo condition. No.
Sir.
Okay.
So, sorry. Hello.
Okay, fine. Thank you.
Swati, can you hear me?
Hello. Yes, ma'am.
Okay, excellent. A nice presentation, Swati. I have one suggestion for you. See, take the same slide, slide number 15. You have given the title as poison food technique. See, that is the methodology that we all should be supposed to know. Okay, so you could have given the title as effect of different treatments against mycelial growth of P. capsicum. Understand? So that will be a proper title for this slide. So please use proper title for the slides in your future presentations. So that is the main information that we get from this slide. Okay, congrats, good work.
Okay, ma'am, thank you.
Moving to next presenter, T2OP6, Sudesh.
Yes.
Audible now?
Yes, sir. Am I audible?
Yeah.
Thank you. I request you to present the second PPT I have. Okay,
it's new, no?
Yes.
Yeah. Is it visible?
Yes, sir.
Yes. Yeah, please, sir.
Okay. First of all, I would like to thank you, Saina ma'am, for this wonderful opportunity and all the coordinate of this national conference. Myself, Sudesh KM, a research scholar of Department of Computer Science, Amrita School of Computing, Coimbatore. And the topic is paddy pathologies, an AI-powered mobile application for early stage severity assessment of rice BLB using deep learning with integrated image and lesion. So as an introduction, rice is major crop in the world, as all you know, and the diseases causing the in rice is like there are major diseases affecting yield and its crop quality. So early detection and severity assessment are critical for the effective disease management. Existing approaches mainly focusing on the classification part itself. As we know that there are a number of proposed methods and number of applications are available using the CNN model and other deep learning model. All are concentrating on the image itself. So limited support for the severity estimation. No, currently we are concentrating on the class. Sir, please adjust. Previous slide, please. Okay, fine. So the major objective of our proposed approach is the early detection, severity assessment, real-time field usability. Moving to the next slide. Recent advancement in medical images have demonstrated the benefit of combining the visual features with the text-based descriptions for improved diagnosis. So inspired by these approaches, proposing and extending our pipeline, like incorporating a description-based feature extracted from the diseased leaf. So these features are generated using a classical image processing technique like lesion size, texture, color statistics, and these all are saved as a CSV file. Using these clubbed information like image features along with the description features, we are fusing these features into a single vector and moving to the classification part or detection part. So multimodal approaches combining images and description features improve the diagnosis accuracy and robustness. So textual description derived from the image features like lesion area, color, texture can improve like visual features are by providing interpretability domain relevant information. Fusing of these features can improve the accuracy and as well as the visual plus texture description can enhance the easiness for classification of our existing CNN models. Next slide. So these are the proposed approach, like proposing a PADI pathologist. It's the name of our application. It's an AI-based mobile application, combines the image-based features along with the lesion-based severity estimation. Designed for the field condition, low resource environment, and especially for the offline usage. And contributions are image information plus the lesion tool. And optimized mobile-friendly pipelines, including, first of all, we are just evaluating the presence of leaf. For evaluating this leaf presence using a unit-based leaf segmentation approach. So using this unit-based leaf segmentation, it's very important for every CNN-based mobile application. The major problem of CNN is for any kind of image, there will be a result, like there will be a classification result. So in order to avoid such issues, such error input issues, we are using a unit-based leaf segmentation first. Then the lesion extraction process using HSV filtering. These are the overall pipeline of our proposed approach. As I already mentioned, there are multiple fusing approaches like fusion of images plus fusion of lesion mask features, then description features. Finally, we are classifying using the MobileNet V2 model. And this, yeah, next. These are the descriptions extracted using the description method like lesion-based computer vision tool. Using traditional computer vision methods, we can extract the lesion features like lesion size, texture, homogeneity, H-means, lesion correlation, everything. And all these features are tabulated. And moving to the segmentation approaches, this is a sample of some leaf segmentation. As you can see, the first image, first row showing the original images, then diseased lesion areas, then the finally red marked area, which is the leaf with the lesion. Next. So moving to the evaluation part, we are getting a validation accuracy as almost 99%. Test accuracy is reached to 98%. And overall, for a particular image, it will take almost 200 milliseconds. The confusion metric showing the classification. Next. For interpretability, we are using a GradCam image analysis where the model is concentrated for classification or prediction. As we can see that the model is correctly classified and correctly concentrated where the actual disease or to severity analysis. Next. Skip next. So the key input modes of mobile applications are auto capture mode, manual capture, then gallery upload. Only the leaf which have the segmentation confidence more than 20% is moving to the next stage. Otherwise, it is just popped a message like no leaf detected. Then manual capture, there are same procedure is happening like it is actually tracking for the leaf is detected or not. Then finally, next. If the leaf is detected with a particular confident, it is moving to the segmentation, like capturing with the detailed lesion characteristics. Next. Next. Yeah. Here we can, first of all, it's classifying whether it is BLB or not. If it is BLB, then severity analysis is, severity analysis, the button is enabled for capturing. And advisory and escalation support, we are using an SMS-based support along with the GPS location of the user into nearest agriculture officer. Whether it is any field is detected, disease is detected or not. Next. And the application provide the multilingual support like English, Malayalam, Tamil, Telugu, Kannada, Hindi. And image-based SMS-based escalation system, which contain the disease name, severity stage, farmer locality, and in the nearest agriculture office's mobile number, or enable the rapid response. Next. These are some sample results of leaf segmentation. Next. And this is the overall application overview. First of all, the user need to register using wishing language, like he can select any kind of these above mentioned languages. Finally, home screen is like the final screen. Next. And this is the sample images representing rice leaf detected or not. Once the rice leaf is detected, it is moving to the classification part, whether it is BLB, the severity enabled, severity checking was enabled, and the remedial suggestions are also provided in the regional languages, whether the user selected languages. Finally, an SMS service is enabled to the nearest agriculture officer, along with all the details of user field information, disease information, like which disease is affected. Next.
Sudesh, please conclude fast.
Yes, fine.
We're already crossed the time.
Yes. So we have developed a paddy pathologist, an AI-based mobile application for real-time detection and severity analysis of rice BL. Thank you.
Hello, Sudesh. Okay. I wonder why did you name your application as paddy pathologist, though you concentrate on a single disease? How are you going to justify this?
Actually, in future we are going to upload it with the different kind of diseases like blast, brown spot, etc.
Yeah. You are concentrating on only on BLB.
Yeah, currently I'm concentrated on BLB itself, sir. And in the future, I'm planning to update the application with another plan, like another diseases.
Okay, I wonder why. What is the result being shown when rice blast imagery is uploaded into this system?
Initially, we are classification was done, like whether it is blast, BLB, or any other kind of diseases. Severity detection is only done for the BLB now.
Okay, thank you.
Yes.
Audience? Thank you, Sudesh, for the nice presentation.
Thank you.
Now we'll have a short break for tea, requesting all the delegates to reassemble at 11:50 a.m.
Now we move on to the next session. Requesting the chair and co-chair on the dais. Next presentation is T2 OP7, differential gene expression in Vigna radiata due to Agrobacterium tumefaciens.
Good afternoon all. I'm Aishat Nebina, I'm MSc Botany student from Central University of Kerala, working under the guidance of Dr. Jasmine M. Shah. Today I'm going to present on differential gene expression in Vigna radiata due to Agrobacterium tumefaciens. Vigna radiata, commonly known as mung bean, is an economically and nutritionally important pulse crop. It is rich in carbohydrates, proteins, 50 to 60% per gram of carbohydrates and 20 to 30% of proteins per 100 gram. Also rich in vitamins, minerals, and it has economic importance as atmospheric nitrogen fixation. Cultivated widely across Asia, particularly in Indian subcontinent, Rajasthan being the largest producer, followed by Maharashtra. And these are the following countries, states. Yield improvement in Vigna radiata has remained stagnant over the past 3 decades due to susceptibility to abiotic stresses as well as biotic stresses, also limited genetic variability available for conventional breeding. Genetic transformation or genome editing offers a promising alternative for crop improvement, yet mung bean, like many grain legumes, exhibits strong recalcitrant to transformation. The transformation efficiency of Vigna radiata remained The transformation efficiency of Vigna radiata remained 4.2% for cotyledonary node and 1.49% for shoot tip. So also the molecular basis of host pathogen interaction in Vigna radiata remained poorly understood. So in this study, a comprehensive analysis, transcriptomic analysis was conducted to elucidate the response of Vigna radiata to Agrobacterium tumefaciens and also to identify and characterize the differential gene expression due to its response. So these are the main objectives of my research. This is the workflow of wet lab experiment conducted to obtain the transcriptome data, and this is the workflow of transcriptome data analysis. I have used ShinyGo, 0.77, NCBI, Ensemble, etc. Coming to the first objective, a total of 814 differentially expressed genes were obtained, of which 457 genes are upregulated and 357 genes are downregulated. Among these 357 genes, downregulated genes, 2 are from mitochondria and 1 is from chloroplast. Then I have done the chromosome-wise distribution of differentially expressed genes. Here, the most number of genes upregulated are from chromosome number 5, and the most number of downregulated genes are also from chromosome number 5. This is the chromosome-wise distribution of DEGs. As we know that this Vigna radiata is not much studied, only 93 genes are upregulated. Upregulated genes are plotted in the graph, and 76 genes are plotted in downregulated genes. Then coming to the top 10 upregulated genes, ethylene responsive transcription factor is the most upregulated gene, followed by extensing 2 like isoform, then putative nuclease, and the others. Then I have performed the functional analysis of top 3 upregulated genes. These top 3 upregulated genes are not yet studied in Vigna radiata, but there are studies cited in different other species. So this ethylene responsive transcription factor ERF017 is a large ERF family protein which is comes under the largest subfamilies of AP2 ERF transcription factor, and it has a binding single AP2 domain. It binds to the GCC box and CRT elements in the promoters of ethylene induced pathogenesis related genes. Coming to the distribution of ERF in legumes, Medicago sativa has cited 159 ERFs and Vigna radiata cited only 81 ERF. These Vigna radiata ERFs are involved in developmental and environmental stress responses. And these are the studies cited in different other species, Cajanus cajan, Cucurbita moschata, and Larix oleogenesis. Then extensing 2 is a hydroxyproline rich cell wall glycoproteins involved in various plant physiological process, mainly plant defense. This overexpression of extensin 2 helps in plant defense. This is a study case done in Arabidopsis thaliana with the pathogen Pseudomonas syringae pathover tomato DC3000. So, in transgenic Arabidopsis, the symptom development was not much prominent as in wild. This is mainly due to this overexpression of this gene. So, this defense was mainly not by activating the major defense signaling pathways like jasmonic and salicylic acid. Then coming to putative nuclease HABI1 gene, this is a domesticated transposase derived gene that encodes putative nuclease enzyme. So this enzyme has a conserved DDA catalytic domain which binds to a divalent metal ion and catalyzes DNA cleavage. And these are the studies cited on putative nuclease HABI1. In Arabidopsis thaliana, the overexpression of this gene has shown tolerance to cadmium responses and also regulate different molecular pathways associated with cadmium element. Then I have done the most gene ontology of the gene ontology of this genes. The most affected biological process is cellulose biosynthetic process, and the more number of genes were involved in biological regulation, and the most affected cellular component was cell wall and external encapsulating structure. And the most affected molecular function was aspartyl esterase activity. Among these, we can interpret that the most differential gene expression was occurring in cell wall related genes. So that may be one of the reason for recalcitrant to transformation. Then I have done comparative analysis of biological process with Arabidopsis thaliana. Here the most affected biological process was defense response and response to stress. In our study, the most affected biological process was cellulose biosynthesis. This is the major difference I have noted. Then KEGG pathways of upregulated genes have analyzed. The stilbenoid, diaryl heptanoid and gingerol biosynthesis was the most upregulated enriched pathway, and these are the genes involved in each pathways. Then I have identified plant factors involved in agrobacterium-mediated transformation. About 49 host genes were identified to be involved in agrobacterium-mediated transformation in Arabidopsis thaliana, and I have identified homologous for this 49 genes in Vigna radiata. From these 49 genes, only 2 genes have been significantly differentially expressed in Vigna radiata. The remaining 47 genes didn't show any significant differential expression in Vigna radiata. Those 2 genes are MPK3, which is upregulated in Vigna, and HD2B, histone deacetylase, which is downregulated in Vigna as well as in Arabidopsis. Even though these 2 are differentially expressed, there is no prominent fold change observed in Vigna radiata. So by concluding that this can be the recalcitrant or the hindrance for transformation to Vigna radiata can be due to these reasons. The weaker differential gene expression may contribute to the recalcitrance. And this, I have already explained the salient features, findings, and these are my conclusions, that this studies will provide valuable insight to the host pathogen interaction during transformation, and this can further used for genome editing and gene transformation. Thank
you. Thank you, Aishat Nabina. Just a question. What was the post-infective duration at which you measured the gene expression?
4th day.
4th day. 4th day. Did you find any symptoms development?
Sorry, sir?
Did you find any symptom development as far as the infection process is concerned?
Sir, the wet lab experiment was previously done by my senior. I have done the bioinformatics works.
That means in all data were generated previously, even the expression analysis. Did you do any wet lab study for
No,
this?
no. I have done the bioinformatics work.
So the information gathered by your senior was just analyzed by you, okay?
Yes.
Okay. The big question is that, when the analysis is done on the 4th day after inoculation, we really do not know whether there is an infection or not, because you say that the percentage of infection by Agrobacterium tumefaciens is very low or too low in nature. So you will not be able to differentiate between the plants which are really infected and not infected. And therefore, there has to be some other mechanism with which you will be able to differentiate the plants which are infected. And otherwise, if it is a single plant sampling process, there could be too many number of errors. Okay. Thank you.
Any questions from the audience?
I think moving to the next presentation, that is
Thank you, Nabina.
T2OP9 by Aparna KS. Aparna, are you ready? Aparna RS, sorry.
Hello, am I audible?
Yeah. Sound korvane.
Okay, sir.
Please. It's T2OP9 online. No, OP8 will do the last. After that, is 2 physical presentations are there. That is after lead lecture of Dr. Selvaraj. Aparna, are you ready?
Yes, sir.
Yes, please.
Respected Chair and Co-Chair, good afternoon, one and all present. I am Aparna, PhD scholar at Department of Plant Pathology, College of Agriculture, Vellanikara. The topic of my presentation is biosynthesis and antimicrobial potential of copper oxide nanoparticles using Pseudomonas fluorescens. Since the conventional methods of plant disease management often results in development of pathogen resistance, residual effect, and other environmental concerns, we need to go for alternative strategies. So nanotechnology is one such field, but when it comes to synthesis, physical and chemical synthesis often leads to hazardous byproducts. So we should focus on biosynthesis of nanoparticles. And in that context, the objective of the study is biosynthesis of copper oxide nanoparticles from Pseudomonas fluorescens and its physicochemical characterization and evaluation of these nanoparticles for its antimicrobial activity against major plant pathogens. Moving on to the materials and methods as part of the study, cultural, morphological and molecular characterization of reference strain of Kerala Agriculture University was done. Next slide. So once Pseudomonas fluorescens was obtained, its cell-free culture filtrate was taken and mixed with 10 ml pentahydrate salt solution at equal parts. The previous slide itself. And once the pH was adjusted, it was then given a heating at 60 degrees Celsius for 20 minutes with intermediate stirring. And we can see the color change from blue to greenish brown. And it was given with 3 wash with deionized water and further dried to obtain the copper oxide nanoparticles. Next slide. Once the synthesis was over, we need to confirm the nanoparticle nature, for which different physicochemical characterization methods need to be done. So as part of that, dynamic light scattering was done, in which particle size was analyzed with an average size which was obtained as 431.7 nanometer. This may be due to the hydrodynamic diameter. A polydispersity index of 0.122 represents a monodispersed nanoparticles. In case of zeta potential, a value of minus 22.67 millivolt was obtained, which suggests moderate stability. For understanding the functional groups associated with the capping for the stabilization of the nanoparticles, Fourier transform infrared analysis was done, that is FTIR. So different peaks were obtained, in which 400 to 700 wavelength was obtained. Peak at 400 to 700 was obtained, which represents characteristic copper oxide stretching vibration. And for understanding whether the nanoparticle is having a crystalline or amorphous nature, X-ray diffraction studies was conducted, and peaks at 35.6 degree, 38.56 degree, and 52.55 degree corresponds to distinct planes suggesting monoclinic crystalline copper oxide nanoparticle. Next slide. For understanding the shape and size of the nanoparticle synthesized, for field emission SEM was conducted. So it revealed ellipsoidal nanoparticles with size range from 28 nanometer to 99 nanometer. For understanding the elemental composition of the nanoparticle obtained, energy dispersive spectra analysis was done. So copper with 76.64% and oxygen at 17.63% was obtained. So once the nanoparticles were synthesized, it was evaluated for its antimicrobial activity against Phytophthora capsicum and Pythium aphanidermatum using poison food technique, and against Xanthomonas oryzae, Pathovar oryzae for using agar-well diffusion assay. So as we can see, in the case of Phytophthora capsicum, next slide, 100% inhibition was obtained in the case of all the 3 concentrations, that is 500, 750 and 1000 ppm. In case of Pythium aphanidermatum, maximum inhibition was obtained in the case of 1000 ppm with 86.67%, and maximum inhibition zone was recorded in case of Xanthomonas oryzae, Pathovar oryzae with 1.37 centimeter at 1000 ppm. So salient finding is that biological synthesis could be done using Pseudomonas fluorescens to obtain copper oxide nanoparticles, and the physicochemical characterization revealed the synthesis of monoclinic crystalline copper oxide nanoparticles. And through the antimicrobial activity evaluation, we could see that even at 500 ppm concentration, significant inhibition of the major pathogens could be achieved. So as a conclusion, these nanoparticles can be effectively used as smart, sustainable and affordable management tool for plant disease management only after doing some field trials. Thank you. I am grateful for my major advisor as well as the advisory committee members and also to Kerala Agriculture University.
Thank you, Aparna. Just a question like, what is the concentration, effective concentration of copper oxide, not nanoparticle, in controlling the, or in vitro control of the pathogen?
Copper oxide nanoparticle answer, sir?
No, not the copper nanoparticles, which you did. But I was curious on getting the information.
I think it is 0.2%.
Copper oxide.
I'm not sure, sir. I need to do that.
You should have done a comparative check with the copper oxide at this particular concentration against the copper oxide nanoparticles. Okay. Another thing,
yes.
Your cell-free culture extract or fluid, whatever it is, CF. That means, cell-free?
Culture filtrate.
Culture filtrate, okay. You have grown the bacteria in some medium, probably King's Bee broth.
Exactly.
King's Bee
Yes,
broth.
sir.
Okay. Why didn't you do a broth alone control for this? I can argue that the biological substances present in the King's Bee medium would have the same character. You say that it
yes,
is just because of
actually, I have tried once, but I didn't get any inhibition, but I have to repeat it again.
Yeah, that is what, unless and otherwise you do a similar experiment with the media alone control and compare the effect. You just cannot say that, it's because of the culture filtrate of the Pseudomonas fluorescens that has provided the additional advantage for the copper oxide nanoparticles. Okay.
Okay, sir.
Thank you.
Okay, sir. Thank you.
Next, we have the lead talk 2 by a distinguished scientist from ICR NRCB, Dr. R. Shelvarajan. With over 30 years of distinguished service in banana research and plant pathology, he has been instrumental in the revival of near-extinct GI-tagged Virupakshi variety of banana. He is instrumental in the development of AI-powered decision support system for banana disease management. And among several other contributions, he has got more than 99 research papers, 2,441 citations with H index of 23 and an I-10 index of 46. He has authored 7 books, contributed 62 book chapters. Several outstanding scientific achievements, including prestigious honors like DST Lockheed Martin India Innovation Growth Program Award, the ICR Hari Om Ashram Trust Award, GS Sannidhi Award, and several other awards. He is a secretary of the Plant Virology Society, vice president of the Indian Virological Society, as well as editor of Springer Journal Virus Disease. He is a fellow of National Academy of Agriculture Sciences. 6 other professional societies also he is a member, and like IAHS, IPS, IVS, CHAI, NABS, and AIPUB. And I cordially welcome Dr. Shelvarajan for the lead talk on artificial intelligence in plant disease detection and management. Welcome you, sir.
Sir?
Second take. Time allotted. Time allotment for me. Okay, let me
20 minutes, sir.
go ahead. Okay. Thank you for introducing me to the delegates who is attending the seminar. And this is, I'm going to talk on artificial intelligence in plant disease detection and management. And all you know that this artificial intelligence being widely adopted in the biological system, even in the medical system, all the radiologist job is being done by this artificial intelligence, and including any scan if they take, they get the result through this the machine learning method which they adopt, and finally, of course, it has to be validated. Mm. It has to be validated by the medical doctor and to give the before giving the report. So here I am going to talk about how it is being adopted in the diseases, especially plant pathogens. Some echo is happening, no? Is it okay, my voice? Hello?
Yes, sir, it's audible.
Audible, no echo, no? Okay. Okay, coming to this, as I said, that artificial intelligence and IoT and the recent developments in the precision farming and the decision support system, which has to be adapted, and for the thing which a lot of developments is happening using this system. And nowadays you might be knowing that even this, ChatGPT and many Gemini, Google Gemini and all are there, and we are utilizing that artificial intelligence, and this is what now we have to go along with the developments happening in the engineering side and this computer science side to acquaint for our plant pathologist, especially for taking decision by the farming community and farmers. So this is very important. So I would like to give a brief about the impact of the diseases, the importance. I think many would have talked about. And you know that, now for by 2050, we have to still produce more and more food actually. So what we produce now we have to increase by 56%. And 56%, if we increase the production only, we can feed a 10 billion population, which is expected by 2050. And this, that FAO estimates 20 to 40% loss mainly due to the pest and diseases. And this loss, if we are reducing, that itself is one of the way of doing. So for this, why this echo is coming for me? Auditorium, okay. Okay, so more than 8,000 species of fungi, oomycetes, and more than 2,500 species of virus species, bacterial diseases, leading to $220 billion loss. It is happening, and if you include invasive insect cost, it is $70 billion is happening. Next. So if you look at the diversity of the plant pathogens, it is keep expanding, and all the pathologists gathered in the seminar might be aware of the diversity in the fungal pathogens, virosphere. Out of 16,000 viruses so far known, 2,500 are plant viruses. And plant viruses are major cause, and for clean plant program, getting free from viruses are very important. And again, bacterial disease still a major constraint in the both field crops as well as horticulture crops. Next. Then here I have given you that the important top 10 viruses which is affecting globally, top 10 bacteria, top 10 fungal pathogens, and top 10 oomycetes, which I have given you. And as all plant pathologists might be aware of these viruses, especially cucumber mosaic virus, I would like to say that more than 200 plant species are being affected with cucumber mosaic virus, including banana. And again, plum pox virus is a major virus. It's a perennial tree, and that's again a major issue. Potato virus Y and X are again a major issue in the potato. And tomato spot wilt virus also a major issue. Then again, tomato yellow leaf New Delhi virus, like that, that's also globally an important viruses. Coming to the bacteria, that Xanthomonas viraceae, PV viraceae is one of the major disease, and Ralstonia, Yersinia, Pectobacterium, these all agglutinin tumefaciens, Pseudomonas syringae, and Ralstonia. These 6-7 species, genus, really affecting globally many crops. Fungi, Magnum viraceae, that is the major disease, that is blast disease. And in horticulture crops means, always fungal, bacterial, viral disease are very major, since they are all propagated through, clonal propagation. So these diseases persist regularly. Oomycetes, very well, it's a very major threat to the many of the crops. Will it be, grapes or many crops, capsicum, and many crops. This is a major, including albugo candida. That's also a major pathogen. Next. And I know that, see, this importance of plant health in agriculture, all you know that, so I will skip this slide. And, plant pathologist role in food security is very important by timely giving the advice and giving a good, better management solutions to the farming community. Next. So the temperature dynamics which I have given research trends, and a lot of papers are now coming on this aspect of detection. Convolutional foods, deep learning, and different methods are being used, sensor-based detection. So now the number of research papers are more coming on the deep learning or machine learning, or use of new technologies are being applied. Next. And the traditional methods, that manual inspection, laboratory testing, field surveys, and testing healthy that in chemical. So different methods of the diagnostics is going on, and this field level diagnostics is very important. All you know about in Florida, more than millions of citrus trees were cut down because of the citrus trichisa virus and other citrus greening and all. So that way they manage the crop, such a good quarantine and regular surveillance and monitoring. Similarly, in Australia also they do regular surveillance and monitoring and strict quarantine measures and timely diagnostics. All these things they make. But countries like India, it's very difficult because, it's a vast country and highly populated, and the movement of planting material, disease infected material cannot be avoided, can be avoided, but it's with strict guidelines can be done. Next. So artificial intelligence, all you know that the method is always, some farmers come with a sample and we identify and we give them idea how to control. Like we will be, like doctors prescribing, we prescribe what all the fungicides has to be applied, how to manage for each and every disease. Our pathologists are having knowledge to give idea to control the disease. Now, to make it simpler, artificial intelligence, the intelligence is being adopted. So, artificial intelligence means it's nothing but simulating the human intelligence in machines that are programmed to think, learn, perform tasks typically requiring human cognitive functions. So these tasks include problem solving, decision making, language understanding, visual perception, and more. AI systems are designed to mimic and replicate the human abilities by processing vast amounts of data, recognizing patterns, and adapting new information. But this can always human intervention is very much needed. But this you can save the time actually. So this type of taking a lot of data, like sometimes in chess games they used to play in the computer, and sometimes the human always win against the computer. So in computer they will all keep all the probabilities of winning, losing, movement of the chess coins. They make chess, they make, but finally the human brain is better than what we bring. So that way, even if we use artificial intelligence, finally you have to validate. So the pathologist role always will be there. So This method of automative repetitive tasks and enhancing the decision support and improve the user experiences. And all you know that when we take a mobile and Google Lens, when we use, take a picture of a one plant, it gives the options. These are all the possible plants names, and we immediately identify. So that is nothing but machine learning. That is because of as many as pictures you upload and give the name it, classify it. Using that, it will be easily they will identify. So Google does this job. So if you put my picture, they will say it is Selvarajan. So if my pictures regularly it is in the Google, and Google will take my picture, and if someone is labeled as a Selvarajan, then immediately it will say this is Selvarajan like. So this is what machine learning. Next. And this AI technologies in agriculture domain, it is being adopted many purposes, and here we concentrate on disease and pest management. Next. And global trends in crop disease distribution, artificial intelligence, just last 24 years, you can see different countries and how it's being adopted, this deep learning, machine learning, and CNN, convolutional neural network, image processing, classification, transfer learning, feature extraction, agriculture, means in segmentation, all these things, it's being adopted. Quite a good number of research papers have come, and it is being adopted. And still a long way to go, but we need to have skills, more skills in learning this. Of course, without the help of the computer specialists, engineers, we biologists alone cannot do this job. We have to do joiningly with them. Next. And machine learning approaches to disease detection, which you know that machine learning, in all this machine learning, all this AI-based detection of the disease and all, we give the answer to the machine already, like banana bunchit of disease. We will give this is the answer. Now I have to feed lot of questions that leads to the answer of the same answer. So in this machine learning, normally the answer is given already by us, and we will be feeding lot of things to make that the machine will say this is Panchitop, like that. So I am saying image processing based disease. For this they use support vector machines, SVM, and also random forest. Random forest is an ensemble method using multiple lesson trees to improve the accuracy and reduce the overfitting, identifying the multiple plant diseases from various leaf textures and colors. So this is a different methods of identifying. Next. And again, this method is decision tree method. The decision tree method also, it classifies the diseases on leaf color, texture, patterns. Then KNN, K nearest neighbors, that is classifies data based on the majority of the vote of its K nearest neighbors, grouping similar plant disease images of classification. So this is also another method. Next. And you can see a lot of diseases already, papers have come. They used the UAV, cameras and photos, different methods they have taken, and in the left side, many crops we have given, many diseases for those crops, using the study scale, either leaves. If any leaves or diseases, it will be like a fruit or tree, and they use multispectral camera or UAV or ordinary mobile. Different methods can be adapted to acquisition of the data. Next. And similarly, for other crops like for horticulture crops also it's given different diseases, guava. For all these diseases already it is being adapted. Next. Yeah, this again the list goes on. Next. On the deep learning approaches in plant disease, that's what CNN I was talking about. CNN is one which is now widely adopted. CNNs are specialized for image analysis by extracting features through convolution layers, identifying the plant disease through leaf images by detecting patterns like spots and discoloration. And you might be knowing about Plant Village database where 50,000, 50,000 and more captured images are there, and that's all classified. So most of the students, they download that and they study in the practical point of view. And transfer learning methods are, it's a deep learning method only. transferring utilizes the pre-trained models. So already if a model is existing, that could be directly used for similar things. It is a, over or it's like copying the same method of the methodology for some other purposes. It means adapting pre-trained models. Same similar models can be directly adapted, and we have to feed your images, and it will do resizing and different different angle it will take and different shades it will take. Then it will lead into the convolution layers, and maximum pooling it will happen, and different modules it will pass through. Finally, it will come to the outcome. Next. So deep learning and object detection architecture and plant disease detection is given here. Different architectures are given, like CNN, BGG, and ResNet 9, and plant disease net, FASTER or CNN. So different architectures are there, and the data set, I said plant village, and some data sets are there more than 50,000. This one is there, and a lot of data sets are openly available, and you yourself can make. If you have got 1 lakh images, you can accurately, 99% accurately, you can detect this, and precision also will be very high. And the category which I was telling, that so a lot of papers are now coming, and in fact we have to have in banana also a lot of work has been done. Next. So deep learning architecture in plant disease detection, again the what I told, only same thing which I was told. Next. And in case of data collection for AI models, you can either use RGB images or multispectral drone imagery or anything, even mobile also you can regularly use it. And this is one which we have taken in our farm using multispectral drone, 120 meter above the ground level we have taken. And this type of data, it will be useful for the identifying the wilt disease. So the wilt disease becomes, the leaf will become epinasty and yellowing. So it is easy to identify using NDVA method and all. Next. And again, this is a AI model which is being adapted for plant disease detection, especially here what you are seeing is a detached leaf method. So this plant village data set, all are they have taken detaching the leaf, they have taken the pictures in the laboratory level, but not in the field. So there are many research papers have come. They say it is not the detached leaf. If you are taking the images in the field itself, that gives better accuracy than the one which we are doing like this. Next. And data pre-processing techniques for AI models in the plant disease. Of course, a lot of pre-processing things has to be made, like what I was telling. If you have taken single photo, that can be made into hundreds by rotating, flipping, scaling, cropping, increasing the brightness and all. Because this is the image, it's like 0 and 1 only. So what these pixels values will be taken 0, 1 only. So this, for a machine, it's entirely different. For our eye, it is different. For machine, it is different. So it's all the pixels and how it is going to do actually. Next. On training AI models in plant disease detection, and this is what I already told. Next. Different tools are there, like TensorFlow, ByTouch for model development, the cloud computing services already. Amazon, AWS, Google Cloud, Microsoft Azure. This all can be utilized, and open source of data set is there. Otherwise, you yourself can develop a data set. That's data set, and classification is the major role of plant pathologist. So take photograph and classify them and keep it. That is the one which very much needed to start this AI-based diagnostics. Next. And here we have shown, deficiencies are not diseases. It is actually we induced nitrogen deficiency in a field, our research field, and we could took the data. You have seen the RGB, but if I show the NDVA also, you can exactly say this is nitrogen deficiency. The top one is, iron deficiency. These things also to really drone-based diagnostics can be done. Next. Then this is decision support system for leaf spot disease management. At NRC Banana, what we are doing is around 9 states we have installed this FUSL machine, This takes 22 weather parameters, and our pathologists are in 9 locations. Weekly they take data. Weekly twice they go and take data on least for disease, age, all the parameters they take, and everything it comes to our server located in Tiruchirappalli. So all this data what we receive and we do using random forest, we use and we analyze. Next. And while analyzing, we come to know that age of the crop and relative humidity, these two plays a major role. And keeping what is happening 10-15 days before this, the RH can be utilized for decision support system, whether a farmer has to go for a spray or not. So well in advance, one can give this one. So already we have got the 2 years data across the country in 9 states and with 22 places we have made this. Next. And another is we have developed a patented gadget along with the SSN College of Engineering located in Chennai. We developed this device which always can detect the Bunchita disease and Cicatricoli spot. Besides, it gives you all the weather parameters also. And we have fixed a camera also which can cover more than 10-20 plants. So especially for leaf spot, one can assess the intensity and other things using this gadget. And we got patent granted just a year back. And this device we are improvising so that this can be utilized in the field level. Next. And based, deep neural network for plant pathogen detection we have done for Bunchita disease and cucumber mosaic virus disease. And the model accuracy, when we did know, we got Inception model, ResNet model, BGG, and out of these three, this Inception version 3 model gave a 96.3% accuracy. And if we add more number of images, still it will be giving a better thing. Next. And Goma, I think you might be knowing about Michael Gomez Selvaraj. He is from CIAT. CIAT is located in Colombia, one of the CGI institute. He has made a very wonderful job of taking different diseases, targeting, leaf, fruit, wilt, and different diseases. And he made the image collection, then he made the labeling. Labeling job is done by the pathologist, followed by data splitting and, using different models he has tried. More than, I think, 6 models he tried and 18 different classes he tried. Next. And he used 70% for, training, 20% for validation and testing for 10%, whatever he has taken. So similarly, he has made for surveillance for the wilt disease also, using human in loop artificial intelligence and the YOLO foundation models. And this is all drone disease, how to detect the Fusarium wilt, Xanthomonas wilt, and how to differentiate from the healthy. So this is also one of the work which CIAT people have done. Next. And deploying, testing the ground level models also, sometimes this wilt disease also, we get a typical symptoms in the ground level, like split of pseudostem and many disease things are there. So too many app which was developed by him, now it's widely adopted everywhere for banana. So in India also we were utilizing and it's doing very well. And those plant pathologists can download this too many app and see when you take a picture immediately it will say it knows which disease. It will say 95%, 96%. Sometimes this, Xanthomonas wilt is not there in India. So sometimes it will show as a Xanthomonas wilt. But you should not say that because this has detected, so I can report like that. You cannot make. You have to laboratory level validate. You have to isolate. Then only you can know. So some problems are there in this app, but otherwise this is doing very well. It can able to detect many of the diseases. Next. And leaf spot disease through CNN, how the model is being given here, how that leaf spot disease can be taken and how we have to detect and do it. Actually, leaf spot disease point of view, we have to bother about the intensity of the severity of the disease to take the decision, not identification. Everyone can identify cicatrically spot, but whether we have to go for spray or not, this depends upon the intensity of the disease, not identification. Everyone can do it, but severity. That's what our gadget, what we developed, patented one. It will give you the severity also. Next. So these are all the different models which we have used, and you can see, I think not ours, this is a recent paper came in 2023, wherein banana fruit scoring beetle, cicatoca, salt rot, pseudostem weevil, yellow cicatoca, then banana aphids. Yellow cicatoca and black cicatoca differentiating itself is very difficult. And so these are all the accurately, if we want to make, we have to use quite a good number of images specific to that. Next. And this is for again banana fruit disease detection networks are given here. This also because fruit detection also very important, especially export point of view. Next. So this is feature inbuilt, how it is being by drone methods, how unmanned aerial vehicle can be utilized for remote sensing and assess. Now even if you want to assess the area under the tropical rainfall in India, we are now tying up with ISRO and can we use satellite-based imaging to know the area in Gujarat, Uttar Pradesh, Bihar and Madhya Pradesh, where TR4 is increasingly spreading. Next. So this is again UAV based that Michael Gomez Silva's work, how they are doing surveillance. And you can see July and August, the differences, how the disease progressing and all one can know. So they take a big area for training, they take a big area for testing, then validation, all these things they make. Next. And similarly for onion yellow, onion downy mildew also similar work has been done. Next. And again this also for onion downy mildew, how a motorized system can be kept in the field and how a disease can be detected. Next. And this is tomato leaf detection using CNN. Next. This is for cotton plant disease detection using deep learning method. Next. And smart farming framework based on the big data IoT deep learning model for disease detection. Here, how big data is being utilized, it's given here. Next. Then rice stock. This is also for detecting the rice diseases, for especially rice blast, how it can be detected. And I think this is in Japan, I think so. This is being adopted and how to detect the disease. And here also, AgriTalk places non-image sensor also. So here they have combined not only images but also weather parameters to give the alert services to the farming community. Next. Yeah. This is also a cloud-based application to detect. Next. And this is algorithm for grapevine orchards. So in grapevine also, it's a perennial system, and how to detect. Again, UAV-based method is adopted here. Next. And powdery mildew, strawberry. This is also very important. You can see the mobile, the vehicle which can pass through the crop and can say where. So these are all actually for the differential rate of spray. So wherever the disease is there, you can spray. Wherever disease is not there, you can reduce the spray. So once you get this data and you can fit into the UAV model, and it will spray wherever the disease is there. If it is not there, it will not spray. So first it will do the mapping. The second one, spray drone will go, and it exactly will spray wherever it is required. So for leaf spot disease, it could be done. Next. And we have also utilized this drone technology for how to spray micronutrient deficiency and also cicatoccal leaf spot disease. We have worked out. And next, the final challenges are there still. A lot of challenges are there. I think, like in computer science, they have made many artificial intelligence, means big data analytics, like that. Many sub in degree level they are making. Similarly, in agriculture also, probably we had to introduce some courses to get more knowledge in this line of work. And the challenges are data availability, because we have to have a lot of data and that should be classified. And improving AI interpretability to build the farmer trust. So farmers also should believe. Then interdisciplinary collaboration should be made. Agronomists, data scientists, policy, all has to join together. And AI-powered solutions that are practical and accessible to all farming communities. The future direction, multimodal AI models, integrating satellite imagery, field sensor, genome data, and prioritize cost-effective, user-friendly solutions tailored for smallholder farmers. They cannot afford costly things. And integration of AI with the genomic tools, climate resilient crop breeding, enabling the development of disease-resistant varieties. So a lot of things which I think in future we have to do for AI-driven disease management strategies, and very importantly, some skill development has to happen. In the wherein we had to bring the engineers as a resource person to give training to the our plant pathologists, especially diagnostic specialists, and again quarantine people and many agriculture officers, horticulture officers who are all in the field level. They work. They should be trained using this type of new gadgets to diagnostics. Thank you. Next. Thank you so much. I can take a few questions if you have. Plus,
thank you, Dr. Silva.
yes, any questions, please?
Before we go into the question answer session, I would like to just thank you for the informative talk. I'm glad that your institution is developing AI-based tools in detection of banana diseases. As you suggested, the severity of the disease is more important than the detection process, we believe. And another thing I would like to suggest is that to have networking with the extension people and the plant diagnosticians, and even we can bring in the suppliers of fungicides and pesticides together in a network such that the farmer gets a photograph, it goes to the diagnostician, it's detected, and recommendation is given back to the farmer. The farmer now is getting connected with the most appropriate pesticide or insecticide dealer, so that the supply chain is ensured at time. This is also most important. We are developing tools, but whether it is being used effectively or it goes to the field is to be checked and then evaluated well. Another thing I would like to suggest, especially with banana, is that of the banana pseudostem weevil, which of course is a hidden insect. We need to develop some technique or technology with which deep spectral imagery or even sound-based system may have some utility in detecting those plants which are infected. Initial infection detection is very, very important as far as pseudostem weevil is concerned. Okay, I hope we scientists would work together and then we'll be able to find out a solution for that. Thank you very much on behalf of Kerala Agriculture University and the organizers of the seminar. I thank you very much, and we may take a few questions if there are any from the audience. Thank you very much.
Thank you, Chairman. Thank you, Chairman. As you said, yes, really, we are working for this detection of Vadiporus longicolis stem weevil based on the sound. This we have proposed a project to the DBT now. We are hopeful of getting that project based on the sound inside the movement of larva and even adult. So that keeping that we are trying now with the Assistant College of Engineering, Department of Computer Science, and ourselves. We have proposed a project already for stem weevil. And coming to another point is that, as I said, that severity is more important, especially least for diseases, unless otherwise a decision cannot be taken. So in this point of view, we have to depend on the weather parameter also. So existence of weather parameter and the disease incidence and the age of the crop, especially in banana, all put together a decision support system could be given, not mere identification of the least spot disease. That's one thing. And another thing is that, as you said, really we need new curriculum, especially in agricultural universities, which we are lagging behind, for this type of use of artificial intelligence in the pest and disease management or crop protection. And this skill has to be taken to the field level extension workers. Unless otherwise, what we develop, it will not reach the farming community. As I said, radiologists always they depend on the AI only. So once they make a scan, and immediately it will give the result. So again, after getting that result, he has to sign only. Otherwise, they have worldwide they have collected the data. Worldwide, they have collected a lot of data, especially the scanning, whether it's a lung or any organ, internal organ, being scanned using the radio. This one that is being adopted very big level. But we do not have in agriculture that much intensive adoption is not there. That can happen, I think, in coming days, for which we need a lot of skill development, and some agriculture engineers has to turn to this type of work. They have to come. Otherwise, it's really now communication gap is there. Our agriculture scientists and engineers, they cannot understand what we say, and they cannot understand what we say. So this, either we should get some knowledge in that, or they should get knowledge on the biology. This is there, a big gap is happening, and sure, I think it will be soon, it will be rectified with the new introduction of courses in the agricultural universities as well as engineering colleges. Yeah.
Any questions from the audience? Once again, we thank you from part of Kerala Agricultural University. Thank you very much.
Thank you for the organizers giving the opportunity. Thank you, Chairman. Thank you, Co-Chairman, and all the rapporteurs and organizers. Thank you so much.
We will be continuing our oral presentation. The next one is T2OP12, G. Durga.
To the pathologically as well as the entomologically. And the damage caused by this fruit piercing moth is estimated to be more than 40% of the production, especially in case of commercial pomegranate as well as the orange ecosystem. And it was Bhumananavar and Viraktamat in the year 2001 where they have recorded 29 species of moths on Gaba as well as pomegranate in Karnataka. And since then, there was no studies made for more than 15 years. And it was my guide and my team where we worked on this fruit-sucking moths exclusively, and we could record more than 50 species of fruit-sucking moths. And this was the first ever studies made in India where we could record more than 50 species. And it was a very challenging task for us since it was nocturnal, where we were there in the field from evening 6:30 to night 10 p.m. So we all generally know that the fruit baits or rotten banana is attracted by butterfly. With the same technology, keeping in mind, we have also designed a trap where we evaluated the different indigenous traps using the baits for attracting and managing this particular fruit sucking moths. So here we have made 7 fabricated traps. The trap 1, where we have used the starfruit puree as bait. In the trap 2, where we have kept an open pan with banana puree as bait. In case of T3, we have kept open pan with guava puree as a bait. In case of T4, we have kept open pan with ripened banana as a bait. So in case of T5, where we have half-cut ripened banana as a bait. So we felt like this moth is feeding on this particular baits, but it is escaping, and we don't know whether we are able to manage it or collect it. So we have used our brains a bit, and then where we have fabricated a design using the nylon net, and we have kept a banana at one position. So moth used to enter from this position, and they used to feed on this, and they were escaping. So further, we have further developed using another technology where we have kept an inserted cone-shaped nylon net structure, and we have hanged banana at 2 positions. Here, when the moth enters from the below, it feeds on the one rotten banana kept here, and then it will try to fly upward because one more banana is kept, and usually generally insects try to tend to move upwards. In that way, it will feed on this banana, and then it used to be trapped here. And then after some days, so the space present inside this cone as well as this structure, here the moths used to be collected. So we were very happy seeing the result that T7, where we have put an effort to effectively control this fruit sucking moths, and we were able to collect the moths. T7, where mean number of moths collected per trap was highest, which was 23, followed by T6. So these are the major fruit sucking moths. These are the major fruit sucking moths, especially the genus Eudosima is the one particular genus where majority of the fruit sucking moths are causing damage. We could collect moths like Eudosima materna, Rithia hypermenistra, Eudosima salamina, Thias coronata, and Thias honesta. All these moths are considered as major fruit sucking moths. And this and further, my study also highlights that just by using the banana bait as a base, we can effectively make a trap, which will become an indigenous trap, especially for monitoring of fruit-sucking moths. And we were very happy to control these major fruit-sucking moths, which are causing damage, especially belonging to the genus Eudosima. Then, with this, I would like to conclude my topic saying that we, the farmers, as well as the agriculturists, are using different chemicals in the different forms for managing of this fruit-sucking moths. So just by using this simple technology or smart technology, we can make use of different things, and we can fabricate a trap where we can effectively manage these fruit-sucking moths as well as the other pests. So it will become an eco-friendly and sustainable option for us. So with this, I would like to thank all my chairperson, Dr. Kaleshwar Swami CM, and I would like to thank the university for providing me the grants during my course of master's research, and I personally thank my friends as well as PG and PhD students who helped me in collecting this moth in the late night. So, and I also would like to extend my thanks to Malabar Plant Protection Society for giving me the opportunity to present my work on this occasion of World Plant Health Day 2006. Thank you.
Thank you very much, Durga. One question, the last one which you showed, the trap was designed by your group, or it was indigenously designed? last trap
Yes, sir.
with the nylon net
Yes, sir.
was developed by you, your group, or it was indigenous?
Sir, it was actually developed by me, sir, because it was a nocturnal pest. I couldn't go alone for collecting those moths. So I used to take help of the friends, any of the friends who was coming. So I mentioned like it is my team.
Yeah, okay,
That
fine.
way I
One question, you could count the trapped moths and all. Did you ever notice the percentage of infestation in the orchard?
Sir, actually we have not gone to that percentage of...
No. See, the thing is that, people are very happy in seeing the trapped insects.
Yes.
This happens in the case of fruit flies also. When you have plenty of fruit flies trapped in a poisonous trap, we get elated.
Yes,
but most of the time, they would have already oviposited in the fruits and then came and got killed. My question is, when you have such a trapping system here, especially when it is tested in an orchard, you should have noticed or rather observed how much is going to be the percentage attack by the in and around the orchard.
Yes.
Did you have a did you do that, or do you have a?
Actually, it is a seasonal pest, sir. Majority of the time, it is a seasonal pest. I got like 6 months of time for me. I initially with a small pan, then later I ended up with this. And it is a purely adult lepidopteran pest. So it is not a caterpillar which will cause damage. So we were 100% sure that whatever moss that we have collected is trying to help in monitoring the pest, sir.
Yeah, the actual assessment of the effectiveness of the trap comes only when you have with you the damage in an orchard. Otherwise, you cannot say that this is effective. It is effective in trapping the insects, but whether it is effective in bringing down the damage based on the population is a big question. I don't know, like I'm not an entomologist. Okay.
Sir, I feel it is directly proportional here because when moths are trapped...
Without data, you don't say that it is directly proportional. Do you have a data? That is my question. Do you have data regarding the damage caused by, occurring in an orchard where such a trap is installed? That's the question. No, you don't have.
No, sir.
Okay, fine, thank you.
Okay.
Congratulations, Durga.
Next presentation. It's T2OP 7. Sorry, 8. T2OP 8. T2OP 8. Dr. B.R. Rajesh.
Good afternoon, everyone. Respected chair, co-chair, and other dignitaries on the stage. Today I am here to present my topic on bioinformatics and biochemical validation of antifungal biomolecules derived from Spingobacterium thalpophyllum NMS02S296 against banana Fusarium wilt. Fusarium wilt remains one of the formidable challenges in banana cultivation globally. This disease is primarily caused by Fusarium oxysporum formispecialis cubens, disrupting the vascular system of the plant, leading to characteristic wilting and ultimately plant death. The conventional management strategies, including the chemical control and the cultural measures, often fail in persistent soil inoculum and while raising the ecological as well as the resistant concerns. So, the bacterial endophytes which reside within the plant without causing any harm to the plant tissue are deeply employed for combating this deadly disease. So, with this idea, we have gone for isolating these bacterial endophytes from resistant cultivars which often harbor diverse microbial beneficial communities in the plants. So, we have gone for elucidating the antifungal mechanism of these biomolecules from the effective endophyte using integrated bioinformatics as well as biochemical validation. So, this is the research flow or the methodology we have followed. So, from the resistant banana cultivar pisang lilin of the diploid and genotype AA, we have gone for isolating the bacterial endophytes from the different parts of the cultivar, such as leaf, petiole, pseudostem, bracts, etc. So, from the different endophytes isolated, we have gone for the dual culture confrontational assay, and it has proved that Spingobacterium thalpophyllum was one of the effective endophytes with significant inhibitory activity against this Fusarium oxysporum formispecialis cubens. This was later subjected to GCMS based metabolite profiling and later whole genome sequencing. And from the GCMS analysis, the most promising compounds were we were subjected for in silico analysis using molecular docking, and this molecular docking gave us a prediction and a direction to the lab experiments where these compounds having the best binding affinity with the target proteins were selected, and we have gone for wet lab experiments. Through the wet lab experiments, we have finally validated the effectiveness of the identified metabolite or the biomolecule. So this were the results of. So this was the result of the dual culture confrontational assay with the different bacterial endophytes. In this, you can see the T15 treatment has where the Spingobacterium thalpophyllum has shown significant inhibitory activity when compared to the untreated control. So the same was done in vivo using the pot culture conditions. So here also you can see the Spingobacterium treated plants, the T1 plants showed significant reduction in the symptom expression, whereas when compared to the inoculated control where there is vascular discoloration, you can see clear-cut vascular discoloration in case of the inoculated control. So in order to elucidate the antifungal activity of this particular bacterial endophyte, we have further processed with the whole genome sequencing of this particular effective endophyte. So we have identified a number of genes associated with metabolism, protein processing, energy, stress response, DNA processing, membrane transport, RNA processing, and cellular responses. This genome annotation has also identified various genes involved in the metabolite production and antifungal activity. We have also gone for the pan-genome profiling of this particular Spingobacterium thalpophyllum isolate. So our test isolate was compared with the remaining Spingobacterium thalpophyllum strains available in the NCBA database. So 3 other strains were available in the NCBA database. We have compared our test strain with the other Spingobacterium thalpophyllum strains, and we were able to identify that this particular species possesses an open pan-genome. This means that as the new genomes are added, the species possess the ability to add new genes capable of bioactive metabolite productions. And also we have compared this Spingobacterium during this comparative genomics. We have identified that our test strain was able to occupy maximum number of MAM genes as well as hydrolytic genes. MAM genes such as elongation factor as well as hydrolytic genes like chitinase genes. So further, this particular effective endophyte was subjected to GCMS-based metabolite profiling. In this metabolite profiling, we have identified a number of promising compounds, particularly methyl 2,4-dihydroxybenzoate, hippuric acid, hexadecanoic acid, etc. So these compounds were docked with the different target proteins of FOC involved in the growth and differentiation of FOC. So this molecular docking analysis revealed that particularly hippuric acid as well as methyl 2,4-dihydroxybenzoate were found to have higher binding affinity with these target proteins. They were found to have higher binding affinity with the proteins involved in the growth and differentiation of FOC. So this is the interaction structure of the methyl 2,4-hydroxybenzoate with different protein targets of FOC. Here you can see the binding affinity interactions with the particular metabolite. So this is also the binding affinity interaction with hippuric acid with the different target proteins of FOC. The binding affinity was found to be nearly 7 kilocalorie per mole. So as a result of this molecular docking studies, we have proceeded with the further wet lab experiments validating the efficacy of this molecule in this in vitro assay. So these molecules, hippuric acid as well as methyl 2,4-dihydroxybenzoate, were purchased in its pure form and they were validated in the wet lab using poison food technique at different concentrations of 1000, 750, 500, 250 and 100 ppm. And it was found that both these molecules were found to be promising, and they were having a concentration based inhibition, particularly at 1000 ppm. Both the compounds were found to have 100% inhibition, and this methyl 2,4-hydroxy was found to have higher inhibitory efficacy when compared to the hippuric acid. So this integrated and biochemical validation has come up with the two promising molecules such as hippuric acid as well as methyl 2,4-dihydroxybenzoate, and this can be used as a sustainable alternative for the management of Fusarium oxysporum formispecialis cubans. We have also done a baseline study for the nano formulation of these two molecules. Currently, the work is undergoing by the subsequent student. With this, I would like to conclude and acknowledge my chairman, Dr. S. Nakiran, and Tamil Nadu Agriculture University for providing me the opportunity. Thank you.
Thank you, Ajish. My question is, what is the surety that within the plant these two compounds, hippuric acid and methyl 2,4-dihydrobenzoate, would be produced? What is the surety that these two compounds are produced within the host plant? This is an in vitro system where there is no host involved. Yes. This is a pathogen and background agent interaction alone. And when it comes to a third party, what is the chance that these two compounds are getting produced?
So these molecules are purely 99% pure molecules?
No, that's not the question. The bacteria may produce some other compound when it is inside the plant. And there could be, unless and otherwise you detect in the in vivo system the presence of these compounds, you cannot say that these two compounds are responsible for the biocontrol activity of this particular tripartite system. Okay, 1, 2, 3. Three things are there. In your analysis, there are only two things. Of course, I appreciate, but still. You will have to extract these two compounds from the pathogen host and biocontrol system, yes, and then prove that this actually is responsible for. Then a big question is that when the bacteria itself can control the disease, why you go for getting its metabolite extracted, and what is the way by which it is getting it should be injected maybe or applied in a different way.
So the survival of different endophytes in this different field conditions may be varying, sir. So utilizing these bioactive metabolites may be a solution for alternative
So what is the
solution.
possibility of getting them synthesized, in chemically? Did you go for the structural aspects of uric acid and the other compound?
Sir?
Structural elucidation of these compounds. Oh, it's okay, fine. Thank you very much.
Congrats, congrats, Sajesh.
Thank you,
I
ma'am.
have one question: whether it is possible to control the disease using this particular compound itself,
Yes.
directly applying.
Yes, ma'am.
So, is it possible to manufacture it or produce it in a large
The
quantity?
extraction will be difficult. We have actually purchased the
Just
molecule.
like how we extract
The
penicillin from.
natural way of extraction will be a difficult procedure.
Maybe we'll have to genetically modify the organism to produce this compound in excess. Is it possible?
We can try.
Any such
Yes, ma'am. We can try that.
Okay, congrats.
Thank you, ma'am.
Any question from the audience? Rajesh.
So the molecular docking experiment was the basis for selecting these two compounds. These two compounds were having the highest binding affinity with the different proteins of Fusarium oxysporum select. Yes. Sometimes in GCMS you will be having higher peak area percentage. Still, the compound may not be having the binding affinity with this particular target proteins. So even if the peak area percentage is less, we should try out. We should select those compounds also for the molecular docking experiment. Yeah. Yes. Actually, culturing and extraction will be a difficult process. We have actually gone for purchasing these pure molecules in its pure form, and then we have gone for applying. Yes. Okay. Those needs to be carried out. This needs to be validated. Thank you, sir.
Okay. Now we are moving to the two flash talks. We are having three posters, but they are presenting. And first, it is T2FT1, Fidanaurine, application of nanotechnology for enhanced crop protection and plant health.
Imagine a future where the diseases can be detected even before the symptom appears, or fertilizers that can release nutrients in a regulated and targeted way, or tiny particles, even which are smaller than cells, that can actually help the plants to survive plant stress and also drought. I'm not talking about a distant possibility, but I'm talking about emerging agriculture reality. Good afternoon, everyone. I'm Fida Naurin, currently pursuing Bachelor's in Agriculture in College of Agriculture, Patnagar, and I'm here to present a flash talk on the topic, Application of Nanotechnology for Enhanced Crop Protection and Plant Health. As we know, today agriculture faces many challenges like extensive dependence on chemical fertilizers and climate change and the incidence of pests and diseases. In these scenarios, nanotechnology is an innovative tool that can be used for mitigating these challenges. So, the first advancement in nanotechnology is the development of nanofertilizers and nanopesticides. These nanofertilizers release nutrients in a slow, regulated and targeted manner, which actually helps to reduce the chemical wastage. And also we know that in conventional fertilizers, a large amount of nutrients is actually lost through leaching and volatilization. And another advancement in nanotechnology is nanosensors. Nanosensors are actually extremely sensitive, and these can detect plant diseases, environmental stresses, and nutritional deficiencies even before the symptoms appear. And these nanosensors actually collect real-time data from the field, and these data can be connected to an IoT systems which can analyze these data, and they can give these results to the smartphones on farmers, which can be used by them for a better planning of agriculture. And then we know that one of the major threats that is faced by global agriculture is climate change. And nanoparticles helps in this too. Nanoparticles helps in water retention and thus by maintaining hydration in plants. And also these nanoparticles can enhance antioxidant activities by stimulating antioxidant enzymes like peroxidases, catalases, which actually helps us to reduce reactive oxidation species inside the plant cells and thus reducing oxidative stress. So, I think even when we are racing towards technology, we shouldn't be doing that. In the cost of environmental security. So for that, green synthesis actually gains importance. Green synthesis is the use of environmental eco-friendly and also less toxic nanomaterials for the preparation of nanoparticles. And in conclusion, I would like to say that these are some innovations in nanotechnology, and this can be used for, and this has huge potential in to transform plant health and also for sustainable agriculture. And I know these innovations are still in developing and in an early adoption stage, but in future it will be having a good scope. And by saying that, I would like to conclude my talk. Thank you.
Thank you, Fida. Just a curious question, okay. One curious question. Have you come across any environmental implications regarding the use of nanoparticles? Like, this is most of the time there are so many non-targeted organisms, non-targeted entities. What actually is the implication with respect to the use of the nanoparticles? Have you come across any such studies, references, or something of that sort?
I think nanoparticles usually focuses on the targeted organisms only, but as technology, it can also be having some of its disadvantages too. But I think we have to go more studies should be there for it.
Any questions from the audience? Congratulations, Fida. It was a nice presentation from the part of a UG student. I immensely congratulate you. Thank you.
Thank you, Fida. Next presentation, T2F FT2, mechanized climber for Arikanet. A review, Ashna P. Assis.
Good afternoon to one and all, respected dignitaries and beloved audience. I am Ashna PSC from College of Agriculture, Parnakat, BSc Agriculture, 4th year student. Just imagine, we are standing beneath a 60-foot arecanut tree. A worker, a rope tied around his waist, a single sickle is in his hands, and a sprayer, a pesticide sprayer on his back. A small mistake from his life to death. This is not just an imagination, these are the realities. These are some of the news headlines like that workers falls from arecanut tree dies and then labor shortage hits arecanut harvest, like that. These are the real newses. India is the world largest producer of arecanut. In India, Kerala, the states like Kerala and Karnataka, this is not just an agriculture for them. It is just their livelihood, it is their economy and it is their culture. Even though single climber may charges high amount during the peak harvesting season, during shortage of labors because of that. So by then the nuts become overripe and diseases become spread and thereby economic loss will occur. At the same time, the manual climbing is also very risky. So the question is, can technologies climb where human struggles? This led to the development of the mechanized arecanut climber. And that arecanut climber Arecanut climber is an assistive agricultural machine that is used to climb arecanut trees safely and for plant management operations and for monitoring, for nutrient application, for pesticide applications, like that. This system combines mechanical designs and autonomically and also sensors and regarding like that. And the basic principle of this project is very simple and it is very innovative. That it is like the machine creates a grip around the tree, and when it is powered, it is the machine grip around the tree when it is powered. And then the motor will rotate the wheels, and then the machine will move upwards. This is the mechanism. And the main advantages of this project is the reduction of the labor dependency and the reduction of the accidents, timely harvesting, managemental operations, or like that. And in future, we can upgrade this system to an autonomous arecanut climber by combining artificial intelligence, remote sensing, and by GPS. So as a conclusion, these types of ideas represents a future step towards safe and sustainable agriculture. Thank you.
Thank you, Shna. As you said, I am asking another question. Is it imagination or reality? Okay. That means, this is an existing entity.
Yeah, these are autonomous, mechanics arecanut climbers are existing, but completely autonomous are still on working project.
Yeah, that means, is there a marketable product or something of that sort? This is currently being used.
I think it's mechanized arecanut climber is currently in use, but the autonomous arecanut climber should
mechanized means a man goes along with the climber. A man or a human being goes along with the climbing device, or rather it goes up and then collects the nuts and come back.
We can control it by remote, or we can control by machine itself. That's why this mechanism.
Yeah, that means, it's where is it being used? Which part of the country it is being used?
This is not using currently, but
That's what, like it's only a prototype or imaginative or
is a prototype.
under development or whatever it is.
Several working are going on this prototype.
Okay, thank you very much.
All the presentations are over now. There are no posted presentations. It's over. The session is over.
Poster presentations?
ഇല്ല മാം ഇല്ല പോസ്റ്റർ ഇല്ല
ഇല്ലല്ലോ
ഇല്ല.
Thank you very much for your active participation, and this particular session comes to an end. Artificial intelligence and plant health management. There were talks which were insightful, and there were oral presentations which are competitive in nature. I wish all the participants and delegates a bright future. Thank you very much.
Thank you, sir. Now we invite Dr. Mini PK to present the memento to the chair and co-chair on behalf of Malabar Plant Protection Society. Thank you, ma'am. Now it's time for lunch break. The third technical session will begin at 2:30 p.m. And we have another important announcement. The technical session 4 will be held at Audiovisual Lab at 2:15 p.m. We are now going to begin the third technical session focusing on invasive pests and pathogens in changing climate. The chairman of the session is Dr. Susha S. Associate Professor, Department of Plant Pathology, College of Agriculture, Vellayan, and the co-chairman is Dr. Nisha M.S., Associate Professor, Department of Entomology, College of Agriculture, Panagar. The rapporteurs are Dr. Siddharth K. and Ms. Anusri. Humbly requesting the chair and co-chair to conduct the session.
Good afternoon, everybody, to the technical section 3,
that is the invasive pests and pathogens in the changing climate. And in this section, there are two lead talks led by Dr. Jairaj P., former program coordinator of KVK Kannur, and Dr. K.N. Anith, the Director of Research of KAU. And first of all, Dr. P. Jairaj, that's having the lead talk on the plant nutrition and plant health management. And first talk is by Dr. K.N. Anith, Director of Research, KAU, and it's by the prospects of endosperm-forming endophytic bacteria as plant growth promoters and biocontrol agents. And Dr. K.N. Anith is currently serving as the Director of Research at KAU and specialized in plant microbe interactions and biological control of plant diseases with decades of research experience in developing eco-friendly and sustainable agriculture solutions. Sir has completed his postgraduates and the doctoral studies in microbiology from the IARI, New Delhi. And then he has received the prestigious Poiskas Fellowship instituted by the Department of Science and Technology. And after that, he has pursued the postdoctoral training in plant microbe interaction at the University of Florida. And Dr. K. Nanith has made significant contributions to the agriculture research, particularly in the development of microbial biocontrol technologies, including widely recognized Pseudomonas fluorescens formulations used for plant disease management, currently being commercially distributed by KAU. And he has also published more than 85 research papers in the reputed national and international journals, and has guided many MSc students and the PhD scholars in their research careers. We are honored to have Dr. K. Nanith with us today as the lead speaker. Let's warmly welcome Sir to this speech.
Thank you very much for the nice words. Good afternoon, everyone. I don't know how best fit this would be to the seminar, but like there is some words, something like changing climate, etc. So I have selected a topic which would somewhat align with the objective of the seminar program. My topic will be divided into these parts, what is microbiome, then what are endosperm-forming bacteria, because I am interested in that, a specific group of organisms which are very recalcitrant in nature, very very much capable of thriving adverse environmental conditions which are present in soil, plant, inside the plant, and everywhere. Those are the endosperm-forming bacteria. Then still cutting down into endosperm-forming endophytic bacteria, those bacteria which are endosperm-formers but still capable of dwelling within the plants. Then I'll be talking on isolation methods, and then the plant growth promoting properties of endosperm-forming endophytic bacteria, and EEB as biocontrol agent, and some specific examples of plant growth promotion, and then biological control, and some questions which could be asked regarding the plant microbe interaction at some functional levels. Then future research programs, etc. Okay, I'm going to present everything which I have done or my group has done. The presentation will be very much oriented towards the results and the methodologies which we have developed in our laboratory. So I have not taken anything from outside. Whatever we have done, I'm just presenting an overview. Next one. Yeah, as you know, microbiome is the microbial community that are associated with a plant. different tissues like flowers, roots, philosphere, etc. The whole genome, genetic makeup of microorganisms which are closely associated with a plant is known as plant microbiome. We have our own microbiome, gut microbiome, for example, those bacteria or fungi or whatever it is present within our gut. Next one. Yeah, if you see a plant microbiome, we can divide them into different compartments, the upper part and the lower part, or rather the below ground part and the above ground part. Above ground part we have philosphere, and below ground you have rhizosphere. These are broad terms, but you do encounter different types of microorganisms everywhere. And one particular niche is that endosphere. You get endorhizospheric microorganisms, you do get endophilospheric microorganisms, you do get within the stems also. So all those microorganisms are in general referred to as endosphere microorganisms or endosphere microbiome. Next slide. Yeah, coming specifically into the endospore forming bacteria, these bacteria are gram-positive bacteria. There's only a single genus which belong to gram-negative kingdom, especially, say, for example, Bacillus, Pinibacillus, etc. They have a peculiarity that at adverse environmental conditions, they produce spores which are capable of thriving under adverse environmental conditions. Say desiccation, nutritional deficiency, less moisture, drying, etc. All these conditions, they produce spores, which are otherwise known as endospores, just because of the fact that the spores are present within the cell. There is a tough integument of the cell, or the cell wall has got some modification so that they are resistant to the action of staining program, resistant to the action of heat, resistant to the action of acids, etc. So they simply survive in the environment where they are. And once they encounter with a favorable environment, these spores start germinating, and then they become a vegetative cell. And the vegetative cell, as you know, would multiply. So this is the importance of spore-forming bacteria. Next slide. Yeah, these are some of the peculiarities. Like it has got a very peculiar cortex structure. Heat shock proteins are more in that. There could be mineralization, especially with a specific compound called calcium dipicolinic acid, which is nowhere present elsewhere. These are some of the peculiarities with which the bacteria is capable of thriving the bad environment. Next slide, please. Yeah, coming to endospore forming endophytic bacteria. How are we going to isolate endospore forming endophytic bacteria? Like if you have a plant sample, you wash it and then surface sanitize it, and you can macerate it under a sterile environmental condition. And the next step is going to be a physical enrichment. Physical enrichment is done by heat processing. Endospores of bacteria have got a peculiarity that even if you heat them up at 80 degrees centigrade for 10 minutes, they will not get killed. But all other bacteria, any bacteria other than endospore, don't get confused between endospore forming bacteria and endospores. Every endospore forming bacteria will produce endospores. Okay. But when I refer endospore forming bacteria, that need not be in the sporulating stage. It could be in the vegetative stage. So when you have a mixture of tissues and then it is getting heated up, all the vegetative cells will getting killed. Remember, the vegetative cells of endospore forming bacteria will also be getting killed. Vegetative cells of the endospore forming bacteria will also be getting killed. But the endospores will remain. So you have vegetative cells of ordinary bacteria, vegetative cells of endospore-forming bacteria, endospores, and first two will be getting killed and the endospore will remain. Now you plate the solution and the endospores will now germinate and become colonies. Okay, and any colony that is coming out after this process is going to be the colonies of endospore-forming endophytic bacteria. Endospore-forming endophytic bacteria. Okay, so then again the colonies could be visualized by a microscopy. This process is known as a conventional one-step enrichment process. One-step enrichment. There is only a single step of enrichment, physical enrichment, that is after trituration, you are heating it up, and that is a single step of enrichment, where the proportion of endospores will be increased when compared to the proportion of vegetative cells. Now we have developed a modified version. This is the next slide. This is just an example. From ginger rhizomes, we just surface sanitized, washed in sterile water, and there is a sterility check which we have to do because we want to make it sure that it is endospore, not a surface-dwelling bacteria. When after surface sanitization, it will be washed in water, the washette will be plated, and there should not be any bacteria coming up there. Okay, so that is what is known as sterility check. Then the rhizomes are crushed, then it will be heated up at 75 degrees centigrade or 80 degrees centigrade for 10 minutes, spread plated. Okay, this is a single step enrichment process. That means 75 degrees centigrade for 10 minutes. Next slide, please. Yeah, here we are doing a double enrichment method. What we do is the rhizomes will be collected, washed, sanitized, and then they will be kept in a hot air oven at 35 degrees centigrade for 2 days. That means the moisture content will be getting reduced so that every vegetative cell of the endospore forming bacteria will be getting converted into endospores. Every vegetative cell of the endospore forming bacteria will be getting converted into endospores. That is the first step. Then again crushed the rhizome and then spread plated. Again once more we are enriching. A vegetative cell that is remaining after the first step of enrichment has to be killed. So the second step of enrichment is again done for 75 degrees centigrade for 10 minutes in a water bath and then spread plated and the colonies will come up. So you make sure that all the colonies that are coming up after two-step enrichment is going to be in colonies of endospore forming endophytic bacteria. Okay, next slide. Yeah, this is a typical example. The first plate shows that without enrichment, you get plenty of colonies. The second, in the lower panel, the first one shows the single step enrichment. And the third plate shows the double enrichment. So there is increment in the number of spores forming bacteria in the case of double enrichment rather than the single enrichment. Next slide. Yeah, this is just a comparison of conventional and double enrichment method. You can see that there is wide gap between the conventional method after heating and before heating, but there is narrow gap in the double enrichment method. That means, the bacteria which you get most often are going to be endospore forming bacteria in the case of double enrichment method. Next slide. Yeah, we have done this conventional and modified double enrichment method in different crop plants. One is the amaranthus. You can see the difference between the single enrichment and double enrichment. This is another amaranthus variety, Co1. Next slide. Yeah, we have published a couple of papers in plant science, biological management of rhizome root rot, based on the results. Then another one in amaranthus also. Next one. Yeah, we see that even there is large difference between the genotypes of plants which are grown nearby with respect to the kind of endospore forming endophytic bacteria. So there is the recruitment process is different in different genotypes and species. That is very important. So species specificity or genus specificity exists in the case of the endospore forming bacterial. Yeah, next slide. So once you get the endospore forming bacteria isolated, for plant growth promotion studies as well as for biocontrol studies, we need to know about the characteristics of these organs. Most often, several of such abilities are tested. One is nitrogen fixation. Actually, nitrogen fixation is assessed based on the functional aspects of NIF genes, but if you do not have any capacity to do that, one thing can be done in a nitrogen-free medium. You can streak plate the bacteria and see if they are coming up or not. It's a putative method. Suppose if they come up, we can assume that these bacteria are capable of fixing atmospheric nitrogen. Other methods are there, like acetylene reduction assay, which requires a gas chromatograph. Then you have other specific NIF-H, NIF-K, NIF-D detection system, where you need to have a PCR machine for that. Then we have P solubilization in a Pikowski medium. Then there is another medium, what is known as NBRI medium, National Botanical Research Institute medium, that also shows P solubilization. Then siderophore protection, these are ion chelating compounds which help the bacteria in controlling plant pathogens. Next slide. Then hydrogen cyanide protection, IAA protection, GA protection, ACC deaminase protection. All these, several of such characters are assessed, and based upon that, you can select the best endospore forming endophytic bacteria. Okay. Next, another thing is that with respect to plant growth promotion, the best way is to assess the behavior of the bacteria in a miniaturized system. The best thing would be a roll towel assay method. Like you treat the seeds with specific endospore forming bacteria and then keep it in roll towels, and then after a couple of days, maybe around 10 days or 15 days, you can see how the growth pattern differs. This is just an assessment with which you can have a preliminary assessment of the plant growth promoting activities of the bacteria. The first thing were in vitro assays, which may not be reliable in nature. That all depends upon the kind of media, kind of laboratory conditions, etc. But this is a bit more reliable because there is an interplay between the host and the bacterial strains. Next. Yeah, you can clearly see this is in the initial stage. So any increment, any marginal increment has to be taken into consideration for selecting the bacteria. Next. Yeah, next slide, please. Once you get the plant growth promotion abilities assessed, the next one is that interactive assay between a pathogen and the bacterial strains which you have isolated. Okay, this is a kind of system what is known as dual culture plate assay, wherein in a plate the fungus and bacteria will be grown together and see if there is an inhibition that occurs. So you can see the differences in inhibition. In lower panel, the first one has very good inhibition. Many of them do not have. So based upon this, you will be selecting the best one. But when you have large number of bacteria, 1,000 isolates you have, it's very difficult to screen in a dual. Each one needs one single plate. And a single plate needs around 25 to 30 milliliters of medium. So it's a time-consuming process. So we have developed, Naishant is here, he was my colleague and student, and then we developed a system in which you could screen a bacteria against 4 different fungi in a single plate. Here, a single fungi versus single bacteria can only be done, but you can do with multiple pathogens. Say there are, in the case of damping off, you have several pathogens. Pythium you have, sometimes you have Phytophthora also that causes damping off. Pythium, different species you have, Pythium aphanidermatum, Merolyticum. So many are there. So if you need to screen a single bacteria against multiple pathogens, each pathogen requires a single plate. But we have developed another system. Yeah, this is a culture filtrate assay. Leave it. Okay. Yeah, once you get a dual culture plate assay, you can see the zone of inhibition and then select the best one. Okay, I am talking about isolation and then characterization and then screening for plant growth promotion, screening for biocontrol. All these steps are to be done one by one after another, and then you have to select the best one. Okay, next one please. Yeah, this is the dual culture plate assay wherein a single bacteria will be screened against a single pathogen. Yeah, this is a graphical representation. Now, next one. We have developed a streaking method known as plus streak method. So what you do is, in the petri dish, we streak as a cross. Then, at the four corners, you will be keeping 4 different fungi. So, 4 different fungi could be screened against a single bacterium in a single plate. Otherwise, it would have required 4 different plates. Now, you require only a single plate. Now, you can measure the diameter of the control and diameter of the test, and then see the inhibition percentage. So this is such a system. Next one. We have also developed another thing. It's called a square streak plate, square streak assay, wherein a square is drawn so that at the 4 different edges of the square you keep 4 different fungi. So that same is the case with the cross streak and then this one. 4 different fungi could be screened against a single bacteria in a single plate. Okay. Yeah, you compare this dual culture plate assay with the modified plate assay. Modified plate assay is, one is square streak, another one is the plus streak. See, for you see the last row, 10 bacteria, if you want to take see the antagonist against 10 fungi, you require 100 test plates and 10 control plates in dual culture plate assay. That means you require 110 plates. But in the modified assay, you need only 30 test plates, 3 control plates, and then total is going to be 33. So instead of 110 plates, you can reduce it to 33 plates. Okay, so you save a lot of time, lot of energy, lot of money. Next one. Yeah, we have published it in Letters in Applied Microbiology. Next one. Yeah, now we have again modified the plus streak method in different way. That means instead of a cross with a single bacteria, we did it with 2 different bacteria. The vertical one is a single bacteria, horizontal one is yet another bacteria. To avoid the cross contamination, what we have done is we have removed at the center agar. Agar was removed from the center, and then the vertical one is bacteria A, the horizontal one is bacteria B. That is present in the graphics. One is red in color, another one is blue in color. So that means 2 different bacteria could be screened against 4 different fungi in a single agar plate. So we named it as modified plus streak method. Modified plus streak. Next one. Yeah, we have again done in a different kind of assay, but this time it is big plates, 20 centimeter plates, which is parallel multiple streak method. And if you see the graphics, the first one is the blue color is bacteria A, red is bacteria B, then again A, B, A. Now, in this 2 different bacteria could be screened against 10 different fungi in a single agar plate method, agar plate. So you save lot of time and money. So see the difference. DCP means dual culture plate. PMS means parallel multiple streak. MPS means modified plus streak. See the last one. Suppose if you want to screen 100 bacteria against 10 fungi, you require 1,000 plates. and 10 control plates, total 1010 plates. And the media, if you consider it as 20 ml for a plate, you require 20 liters of medium for if you want to screen 100 bacteria against 10 fungi. But in parallel multiple streak assay, where you use larger plates, which may have 50 ml of the medium, you only require 50 plates and one control plate. Total is 51, and the total amount required is 6 liters. And in modified plus streak assay, you require 150 plates, 3 control plates, total 153. But since the volume is 20 ml per plate, it is going to be 3 liters. So you see the difference. Suppose if you do the conventional dual culture plate assay, you require 20 liters. Modified parallel multiple streak 6 liters, and then modified plus streak only 3 liters. You save a lot much of money, and only thing is that you have to be very careful in assessing that there should not be any contamination. Next one. So we have also published in 3 Biotech. The Nation is the lead author in this paper. Next one. Yeah, another thing is that sometimes bacteria produce volatile compounds, which are volatile organic compounds, which are very important in plant disease control. It's usually assessed by a paired plate technique. You can see the difference in the growth of the pathogen in the paired plate. In the lower plate, the bacteria will be streaked. In the upper plate, the fungus will be placed, and then see the growth of the fungus, and the inhibition percentage will be worked out. Next one. Yeah, this is non-volatile organic compound production. This is assessed by incorporating the cultural filtrate into the medium, and then over which the fungus will be grown. Yeah, once you have these things, isolation is over, the bacteria are characterized. Now you have assessed the initial potential of the plant growth promoting traits, then done a roll towel assay, then antagonism is tested. Now the next thing is that to see, when you have the antagonism tested, there is only 2 players. One is the antagonist, another one is the pathogen. There is no involvement of the host. But wherever there is a host involvement, there will be 3 players. One is the host, second one is the biological control agent, third one is the pathogen. Such a system can be developed. It's very easy. It is usually done by detached leaf assay. You detach the leaves, wash it, and then spray with the bacterial solution, and then inoculate the fungus. The pathogen, whatever it is, fungus or whatever it is. Then you see incubate at a very humid condition, and then find out the development of the lesion. The first one, the strain WL2 limited the development of the lesion very well compared to the inoculated control. Next one. Yeah, this is done in different systems. One is black pepper. Then you have detached fruit assay for Colletotrichum. So these things will tell you what may happen in the field. In the dual culture plate assay, we have done some research in that. Dual culture plate assay will give one result. The detached leaf assay will give another result, and there is not much correlation between these two when it comes to the field. So you have to be very careful in assessing anything which give large inhibition in the dual culture plate assay may not give very good performance in the detached leaf assay. But detached leaf assay or detached fruit assay is comparable to the field condition. Therefore, you may have to go for the result, depend upon the results which are in the detached fruit or leaf assay. Okay. Now, yeah, this done in stem assay also. What we have done is black pepper stems were cut and then dipped in bacterial suspension and dried and then pinpricked and inoculated with the Phytophthora capsicum and see the difference in the lesion developed. So we can screen like that also. Next one. The same thing can be done for induced systemic resistance. Induced systemic resistance is a thing where the bacteria induces some enzymes or defense-related products within the plant system, and then make the plant resistant, or rather somewhat tolerant to the infection. What we have done here is that in the first one, we took the cuttings and then dipped the lower portion alone in the bacterial suspension, so that only the lower portion, the cut end, will be dipped in the bacterial suspension, so that the bacteria will not traverse in the above part, and then inoculated in the upper part. We made sure that the bacteria does not go. We got good. The first one is control, and others are treated ones. So what can be done is that we can ensure that the bacterial antagonism is not due to the direct activity, because there is a spatial separation between the inoculated site and the bacterial treated site. So what could be the reason? Be the enhancement of defensive capability of the plant or plant part with the help of the bacteria. To make sure that we have done another thing. Same experiment, but this time what we did was, after dipping the cut end of the cuttings, we took it and then wiped the whole part with alcohol, so that we made sure that there is no bacteria present. And again repeated the inoculation with the pathogen. Almost same results. We could get almost same results with alcohol wipe and without alcohol. So we can, instead of the direct antagonism, we can go for the indirect antagonism by way of the induction of resistance also. That screening could also be included in our study. So you have isolation, then characterization for plant growth promoting traits, then biocontrol traits by plate assays, then simulated results by detached fruit and leaf assay, and last one is going to be the test for ISR activity. This could be done in seedlings also. You take seedlings, add the bacteria into the root zone, and then if it is a foliar pathogen, infect in the seedling stage itself, give the infection in the seedling and see if there is any difference or not compared to the control. Very easy. Okay, next one. So, the plant growth promotion usually appears in upper part of the plants as well as the lower part of the. Most often, it is very correlated. Larger the volume of the root biomass, there will be more effect in the upper part. Next one. Yeah, these are some of the examples wherein we have selectively used different endospore-forming endophytic bacteria, and then the parameters which we took for assessing the plant growth promotion, like always you go for dry weight basis. That is more reliable than the fresh weight basis. Next one. Yeah, this is yet another example which we have done. Just wanted to show that the plant growth promotion is a factor that is very much dependent upon the root development. Next one. Yeah, here is an example where the leaf blight pathogen was controlled by spray with endospore-forming endophytic bacteria. One thing is that these endospore-forming bacteria are recalcitrant. Therefore, if you spray, they will stay there. They won't get killed. But when there is moisture content, they will start germinating and start working. Next one. Yeah, these are all some of the examples. Okay, I'll rush off. Okay. Next one. Yeah, then there is a simple method by which you will be able to see whether these bacteria, endospore-forming bacteria, are capable of surviving in the plant structure or the soil, etc. Only thing is that this is what is known as relative abundance. suppose if you treat a plant with the endospore forming bacteria and then grow them, if you want to see whether there is sufficient colonization, sufficient multiplication of the bacteria in the rhizosphere of that particular plant, you just uproot, get the bacterial suspension, heat it up and then plate and find the number. Same will be done with the untreated control. Same soil, same condition, untreated control, where you uproot the plants and get the rhizosphere soil, make a suspension, heat it up. So you have endospore forming bacteria naturally present in the control system, endospore forming bacteria naturally present plus the applied one in the treatment. So you have the number of colonies in the treatment minus the control divided by the control will give you percentage abundance of the bacteria. And we thought first when we proposed this, we thought it will be rejected. This particular theory will be rejected. But I'm happy to share it with you that this was taken very positively by the reviewers, and then I think we have more than 30-35 citations on this particular aspect itself. Next one. Yeah, there is a big question. And you are getting too many number of bacteria in the isolation process, whether to go for single bacterial application or a consortium. Okay, next one. Yeah, in this experiment we did compare Bacillus pumilus and a consortium of 3 different bacteria: Bacillus amelia-liquefaciens, Bacillus valescensis, and Bacillus pumilus. 1 versus 3. But there were so many parameters which we have checked. Many were positive for the single bacteria rather than the consortium. Because in the consortium sometimes there will be less cross-compatibility. There will be more incompatibility between the bacteria that are present in the consortium than in the single. But contrasting results could be obtained. Next one. Yeah, here too, here some, see the fresh weight 16.53 in single inoculation, it is only 9.5 in the consortium. You remember, that particular bacteria is a part of this consortium. The first one is a part of the consortium, but still there is, less growth. Most of the cases, the first lane, the single inoculation was better than the consortium inoculation. Next one. Yeah, here too, there was some confusion, like when 2 were combined, there was reduction in the parameter scale. Okay, next one. Yeah, please. Yeah. Another question, whether the host-derived endospore-forming endophytic bacteria perform better. Suppose, you can isolate endospore-forming bacteria from tomato. You can isolate it from amaranthus. You can isolate it from cowpea, whatever it is. The cowpea-derived bacteria perform better in cowpea, or cowpea-derived bacteria perform better in tomato, or tomato-derived bacteria perform better in tomato or in cowpea. This is a big question, because is there any relationship between the genus of the host plant and that of the bacteria? That we have seen that there exists. So better to, suppose if you want to have the bacteria isolated and tested for disease control or plant growth promotion, better go with the exact host. Suppose if you want to do research in rice, try to get it out of rice rather than getting it from some other crop. So we have seen that EEBT is tomato derived, EEBC is cowpea derived. In cowpea, you see the germination percentage is 100% when EEBC, cowpea derived bacteria, were used. Same is the case. All parameters were more in the case of cowpea when cowpea derived bacteria were used. Okay, even it was less in the case of when the consortium was used, EEBT and EEBC. Next one. This is the same case. In cowpea-derived bacteria performed well in the case of seedling vigor index 1. Next one. In tomato, it is reversed. Tomato-derived bacteria performed well in the case of tomato. This is seedling wear index 1 in the case of tomato. Here, tomato-derived bacteria performed better in tomato than cowpea-derived bacteria in tomato. So there is some host specificity occurs. Most often what we do is somebody would isolate from some crop, we will borrow it and test in our crop. And then we say that this is good. You have better choices. Then suppose you have the host plant in your locality, try to isolate and then use the same thing. Next one. Here it's the same case, SCV, that means seedling vigor index 2. Same is the case, tomato, good. In plant growth, that was in plant growth promotion. With respect to the damping of suppression, same thing. In cowpea-derived bacteria performed well. Next one. In tomato-derived bacteria performed well. And damping of suppression. Next one. Yeah, these are some of the parameters, but I wanted to say that like it is better if you have those bacteria which are present in the exact host than going forward with a related species or genus. Whether we will be able to combine endosperm-forming bacteria with other endophytes. We did research in which some of the bacteria were combined with the beneficial endophytic bacteria, Piriformis poroindica, and of course we got positive results with some bacteria, but not with all. There also is the relationship between the two different bacterial endophytes as well as fungal endophytes. Next one. Yeah, you could see when there is combination, sometimes it outperforms, but in some other combination it doesn't. Yeah, leave it. Next one. Now, some of the advantages with respect to the endosperm-forming endophytic bacteria, plant growth promoters, and biological control agents is that their formulation is very easy because you don't have to. They will survive in water for 100 years. They will not get killed unless and otherwise there will be large changes in the environment. Okay, so you can have the cells collected and then put it in water. They will stay, no issues. They will form endospores and then stay for hundreds and thousands of years. There is a better survival. You don't have to add preservatives, usually polyvinyl pyrrolidine or glycerol or trachelose, mannitol, etc. are added to preservatives in liquid formulation, but those are not needed. You can easily make liquid formulations and very easy to store it. This is with respect to the endosperm-forming bacteria. Not only the endosperm-forming endophytic bacteria, but any endosperm-forming bacteria. Okay, next one. Yeah, we need to know novel delivery mechanisms in the plants. Your research, Ajesh, okay. Your research has shown that there are different metabolites that are capable of inhibiting the pathogens, and of course a synthetic development of such metabolites will pave way forward for developing novel fungicidal compounds. That is very important, and it is very important to see or derive the isolates with multiple traits. That is very important. Plant growth promotion, then another one is biocontrollability, then induction of resistance. Then you have another thing is that post-harvest preservation. Some of the bacteria, when applied to the root zone, will produce fruits which will have more preserved capabilities. We may wonder how, but sometimes there are processes known as suberization, cauterization, etc., which will help the fruits stay afresh for many times. Okay, so anti-herbivory, it's another thing. Sometimes application of some of the bacteria will prevent the herbivorous activity of insects. So you can have multiple strains with multiple traits which could be combined as a consortium and then used. Okay. Next one. Finished. Okay. Thank you very much for the patient listening. I'd be glad. I would be glad to answer a couple of questions if there are any. Okay, please. Pardon?
Under nature, in vivo plant condition.
In vivo plant condition. Okay. So this is a good question. You mean to say that plant is always with full of water, and then always it is bacteria are happy to be there. Okay. That is your question. Okay. I can understand. See, in an ideal condition, a spore-forming bacteria will never die. There is no death for them. But it dies because of several other conditions. Sometimes there will be toxic things which are produced by the plants. You don't know. See, Dr. Srimar is gone. Okay. Srimar would say that the worst toxic factories are plants, because there are n number of compounds which are produced by plants. So what happens is that at times the plant itself will produce some of the toxic compounds. Then those bacteria which are present within, get converted into spores. They have to survive. They will go to the next generation. Most of the time what happens is that in plants, the expression is not throughout their life cycle. Sometimes it is spatially separated, sometimes it is temporally separated. A young plant may not have toxin, but when it flowers, it will produce toxin. But these bacteria need to survive in the toxic condition to go to the next generation. There is horizontal as well as vertical transition of the bacteria from one generation to another through seeds, or from one population to another through other mechanisms like pollen grains. They do go. But don't ever think that these bacteria are always in the vegetative stage in the plant tissues. Sometimes there will be deficiencies with respect to the nutrition. Then the vegetative cells get converted into. Now, sourcing allocation is very, very important in the case of plants. Sometimes, it gets transported just to the fruits. What happens to the bacteria that those are present in the carbohydrates and sugars are definitely going on to the some other parts. So they encounter a kind of question mark. So they get converted. So some temporary drying, permanent, not permanent wilting point, but it is temporary drying. At that time, the sap concentration is going to be very important. Plant sap concentration. They are there in the cortex cells and in the phloem and xylem, etc. They may encounter some uncomfortable situation, and during the uncomfortable time, they get changed into endospores. Any other question? Please.
Genus specificity. You are referring to cowpea and tomato. You are referring to cowpea and tomato. Genus-specific endospore. Isn't that natural? Isn't that we naturally expect, sir, that a cowpea endospore will be
endophyte.
Endophyte will be more favorable to cowpea than tomato. I mean to say that isn't that naturally selected? As isn't that
need not be. We have encountered a situation where some of the endophytes derived from the host plant adversely affect the growth of that particular host. We have come to the in one of the researches with brinjal. Brinjal, we got a number of endophytes from brinjal. But some of them were unfavorable to brinjal itself. Okay, but they were good for tomato. This is a condition. This is logical in nature. Like endospore forming, endophytic bacteria which are host-specific in nature, will have more favorable action in. But what is there is chance that the same bacteria would be present in both. Same bacteria, when you take a little bit of soil and then plant two different genus in the same soil, in the same cup, one tomato and one cowpea. Uproot them and then see the microbiome pattern within. There will be a lot of difference. There will be a lot of similarities also. So the same bacteria could be present in host 1 and host 2. In there, the question is something different. You may not be able to differentiate based on the colony morphology. We say this is A, the other one is B. Sometimes A and B will be C, same. Such situations can arise. I just wanted to say that, in my experience, it's better you go with some host derived. There are people who will look forward, look for endophytes isolated from, say, plants with antifungal properties. They think that anything that is present in will also have antifungal property. That is the wrong notion. That's the wrong. There are problems. And, you must be knowing. Okay, the classic example is Taxol. I just forgot about the plant. Taxol brevivitor. I don't know. Taxol is an anti-cancerous drug. Okay, and then there are endophytic fungi isolated from that particular plant, which in culture condition started producing Taxol. The fungi that will be present, that are present in the plants, started producing Taxol in culture. People thought that there is a big revolution and then we can now industrially produce Taxol without cutting the plants. That particular plant is in the Himalayan region. There is wide deforestation with respect to such plants. People thought that this was around 20 years back, but it was not materialized. After some time, the character is lost from the endophyte. Such things do happen. There are more unpredictabilities than predictabilities in such plant microbe encounter reactions. Anything else? Thank you very much for your patient listening. I think I exceeded my time limit, but at times it happens. Thank you very much.
Thank you, sir, very much for the great insight lectures on the endophyte forming bacteria, endospore forming endophytic bacteria, and have a very basic techniques itself for the researchers who had doing in this field. It is very helpful, and also thank you, sir.
Thank you, sir. Now we invite the chairperson to present a memento to Dr. K.N. Anith on behalf of Malabar Plant Protection Society.
Next, our lead talk is by Dr. Jairaj P., the former program coordinator, KVK Kannur, and sir is giving talk on the plant nutrition and plant health management. And Dr. P. Jairaj has completed his graduation and post-graduation from KAU and obtained his doctorate degree from IRI New Delhi. And after joining KAU, he has served a long period as program coordinator at KVK Kannur. During his tenure, he implemented several developmental programs and introduced many innovative practices to his credit. He has also guided many PG as well as PhD students. Dr. Jairaj, after the retirement, presently active in farmer consultancy and solving field level agriculture problems. Sir, I cordially invite you, sir. And sir, due to the time limit, sir, please limit the talk for 20 minutes.
Thank you for your nice introduction. I don't know whether I can complete within 20 minutes because it's actually an academic session. But more oftenly, the academics are not very much interested in the real field conditions. That is the reality is actually occurring in all the research stations. So what I feel is that so the resilience measures in agriculture has to start from soil. And if where we have to start, that is the main question. Here you can see some cross-section of different soil parameters. What actually I feel is that all our technologies will prevail well in matured soil. But where we are having that matured soil, we are having immature or young soils where most of the agriculture field situations are prevailing, and all our technologies are fails to perform in such soils, whereas in the matured soil it will do wonders. So this is the real problem, how we are going to assess the exact field situation with respect to our findings, whether that findings will be suited to that particular situation, then that definitely the farmers will adopt that technology. Otherwise, farmer will reject those technologies. This is actually happening in the field situation. So why this? Only because of the heterogeneity of our field situation. Because in Kerala, we are having 23 different agroecology units. There you can see that the Kaipad, Katamballi, Wayanad situations, Palakkad, Kol, Pokkali, Kuttanad, all are having a heterogeneous soil environment where we are pumping our technologies without doing the multi-locational trial in such a situation. So how we had to place our technologies in those systems, there lies this is the main concern faced by the farmers and development officers, the poor soil health status leading to pest and disease incidents, and the low productivity in majority of the agricultural ecological units. So how we are going to address these issues? So many other social issues are there because the extension is the main agency where our finding has to lead that particular areas of concern. Say the challenges faced by the development offices. Largest unorganized sector and only 5 to 10% of the progressive or educated farmers are there. No vibrant model. More than 40% youth are out of agri sectors. And who are going to take up our technologies for the future or our Vision 2050? So in that context, we had to pump our technologies in such a way that agripreneurship guidance should also be there along with our findings, then only the farmers will adopt it. Then this is the present issue and challenges faced by the real farmers, because the farmers are forced to move from the scientific practices to traditional or organic by the public, finding fault with the modern agriculture without knowing the real facts. Who will address this issue? Whether farmers can address, or researchers can address, or the academicians can address, or the politicians. So this is the real issue, situation prevailing in Kerala. We are having so many technologies, but who are taking these technologies? By the social stigma of the scientific technologies are not worthy. We are going back to the organic or natural farming system in our country. So this type of so many issues are also prevailing in our country. Then coming to the problem cost diagram. The low productivity is the main issue addressed by the farming community. And we are having many programs and many technologies. Whether any of our technologies completely demolished the problems in the farmers' field, we are answerable. So how the extension system is failing. So see, poor soil health, high incidence of pest and disease, many progress, many technologies, still the problem remains same. So how we are going to address this? Because this is actually the resilient agriculture. How we are going to make the agriculture a profitable one to the farming community so that our economy can also be improved. This is just because the wide variation, cropping pattern, nutrient status, management strategies, ensure adequate availability to nutrients and improve soil and ultimately productivity. This has to be in a holistic aspect of how we are going to address this. So these are all the requirement, policy decision with a clear vision, convergence to solve problems, rural self-sufficiency of foods and other items, and specific rural crop production plan. Integrated approach of doing things in all available ecosystems. So where we are lacking, we are still providing technologies for rice, coconut, vegetables, banana. Have you ever developed a technology for a particular ecosystem? Because this is actually a situation of Kerala where we are facing homestead ecosystem, wetland ecosystem, commercial ecosystem, plantation ecosystem, and integrated farming systems. So how we are holistically addressing the ecosystem perspective of pumping our technologies or findings to that particular situation where we are having a eco-friendly technological input. Then, so what we have to do is, for a resilient measures, as a resilient measures, in RBM concept has to be practiced, result-based management. What should be the minimum productivity to achieve in a unit of land, whether it is in homestead or a commercial or a IFS or in a monocrop culture or a plantation. This type of thrust area in different ecosystem has to be highlighted. This is what actually now the farmers are doing in certain locations. And this actually, because here the plant protection scientists or students or researchers are attending this conference, what we lack is that the physical properties, chemical properties, and biological properties, along with the agronomic practices and the tillage operations, definitely provide support to the soil productivity, which in turn produce the crop health status, and that will create better production. At the same time, the climate is also influencing soil as well as the crop health. Whether the influence from the physical, chemical, biological, and the agronomic management practices is more on plants, that is the best climate resilient practice. So in that context, we can do a lot of things, because the NICRA, the basic principle of NICRA, National Innovation of Climate Resilient Agriculture, is combination of available technologies suited to that particular situation to have a minimum level of productivity is ultimate aim in NICRA. So these type of avenues are also there to explore for the future research areas. Then coming to how we can address these things. Knowledge, skill enhancement, business. What we are lacking in agriculture sector is business ideas is lacking. We are having so many technologies, but we are not providing any business ideas to the farming community. See, this is the ultimate income you can achieve from your unit place of land by adopting our technology. We are not providing the business idea to farmers. We are providing the scientific principles and technologies only. Along with the technology, we have to provide the business ideas also to make our agriculture a profitable one. Then, for that context, we can have a protocol approach for different crops. That is, protocol approach for rice, coconut, banana, vegetable, spices. That too with AU based. How much will be the cost of production? How much will be the minimum productivity? What will be the return? Similarly, how we can have the convergence of financial institution, hand-holding the beneficiaries till the end, traceability and the quality of the produce. This type of holistic approach, along with our scientific interventions, will definitely make a change in our agri scenario. So what should be our work plan? We have to start from the management of existing situation, where we have to start the soil health management, crop health management, disease management, IDM, IPM aspects, drought management. Along with that, new planning, how we are going to take up new planting in such situations, and what are the requisites for that things. So these type of interventions also to be carried out. Then coming to the soil health. So you can see three different soil profiles. The extreme left is the typical Kerala profile. The middle one is the Karnataka profile, and the right one is the North India Haryana profile. So you can see how the productivity status of Kerala is mainly because of our heavy rainfall, where the iron aluminum toxicity is more in the upper crust of the soils, and the calcium, magnesium, silicon, potassium, boron deficiencies are more due to the leaching problem. So how we are going to address that? That is mainly because our soils are 1:1 non-reactive clay kaolnate. So any nutrients we added to the soil cannot be hold for a longer time. That's why the split application or precision farming is performing well in Kerala, where we are providing nutrients at a regular interval. So considering the soil ecosystem, how we are placing our technologies suited to our soil. That is ultimately the soil crop management where starts. Then this slide, almost all of us have studied this chart. I am going in depth to this because the 25% there, 25% water, 45% mineral, and 5% organic matter is the ratio. We are actually it's a good soil. Then coming to the minerals, you can see that red circle where the clay, silt, and the long combination. That is, it's having a perfect combination, which is having the productive soil. We call it a productive soil. It's having a perfect combination of sand, silt, clay. And we can see this perfect combination only in the foothills of Kerala. Not in high hills, not in midland laterite, not in the coastal area, only in foothills. That's why our technologies perform well in foothills. Then for other part of the Kerala, we had to do dynamic corrections in our package, which rarely occurs. That's why our technologies are rebounding. Our technologies are performing well in foothills means that, the soil physical and structural characteristics are suited to that particular situation. That's why many of the technologies are not performing well in different areas. So that issue has to be addressed as a researchable issue, especially in Kaipad or Pokkali areas where farmers are telling that we are not adding anything. But actually the eroded nutrients are accumulated in their area, and that is why the crop is growing well. We are not addressing that situation. So every situation is unique, and we have to mold our technology suited to that particular ecosystem. Then coming to the nutrition, because we all may be knowing all this, but as per Kerala context, among major nutrients, potassium, among secondary nutrients, calcium and magnesium, among micronutrients, zinc and boron. The 5 nutrients are creating issues in our production, in crop production system. So along with our nutrient management strategy, ensure that these 5 nutrients are available to that particular crop. Definitely that crop will be a good one, and we can do minimal corrective measures, especially the plant protection measures, if we provide these nutrients in that particular situation. So the nutrition physiology of crops. When a crop is attacked by a piercing or sucking insect, at what stage? Generally, at the flowering stage onwards, the pest attack emerges. What is the reason for the pest attack during the flowering stage of a particular crop? Why it is not in the vegetative phase? So this type of nutrition physiology is another area where to do well-based research to have a nutrition package for the particular crop in study. Then coming to the deficiencies, you can see the deficiency graph is growing at an alarming rate. Out of the 16 elements, 13 elements are now deficient. By 2030, again, it will be going to increase. How we are going to address this issue? So these type of issues are emerging without knowing the knowing our technical drawback in addressing the real issue. So this is also another issue where this plant protection measures, because if we provide nutrients in such a way, PP measures, we can have minimal PP measures. So then coming to why the pests and diseases are more in certain areas. This is mainly because of the nutrient interactions. Because you can see, if nitrogen is more, means the potassium deficiency will be guaranteed. Now, with respect to the nutrient subsidy, major issue due to the price and nutrient subsidy, the farmers are forced to use nitrogenous fertilizers, omitting other nutrients, which creates a nutrient imbalance, and that will issue the stress tolerance mechanism will be broken, and incidence of pests and diseases are coming. So this is the nutrient interaction, the potassium-calcium ratio, how it performs the proper growth of the plant. Then if chlorine is more, because now the trend is that farmers are using the Japan water for irrigating their vegetables and creating issues. So this type of issue, then if P is more, definitely zinc and boron will be deficient. So this is a peculiar situation in Wayanad soils. We can see the alloying of black pepper in Wayanad, where the issue is the high P content in the surface soil. So these type of issues are very much common. Then how to address those things with our technology? So we have a result-based management, target-oriented approach we already studied, and this how we can do this. This cultural management almost all the farmers are doing. Soil health management very rare are using. Nutrition management hardly few farmers are using, and that is creating the reduction in the crop health and ultimate yield. The major problem is the farmers are forced to take more emphasis on disease management and pest management rather than soil health and nutrition management. That is a real situation. So an integrated approach of doing things is absolutely essential to have a better performance in the field. So this is a particular example of post-monsoon alloying of black pepper. This is in Wayanad. You can say so many reasons are there. Foot rot, mealybug, nematodes, nutrient depletion. How we can differentiate these things? This is another challenge. The complex symptoms we are seeing in almost all the crops. Then the next slide you can see. This is also post-monsoon alloying. This is actually the monsters, as you can see, the typical second leaf alloying symptom in black pepper, especially from November onwards. That is mainly due to the moisture stress and potassium deficiency. That is the berry maturity phase, the sourcing relationship from the second leaf, the nutrients will be moving for the berry formation, and the second leaf is showing alloying symptom. And if it is seen in heavy bearing vines only, not in the normal vines. Especially in the Pannur varieties in Wayanad, any field you can see this symptom is common from December onwards, before harvest. So how we are going to address it? This is due to actual nutrition deficiency. But farmers mistook that it is a nematode and they are doing nematode management practices. So how we are going to address the real cause of the symptoms, or what is the real cause and how we are going to manage it. So an integrated approach of addressing all these complexity has to be there. Then we studied along with our Sugandhi project in Wayanad with the pH and soil microbiology. I haven't seen any publication along with the pH and the microbes, because it's actually doing wonders. Because here we collected soil samples from diseased and healthy plants of pepper vines from Wayanad, and we can see the pH difference. In the healthy vines, the pH is almost above 6, whereas the disease it is below 5. Then we studied the presence of Phytophthora in the diseased and healthy plants. In the same plantation, you can see diseased and healthy plants. We took samples from the rhizosphere from the diseased and healthy plants, and we could see that all the diseased plants are having presence of Phytophthora, whereas all the healthy plants are also having presence of Phytophthora, but the vines are not expressing any symptom of foot rot. So how this happens? So the soil pH management in the rooting zone itself arrest the pathogens to create a attack of foot rot in that particular situation. This is an area we have to do further research, especially for a spice crops where the root is having only surface feeder. It's not have a deep rooted plant. So this is highly susceptible. So this type of observation, this has to be taken, carried forward for future research and other things. Then I will show a few photographs how the acidity is going to reflect in our crop. This is the nursery of the paddy field due to severe acidity, how the nursery performs. You can see the symptom of the iron element toxic due to acidity. Similar situation, this is also the acidity symptom. Whether these microbes or other organisms also perform like this in the acidic environment, I think nobody has studied. So how the growth hormonal results will help the growth to stay. So you can see the root deficient, root in growth parameter, the layered roots in paddy field, typical case of acidity. What is a clustered root? You can see the layered root there. So these type of below soil symptoms, we are very rare. This is the Kaipad situation. This is a Kudiru, where at the grain maturity stage, all the plants were fallen down. Then we uprooted a few fellows. We can see that no healthy roots were there in the Kaipad situation. But our technology is that we have to provide 1 ton of lime in Kaipad soil, but nobody is providing ameliorants in Kaipad soil or Bokali soils. So this is a technology, but the farmers are not adopting it. In a social stigma that is 100% natural. So how we can protect the natural ecosystem? We are having the technology symptoms, but how we are going to take up these challenges? This is 100% natural rice, but if you provide 1 ton of lime in those areas, means these issues can be addressed. So these type of field reality, then this is another case, how within 15 days, how the paddy is changing with the ameliorants. You can see 3rd October, within 5 days, see the root development. Then within 5 days, see the foliage coverage. This is really the technology, and this will create a microclimate for all the soil fauna and flora to survive there. And you can see even the earthworm castings are also there in the well-managed plot. This is the photo from Pariyaram Panchayat, which is under northern Midland lateral. What we say that it is a poor fertile soil. This is the situation of, you can see another thing also, in all our wetland ecosystems are located in the Midland laterite area, considered to be very poor in productivity. All our wetlands, double crop or triple crop wetlands are in the Midland lateral. That is our beauty of our nature. So how we are going to protect that wetland ecosystem? Only with our technologies, not with the, what we say that the slogans like natural farming, organic farming. These are the findings and how best we can provide these technologies. You can get a lot of rhizobial bacteria in this situation. Whether have we collected anything from such situation, real field. So these are the way forward potential of that. So this type of, then another thing is that qualitative performance of crops. You can see the leaves are green and grains are golden. This is actually crop health without any pesticides. So how we can make or plan in this situation with our technology? We are having the technology, but this type of performance has to be there in the farmer's field with our continuous support through the extension system, what we lack. So that type of convergence is there with the scientists and extension functionaries. So a few photographs, these are a typical case of potassium deficiency, boron deficiency, potassium deficiency. This is a well-managed plot with 100% fruits that you can never see any abnormal shaped vegetables in that plot. So vegetable farming should be there in such a way that we won't harvest abnormal fruits. that is the quality of that garden. So these type of interventions is highly needed in our commercial ecosystems. Calcium deficiency, boron deficiency, calcium and magnesium deficiency. Then the nutrient requirement. Our common plant is coconut. What we are doing? We are providing simply nitrogen fertilizers again, and ultimately what will happen is this is the related requirement of nutrients for coconut. And if you are not providing in this ratio, the crown will be like this, because the leaf will be in a straight line, there is no flexibility, and even with a single wind, the tendermost leaf will break and it will cause a bud rot disease. So what is the cause of bud rot? We are providing fungicidal treatment. Actually, it should come that the IPM and INM strategy to be adopted in all the ecosystem, and we are still lacking how to promote that system again. These are few examples where in our KVK Kannur had done. This is a banana plantation where we provided a standard operating protocol for 6,000 palms. Hardly 13 palms are infected with pseudostorm borer. This is another situation. This is the bitter gourd. How the nutrients do wonders in cropping system. That is one of the things. And how best the ecosystem is surviving with respect to the beneficial insects also, so that the insects can be controlled by the beneficial insects. Then you can see the crop health management of black bitter gourd, how it performs. This is the plot of the Kariman District Agriculture Farm, where we provide our standard operating technology for the seed production. You can see the shape and of fruits. All fruits are uniform. This is actually our technology can do. So these type of interventions is actually, especially now the farmers, especially the precision farmers, are doing wonders using all the nutrition techniques. These are all, you can see, no abnormal fruits is there. And another thing is that we provided a QR code system for the ready recognition. You can scan mobile and can take the standard operating protocol for all the crops, instead of going through entire package of practices. And the dynamic corrections also we can do in the farmer's field. So these are in a nutshell. Thank you for inviting me. I don't know whether this will suit for the resilient technology. Anyway, our technologies are there. Along with the convergence, so many beneficial microorganisms can be isolated from the healthy cropping situation. So I think the new researchers and PG and PhD students have an eye-opener to address the real situation and how we can explore that situation for the benefit of the farming community. Thank you all. I think within time finished.
Thank you, sir, for the informative lecture. Sir, the nematodes, the fungal pathogens and the soil nutrients all have impact on the root development. So what about the nano fertilizers? Is the foliar application can help the farmers?
No, actually the fertilizer use efficiency, nutrient use efficiency, we will discuss in such a way that for the major nutrients, it is hardly 30 to 40%. For potassium, it can go up to 80%. For the micronutrients, it may go up to 2% only if you are providing in the soil. Then coming to the nano fertilizer means the fertilizer actually the plants are absorbed through ions from the soil. Nano we usually recommend through foliar means. The atomic size is far smaller than the nanoparticle. Then whether the nanoparticle will do positive results in plants, it is still under study, because in our KVK system also we conducted trial for the nano urea with the 2 ml per liter and 4 ml per liter, but the results were not at promising level. But because the nano urea, it contains around 20% nitrogen. That means half a liter bottle contains 20% means 4 gram nitrogen. And the firm is telling that it can replace 45 kg of urea. The urea is having 46% nitrogen, and if the efficiency is 10%, that will be far superior than nano urea. Still under study. So that is a question. I think last KVK annual review workshop, I only put a suggestion to the soil science department to have a study on nanoparticles of urea along with the traditional fertilizers and how it performs. Similarly, with respect to the pipe cultivation in black pepper along with the dead standard and live standard. So these type of issues has again to be studied in a detailed way. I think Dr. Tulasi and Dr. Mosa is in the line to address the performance of nano fertilizers.
Thank you, sir.
Any other questions? Okay.
So on behalf of the organizing committee, we extend our sincere thanks to Dr. Jairaj sir for coming here and delivering a informative talk.
Thank you, sir. Now we invite the chair and co-chair to present a memento to Dr. P. Jairaj on behalf of Malabar Plant Protection Society.
Now we are moving towards the oral presentations of this technical session 3. The first one is T3OP1, Sneha Mohan.
Will the participants please stick on to the time? Okay.
Total time provided for this oral presentation, 5 minutes for presentation and 1 minute for discussion.
Good evening, respected dignitaries and my dear friends. I am Sneha Mohan. Today I am here to present on the topic climate-driven dynamics of finger millet blast in Kerala, implications for resilient plant health management. 2023 has been declared as International Year of Millets by United Nations. As finger millet is one of the major millet, it's nutritionally important and a climate resilient crops. But the finger millet is majorly affected by fungal disease, blast disease. It's a major constraint in the finger millet cultivation, which causes an annual loss of 28 to 36%, and in endemic areas it causes an yield loss of up to 80 to 90%. So, as part of my research, the objective was formulated to assess the distribution of finger millet blast across Kerala and analyze its association with the microclimatic factors, as the disease is
majorly favored by the environmental conditions. So, as part of the research, a field survey was conducted in 10 locations of 6 agroecological units of Kerala, that is AU 3, 8, 11, 15, 16 and 18. And the finger millet leaf samples showing typical blast symptoms were collected and the disease incidence and severity were assessed. The weather parameters during the survey period were recorded and a correlation analysis was performed and the isolation of the pathogen was done from the infected leaf samples. So these are the areas surveyed for the finger millet blast symptoms in Kasaragod, Palakkad, Idukki, Alappuzha and Thiruvananthapuram district. As the result showed that the symptom appeared on all stages of the growth, that is the symptom appeared on the vegetative, flowering and ripening stages of the crop, and the symptom varied from a small brown spot to large spindle shaped lesions which coalesced to give a blighted appearance, and the percent disease incidence was assessed, which observed 20.67% to 56.33%, with the highest at Shantambara region of Idukki district and the lowest at Pullurpediya region of Kasaragod district, and a percent disease index of 17.76 to 52.6% was recorded, with the highest at Shantambara of Idukki and lowest at Mavelikara of Alappuzha district. These are the symptoms of finger millet blast. And isolation of the pathogen was done and it showed a grayish white appeared colonies with the hyalin and septatum hyphae with the conidiophore, the conidia attached to conidiophores, and the conidia were pyriform shaped with the 2 septate and 3-celled conidia and also formed aprosorium. So, the weather parameters were correlated with the disease parameters, and it showed that the maximum temperature showed a negative and significant relation with the disease incidence and disease severity, while the relative humidity showed a positive and significant relation with the disease incidence and disease severity. That is, the maximum temperature reduced the blast development and the relative humidity favored the blast disease development. And correlation, the graphical representation was done with the weather parameters and disease parameters, and the bar graph shows the percent disease incidence and severity, while the line graph shows the weather parameters. As it highly indicate that in Shantambara region, the relative humidity was higher and the maximum temperature was lower, and also the moderate rainfall and the lower minimum temperature was observed. So this region observed with a higher disease incidence and intensity. And while the Pullurpediya region with a higher maximum temperature and lower relative humidity showed a lower disease incidence. So, the result indicate higher disease incidence was observed under the high relative humidity, and a moderate rainfall favored the disease development. Also, lower temperature showed higher disease intensity, and this higher humidity always affect the spore development and conidia formation and aprosorium formation during the spore germination. So, this high humidity always favors the disease development, and a temperature greater than 30 degrees Celsius reduced the sporulation, because water is essential for the sporulation to happen. So, it reduced the sporulation and lesion development, and lower minimum temperature it increased the dew retention, and also the moderate rainfall increased the leaf wetness during the disease development. So, coming to the salient findings, the blast incidence and severity varied across Kerala agroecosystem with climate, and the disease incidence and severity showed a positive correlation with the relative humidity, while a negative correlation was with the maximum temperature. High relative humidity, adequate rainfall and lower minimum temperature favor the blast development in finger development, while the higher maximum temperature suppress the disease progression. While the disease was observed higher in the cooler and high humidic ranges, and these findings highlight the importance of this microclimate based disease forecasting approaches, as this blast disease is developing into new hosts. So, a climate based, microclimatic based disease forecasting and disease management strategies are required for to prevent the spread of the disease. And so, I acknowledge Department of Plant Pathology, College of Agriculture, Vellani, and Kerala Agriculture University for providing infrastructure and funding support for my research. Thank you.
Thank you, Sneha, for completing in the stipulated time. And also, I have only one question to ask. So, this Pyrochlorella is a very host-specific pathogen, and now it's gaining very importance. And can you give which are the adaptations of this pathogen for this changing climate to become an epidemic pathogen?
Ma'am, Pyrochlorella is it's crop specific. That is, Pyrochlorella is affecting mainly it is affecting cereals, but it's also now affecting the ginger. Ginger is the main crop recently affected by this Pyrochlorella. So it's changing its host habitat. It's changing with the climatic conditions are favoring this disease. So it is adapting new host and new regions. So it is spreading to new host regions, and also banana also reported recently with this Pyrochlorella.
So I'm just asking the adaptations, how they can adapt to these climatic conditions, which are the adaptations that this particular fungus is having, which is to our climate.
Spore development.
Just how it become an epidemic in our condition.
The cooler microclimatic conditions here, that is the higher relative humidity and this changing this lower minimum temperature here is giving favorable conditions for this species to adapt. Sir, agronomic conditions, that soil conditions were analyzed. Then fertilizer, variety. Sir, it is mainly local varieties they were cultivating during the survey period. So, a difference. So during the survey period, sir,
Okay, we will move to the next presentation, that is in online. T3OP2, Anton.
Yes, sir.
Are you ready?
Yes, sir. Good evening, dear teachers, scientists, and my dear friends. I am Anne Tom, and I would like to present metagenomic insights into phytoplasma association with tapping panel dryness in Hevea brasiliensis. Next slide. Latex from Hevea brasiliensis is the major source of natural rubber, and the production of natural rubber is directly dependent on the production of latex. Tapping panel dryness syndrome is an important serial killer disease which directly affects the latex production by blocking the latex biosynthesis in the tree. And the etiology remained unresolved since its first report from Brazil during 1887. Objective of the study is to elucidate the role of microbes and physiological parameters in inducing tapping panel dryness syndrome in Hevia brasiliensis. Next slide. Experiments like survey, anatomical investigation, characterization of phloem-limited microbes, bark grafting to test the transmission, and biochemical analysis has been done. Next slide. Survey was conducted at major rubber growing tracts of Kerala for person disease incidence and severity. A severity scale has been developed based on Abraham and his co-workers during 2005. Next slide. Among the major clones cultivated in Kerala, RRII 414 has reported to have more highest incidence and severity in tapping panel dryness syndrome. Next slide. On analyzing different tapping systems, tapping has been done by making a cutting across the bark covering its half the circumference, which is denoted by S2, and D2 and D3 denotes the frequency of tapping. Among these two different tapping systems, high frequency tapping, that is S2D2 tapping, which is done once in 2 days, has shown the highest incidence and severity. Next slide. Highest incidence and severity has found in the oldest trees, which means that tapping panel dryness syndrome, severity and incidence of the disease increases with increase in the age of the tree. Next slide, please. Among the plantations, we also observed a spatial spread of the disease along the rows in the direction of tapping, and these number of rows varied from number 1 to number 14. Moreover, next slide, please. Occurrence of TPD symptoms was also observed in the untapped tree or in the virgin bark, and it is manifested as necrotic bark or absence of latex in the bark. With this indicates the presence of a pathological or a biotic factor in inducing the syndrome. Therefore, next slide. Anatomical investigation of soft bark, which is rich in latex vessels and sieve tubes, has been done. Next slide, please. Tangential longitudinal section of the bark has been stained with oil redo, and it was observed that a dark grayish discoloration and deformed latex vessels was observed in TPD affected tree in comparison to deep red spindle-shaped latex vessels in healthy tree. Next slide, please. Sieve tubes in TPD affected tree, which was stained with Congo red, has shown blockage in the sieve plates. Next slide, please. Which was more prominent in the higher magnification, and this blockage was assumed to be due to the callose deposition, which has a high affinity for Congo red. Next slide, please. On observing the sections under confocal laser scanning micrograph, which was stained with DAPI, fluorescence was observed only in TPD affected bark, and in healthy it was completely devoid, indicating the presence of microbes, especially phytoplasma, in the sieve tubes. Next slide. Which was observed to be in the size range of 400 to 1000 nanometer under scanning electron micrograph. Next slide, please. Therefore, phloem-limited microbes has been characterized using metagenomic analysis of the soft bark region of the tree, both healthy and TPD affected tree. Next slide, please. Next slide, please. Under metagenomic analysis, the major phyla has shown to have no significant difference in healthy and TPD affected tree. Similarly, next slide, please. At genera level also, similar result has been observed, where none of the major genera has shown a statistical significance or difference in both healthy and TPD affected tree. Next slide. Diversity, both alpha diversity and beta diversity, was shown to be more similar in healthy and TPD affected tree, which means there is similar species richness and evenness in healthy and TPD affected tree. This indicates that there exists a core microbiome in Hevia brasiliensis, which is not affected by the tapping panel dryness syndrome. But, next slide. There were few genera which were exclusively present in TPD affected tree, and the most notable one is Phytoplasma with an abundance of 0.015%. Next slide. And on amplifying the 16S rRNA gene of Phytoplasma using the universal primers, amplification was observed only in TPD affected bark. Next slide. And sequence analysis has shown that these sequences of 16S rRNA was more similar to Phytoplasma rice yellow leaf causing Phytoplasma belonging to the group 1 with Acolaplasma as the outgroup. Next slide. On analyzing the expression of 16S rRNA in healthy and TPD affected tree, we found that highest expression was found in partial TPD affected, that is 50% severity affected tree than in comparison to that of total TPD affected tree, indicating the obligate nature of the pathogen. Next slide. Transmission of the syndrome has been tested using grafting the TPD affected patch to that of the healthy tree. And next slide, please. And the grafted tree has shown the absence of latex or latex flow in comparison to that of the healthy tree. Next slide. About 70% of the graft was successful, and all the grafted trees had shown the TPD occurrence, and only 70% of the tree has shown the amplification of 16S rRNA gene of phytoplasma. This indicates the transmission of syndrome from TPD affected tree to that of the healthy tree. Next slide, please. Biochemical analysis has been done for total sugar, reducing sugar, phenol content, peroxidase, and polyphenol oxidase using standard protocols. Next slide, please. Reducing sugar and total sugar were high in TPD affected tree in comparison to that of the healthy tree. Next slide. Phenol content and peroxidase content was also higher in TPD affected tree in comparison to healthy tree, but polyphenol oxidase was higher in healthy tree in comparison to that of the TPD affected tree. Next slide. With this result, we proposed a disease model where we found that though there are some host predisposition factors relating to the stress-inducing factors, involvement of phytoplasma can cause phloem dysfunction and can block the sieve tubes, thereby increasing the sugar content in the TPD affected tree and activating the defense mechanism in the plant, leading to phenol accumulation, peroxidase activity, and thereby causing hypersensitive response, programmed cell death, latex flow disruption, and finally leading to tapping panel dryness syndrome. Next slide. Future prospects for the study include sequence analysis of phytoplasma in different clones and geographic areas, transcriptome analysis of healthy and TPD affected bark to understand the host-pathogen interaction, and development of kits for rapid detection of phytoplasma. Thank you. I take this opportunity to thank my major advisor, Dr. Sainamol Kurian, and Rubber Research Institute of India and Malabar Plant Protection Society for giving me this opportunity to present my presentation. Thank you.
And it is a very nice presentation. So, what about the output, the conclusion you have not mentioned?
Yes, ma'am. Conclusion was mentioned as a disease model, where we conclude that presence of phytoplasma, which is a pathogen, obligate pathogen, blocks the sieve tubes, and these sieve tubes, which provide the latex vessels, photoassimilates, especially sucrose for latex biosynthesis, is completely blocked there. And this blockage and invasion of pathogen activates the defense mechanism in the rubber tree, which is leading to phenol accumulation, peroxidase activity, and hypersensitive response, and is manifested as the symptoms like cracking and typical TPD symptoms, and also due to the lack of photoassimilates for latex biosynthesis, latex is not generated, and which is the ultimate reason for tapping panel dryness syndrome.
Okay. Any other questions?
Next presentation. Thank you, Anton. Next presentation is
Thank you, sir.
T3OP3, Anushree SG.
Every plant you have ever eaten has survived because it could differentiate between a friend and an enemy
with no eyes, no brain, no immune system. My research reveals exactly how Arabidopsis does this at the level of individual genes. Good afternoon, everyone. I am Anushri SJ from Central University of Kerala. My research is the comparative transcriptomics of Arabidopsis thaliana due to beneficial and pathogenic Pseudomonas under the supervision of Dr. Jasmine M. Shah. Plants constantly interact with both beneficial and pathogenic microbes. The transcriptional reprogramming is the key molecular response. We have infected Arabidopsis thaliana, the model plant, with two bacteria with the same genus, that is Pseudomonas fluorescens, PF, which is a plant growth promoting rhizobacterium, and Pseudomonas syringae, which is a plant pathogen. The objectives include the comparative differentially expressed gene analysis of PF versus PS responses, functional enrichment analysis, regulatory network analysis, and comparative defense pathway analysis. Here, the method of infection plays an important role. We have used the flood inoculation method, which reduces the background noise, unlike the syringe infiltration method, which causes wounding. These are the softwares and databases used for the transcriptome analysis. Coming to the results, when the Pseudomonas fluorescens, the beneficial bacterium, met Arabidopsis, the Arabidopsis genes differentially expressed were 228, and for the pathogenic bacterium it was 148. And both the treatments had a common of 50 DEGs, out of which 44 were upregulated and 5 were downregulated, but one particular gene in the chromosome 4 was found to be upregulated due to the beneficial bacterium and downregulated due to the pathogenic bacterium. This could be a potential molecular switch between a mutualistic and pathogenic signaling. And also, coming to the topmost unique upregulated DEGs, when the beneficial bacterium met with Arabidopsis, the major genes which were expressed include BHLH family members, which are involved in iron acquisition. And for the pathogenic bacterium, many signaling genes and barrier proteins were highly upregulated. And for the downregulated genes, a long non-coding RNA for the beneficial bacterium was found to be highly suppressed, but it was non-significantly upregulated in the pathogenic bacterium. This could also be a potential molecular switch between beneficial and pathogenic bacterium. The PS also showed another gene which was highly suppressed, and it showed opposite behavior for the beneficial treatment. These are the top genes which were shared for both the treatments. Chromosome-wise distribution of the total DEGs also revealed clustered and non-random arrangement. Gene ontology for the beneficial interaction shown iron homeostasis activated and ISR-associated priming, while pathogenic interaction revealed stress and immune signaling, which were highly enriched. And the shared geosignatures were defense signaling, which was highly enriched. And this could tell that Arabidopsis uses a conserved microbial response, but it deviates its transcription according to the microbial lifestyle, where the beneficial microbes prime adaptation and pathogens trigger defense acylation. KEGG analysis revealed for the pathogenic treatment, the highly enriched KEGG pathways involved the monoterpenoid biosynthesis, plant hormone signal transduction, and terpene backbone biosynthesis. For the beneficial bacterium, the highly downregulated KEGG pathways included the MAPK signaling pathway, protein processing in the ER, and Q10, suberin, and VAX biosynthesis. And who regulates these? The transcription factors. The transcription factor analysis revealed 28 DEGs which were upregulated due to the beneficial bacterium, and 13 DEGs which were differentially expressed due to the pathogenic bacterium. And the transcription factor dominates the BHLH family for the beneficial bacterium, as well as ethylene responsive genes, and it was mainly involved in ISR and iron acquisition, while for the pathogenic bacterium, major genes which were transcription factors which were downregulated were the developmental genes. And also there was one long non-coding RNA which I said earlier. It was highly downregulated due to the beneficial bacterium, but it showed non-significant enrichment for the pathogenic bacterium. It could be a potential molecular marker for the beneficial colonization. And comparing the defense for both the treatments, for the beneficial bacterium, the defense was primed but not activated. But for the pathogenic treatment, there was active defense seen and targeted gene cluster repression was observed. For example, the marker gene for SAR, that is DIR1, was highly suppressed due to the beneficial treatment, which shows that the plant is suppressing its own immune system to allow colonization of the beneficial bacterium. Coming to the conclusions, PF induces broader reprogramming than PS, and 50 common DEGs were common for both the treatments. PF, that is the beneficial bacterium, activates iron homeostasis plus transcription factor reprogramming, while PS activates terpenoid defense plus cell wall reinforcement. Major genes which were differentially expressed for beneficial bacterium include the BH-LH family, and a long non-coding RNA in the beneficial bacterium could be proposed as a molecular marker for beneficial interaction. And in the future, applications include the identification of candidate genes for CRISPR-based disease resistance engineering and PGPR-based biocontrol optimization. Thank you.
Okay, thank you, Anishri. And you have done with the gene expression studies, or anything with the that's the pathways or anything you have studied, or there is only the discussion part only, the pathways and thing. So with the gene expression only you have done. The others, sir? Okay, you have not given any results on this pathway analysis on that.
I have just shown the conclusion of
you have just concluded that, but you have not.
I have not shown the pathways exactly.
You have done this.
And these are also the conclusion of gene ontology analysis. I have so many maps which I cannot include due to the time shortage.
Okay. Any questions? Okay, thank you, Anishri.
Thank you, Anishri. Before tea break, one more session, that is T3OP4, Tirumala Sami.
Yes, hello, is it audible?
Yeah, audible. 1 minute.
Oh.
Yeah, presenting.
Yeah.
Slides are visible?
Yes, visible.
Yeah, okay. Please, sir.
Thank you.
One minute.
Yeah.
Yeah, sir, please.
Can you enlarge that? Can you focus?
Yeah.
Now
it's visible?
Yeah, it's visible.
No, I think the file is not.
It not downloaded properly, maybe?
Yeah.
Is it downloaded?
Because due to some pictures, we could not able to make a big screen actually.
No, it's mostly pictures only, actually. Photographs.
Yeah. No, is it visible now?
Yeah, for me it's visible.
Yeah. Okay. Yeah, please, sir.
Okay, thank you. Respected Chair, Co-Chair, distinguished scientists, and dear participants, very good afternoon to all. I am presenting the host range, resistance screening and defense strains against the bacterial wilt and shoot and fruit borer in Solanum species. Next slide, please. India is having huge genetic diversity in eggplants next to China, and we are having wild species also. At National Gene Bank at NPPGR New Delhi, we have 3,786 germplasm accessions belonging to 21 Solanum species. At our medium-term storage regional station Thrissur, we have 285 germplasm working cultures belonging to the 17 species. This, next slide, please. In that, the three major species are cultivated type. We are Solanum melangina, which is normally called a brinjal eggplant. Another one is the Solanum ethiopicum, which is an Ethiopian eggplant, which is grown in our northeastern states also, and it is consumed. And in another one is the macrocarpon, the Govoma eggplant, which is cultivated in hill areas as well as in some of the plain areas, which is also edible, but less consumptions. Besides these three species, the species of Solanum tarvum fruits and also leaves of Solanum americanum and Solanum trilobatum are also edible. Next slide, please. The major biotic stresses of eggplants and its wild species are fruit and shoot borer, which is the national importance also, and another one is the bacterial wilt. Next slide. The bacterial wilt, which is caused by the Ralstonea solanacearum, which is recently renamed as Ralstonea pseudosolanacearum, which exclusively infects the Solanum species, which belongs to the race 1 and Biovar 3. It is a lethal disease mainly in the high rainfall area of coastal Goa, Karnataka, Kerala, Tamil Nadu, Andhra Pradesh, Orissa, West Bengal, and northeastern states. Wherever high rainfall followed by heavy temperature is there, the disease is more severe because it needs the pathogen to invade and also cause disease. So high rainfall and high temperature is essential. So the yield doses vary from 75% and sometimes up to the 100% based on the disease incidence on severity. The pathogen is difficult to control because of its soil inhabitant nature and it's having wide host range. So we go, next slide please. Next slide. So we have screened the 182 accessions what we are having at our medium-term storage belonging to 17 species collected throughout the India, mainly from Kerala, southern states, mainly Kerala, Karnataka, Tamil Nadu, Andhra Pradesh, as well as the Andaman Nicobar, which is very potential for our materials at our National Gene Bank. We have 17 species we screened for that bacterial wilt diseases. Next slide. By the artificial inoculation technique, where 30 days transplanted material are injured by using the knife at 4 sites, and the inoculum at a concentration of 7 into 10 per 8 colony forming unit per ml were used for making the infection. The symptoms appeared within a week or up to the stage of harvest, and we have recorded at a weekly interval. The cumulative data were used for analysis, which is a 3 years data I am representing only in the nutshell form. Next slide, please. Where we have out of 182 accessions, 16 accessions where we found resistance, and 29 accessions were totally succumbed to the diseases. In that, the Solanum melange, there were 12 accessions were resistant. Many accessions were also reported from the IAHR as well as IVR, and also from many state agricultural universities. These are the potential for breeding purpose we can use, or we can directly we can use it for the backcross method, and we can improve the quality of the resistance, and with high yielding we can release. In the another one is Solanum ethiopicum. We found 2 accessions. Though it is not edible, we can use this accession as a rootstock for making this grafting for the high yielding variety. This Ethiopium is highly compatible. Up to 60 to 80% are success for the grafting. And another one is Solanum incanum, which is also resistant to that bacterial wilt. What we found that morphologically many plants are very thorny and which are very drought resistant, that most of the species are susceptible to that of the bacterial wilt, like species of Solanum viarum, Solanum lasiocarpum, Solanum virginiatum are highly susceptible to that of the bacterial wilt. What we have predicted it may be resistant, but in reality it was not found. And in the background work we are doing for decoding of elicitor receptor in that, and also pre-breeding by two-way and three-way crosses by incorporating resistant in the bacterial wilt with the cultivar type. And next slide please. Coming to the other part, the fruit and shoot borer resistant, which is nationally important. This infestation caused very severely at the post-rainy season. So we have planted all the 182 accessions in the October, November, and we have maintained up to February, March. So we found that 5 species, Solanum viarum, Solanum cymbrifolium, Solanum exermatum, Solanum elongifolium, and capsicoides, which are totally free from the infestation. Neither shoot infestation nor fruit infestation was found in these 5 species. In the 2 species, Solanum violaceum and also Solanum virginianum, we did not find any shoot infestation, whereas fruit infestation was found in these 2 species. Next slide, please. This is the 3 species which we were found newly that Lucinodes arbonalis, that is fruit and shoot borer of eggplant, infest these 3 species, which is the first report to the world. That is a trilobatum, Solanum trilobatum, infested by the Lucinodes arbonalis. We are seeing in the pictures. This is the borehole, the excreta, and the larvae inside. This single larva where we found in the fruits of this trilobatum. And shoot also infested by the Lucinodes arbonalis. This is the another one, Solanum violaceum. This is another species which is entirely thorny, and some are non-thorny species also there. And both the species are infested by the Lucinodes arbonalis. The highest number of fruits infested by single plant we found in this previous slide, please. Previous slide. Highest number of fruits were infested in the Solanum violaceum if you will take into the number of fruits per plant. This is the one. Another one is the Solanum lasiocorpum, which is highly hairy in nature, both the and also thorny. Both the fruits and leaves were covered by the thick hairs. In spite of these thick hairs, the larval feeding were we observed in the Solanum lasiocorpum, which is also collected, one species collected from the Kerala and another species from the Andaman Nicobar Island. These also infested by the Lucinodes arbonalis. Next slide, please. Besides, there are thorny species which are hardy species like Solanum virginium also susceptible, which were infested by that, and Solanum americanum and Solanum nigrum, which is also infested. Where in this case, the small number of fruits,
can you conclude this presentation to give a conclusion to it?
Yes. This is the last slide. Previous slide, please. This is it makes the cluster and it fends. The remaining species like tarvum and other eggplants were already reported. Next slide, please. These 4 species of the 5 species like Solanum exermatum, Solanum cymbrifolium, and capsicoides, elongifolium, and viarum were totally free from this, and there were no infestation of this Lucinodes arbonalis. So, next slide, please. So we found that some of this is last. Some of the polyphenols and phenols and pericarp thickness we were found as the component for resistance in the Lucinodes arbonalis. And we are combining this, we are combining this Lucinodes arbonalis resistant gene plasm along with the bacterial wilt by using the 3-way or 2-way crosses for incorporating resistance, and combined resistance programs are under the way for development of both the pathogen and as well as the pest of that eggplant. Thank you. Next slide, please. Thank you so much.
We are in short of time, actually. So, dear participants, be in time because we are the program is already delayed for 1 hour, and now we have a small tea break for 5 minutes. Yeah.
Tirumala Swami, it is a nice presentation. And what about Solanum thoruum? And it is widely used by many farmers for grafting
Yeah, yeah.
But you have
Yeah.
not mentioned it is not promising.
Tarvum is resistant to bacterial wilt. No, it is sure. There is no doubt. But the grafting success is 60% plus only. And there are some problems associated. Means if the grafted material fruits are having some bitterness, something, the dislike factors are associated. So there are some research components is always going on. But tarum also good material. We can use it as a rootstock for grafting. Besides, we have found on more species like Ethiopium eggplant, which is also easily multipliable, and there is no bitterness in that. So this research is under the way.
Okay. Okay. Thank you.
Yes. Thank you. Thank you.
Thank you, participants. Now it's time for a short tea break. The presentations will be resumed after the break at 5:10 p.m. As we are running out of time, I request everyone to reassemble sharply at 5:10 p.m.
I call upon Nandita KV on the topic identification and characterization of fungus, viruses affecting marigold, T3OP6.
Good evening everyone, I am Nandita KV and today I am here to present on the topic identification and characterization of viruses affecting marigold, Tagetes species in Kerala. Good evening everyone, I am Nandita KV and today I am here to present on the topic identification and characterization of viruses affecting marigold, Tagetes species in Kerala. Marigold is an important ornamental flowering crop which belongs to the family Asteraceae. It is mainly cultivated in India for religious, cultural and commercial purposes. Many farmers are increasingly adopting marigold cultivation due to its high profitability and short duration. However, there are many constraints in its production, among which viral diseases are a major concern. Moreover, marigold acts as an excellent trap crop, helping reduce pest incidence in nearby crops. So when such marigold plants becomes infected with the virus, it helps in transmission of the virus to other economically important crops. Hence, the present study was undertaken to identify and characterize the viruses associated with marigold. Next slide. Coming to materials and methods, a purposive sampling survey was conducted in marigold growing regions of AU 10, 22 and 23 of Palakkad district AU6, 10 of Thrissur district and AU6 of Malappuram district. Disease incidence and disease severity was calculated for all the fields. Different types of symptoms were collected and maintained in local lesion host Chenopodium amaranthicolor. Morphological characterization of the virus was carried out by using transmission electron microscopy. Serological detection of the virus was carried out by ELISA, and molecular detection of the virus was carried out, followed by in silico analysis. Transmission studies and host strain studies were also conducted. These are the different symptoms observed during the survey, which included green mottling, mosaic, leaf curling, purpling, puckering, shoestring symptom, chlorosis, and leaf curling. All the samples were mechanically inoculated into Chenopodium amaranthicolor by mechanical transmission. Next slide. Coming to results and discussion, the disease incidence ranged from 2.5 to 45%, and the disease severity ranged from 0.03 to 39.6%. The major symptoms observed during the survey were mosaic, mottling, leaf curling, and purpling of leaves. 2 samples were finally maintained in insect-proof net house by mechanical transmission, and the samples produced local lesions 5 to 7 days post-inoculation. These are the 2 samples maintained in insect-proof net house, which produced local lesions in Chenopodium, and the samples were then sent for electron microscopic studies to IARI New Delhi, and the electron micrograph sample shows that for the first sample, the electron micrograph shows flexuous filamentous rod-shaped particles which are indicative of potty virus, and in the case of second sample, the particles showed isometric particles of 20 nanometer which are indicative of cucumber mosaic virus. Next slide. 2 samples tested positive for CMV in ELISA, and 1 sample tested positive for potty virus. RT-PCR with universal degenerate primers MJ1 and MJ2 resulted in amplification at 300 base pair. The samples were then sent for sequencing. The obtained nucleotide sequence was analyzed by using NCBI BLAST. The isolate showed maximum nucleotide identity with Zucchini tigre mosaic virus 97.69%, and it also showed similarity to papaya ring spot virus 95.36%. None of the samples tested positive for tobacco streak virus, tomato spotted wilt virus, Faba virus, and marigold cryptic virus. This is the in silico analysis. Phylogenetic tree was constructed using Megatwell software by comparing different potty virus isolate, and our isolate clustered with papaya ring spot virus, indicating that the potty virus isolate is most probably papaya ring spot virus, and the amino acid sequence was predicted using XPressy translate tool, and the sequence was compared using BioEdit software, and the sequence identity metrics revealed that the sample mostly was identical to papaya ring spot virus. Next slide. Looking into transmission studies in detail, Chenopodium amaranthicolor was inoculated at 8-leaf stage using 0.01 molar potassium phosphate buffer pH 7 at 1:10 ratio. Small blotchy lesions appeared 5 days post inoculation for cucumber mosaic virus and 7 days post inoculation in the case of potty virus. You can see the disease progressing from the 5th day to 10th day in the case of cucumber mosaic virus, and when inoculated on marigold, the symptoms were observed 18 to 20 days post inoculation. In the case of second sample, the disease progression started from 7th day, and you can see the progression till 10th day, and in marigold plants, the symptom developed on 20th day post inoculation. Next slide. This is the symptom expression of CMV in different host plants. Vein banding was observed in cucumber, green mottling in pumpkin, chlorotic spots in ash gourd, vein banding and chlorosis in cowpea, leaf curling in chili, vein banding in Cinderella nodiflora. In the case of potty virus, similarly, vein banding was observed in cucumber, chlorotic spots in pumpkin, leaf curling in blackgram, vein banding in cowpea, leaf curling and vein banding in watermelon was observed. Next slide. This table indicates the host strain studies of cucumber mosaic virus. Next slide. Similarly, this indicates the host strain studies in detail of potty virus. Next. Finally, we identified that 2 viruses we got were CMV and PRSV, which are aphid transmitted. So aphid mediated vector transmission was carried out. Aphids were collected and reared in polyhouse, and a pre-acquisition starvation period of 2 hours was given for the virus. And finally, they were fed with infected samples and were released on healthy plants, and the symptom expression was obtained 18 to 20 days post inoculation in the case of CMV and 20 to 22 days post inoculation in the case of potty virus. Next slide. So in conclusion, the present study identified cucumber mosaic virus and potty virus as a major virus affecting marigold in Kerala. So this is extremely important in the case of Kerala because during the festival season, marigold is widely cultivated in Kerala. So this becomes a significant study. Thank you.
Okay, thank you, Nandita. You have done a good job. And you have seen the associated vectors and all. And you have identified 2 viruses. And is it occurring as complex form or is separated in different plants?
CMV and potty. No, ma'am, in different plants.
Plants, there is no complex, is there?
No, ma'am.
Have you tried in the re-inoculation, any combined infection, is anything observed in that? Or when you are re-inoculated it only?
Yes, ma'am.
So in that case, you have inoculated separately or any complex, both the viruses
No, ma'am.
simultaneously?
No, ma'am, I have inoculated separately only.
Separate.
Yes, ma'am.
Okay.
The next oral presenter is T3OP7, Shaili C.
Good evening, I'm Shaili. I'm presenting on the topic radiosensitivity of Karuppu Kavani, a medicinal rice land race to gamma rays. So, coming to the introduction, paddy land races are native varieties of rice that are indigenous to particular areas where they are cultivated for a long era and they have evolved many good qualities due to the natural selection or by farmers-driven selection. And I have selected Karuppu Kavani for my study, and it is a land rice from Tamil Nadu, and the seeds and grains are black in color. It is an antioxidant-rich superfood, and this is a grain, and it improves heart health by lowering LDL cholesterol. It manages blood sugar for diabetes due to a low glycemic index, and it aids in digestion, and it lowers inflammation and promoting weight loss. And I have used gamma rays, which is a physical mutagen, widely used for creating variation among different crop plants. So coming to the objectives, to study the effect of gamma rays on germination and seedling growth of paddy land race, Karuppu Kavani, to find out the LD50 value, to study the different types of mutations that occurred in the particular variety. And materials and methods are, I have used Karuppu Kavani seeds with 12 to 13% moisture and irradiated the seeds from, with gamma rays from IIHR Bengaluru, and the germination study was conducted in the laboratory with Petri plates, and M1 generation was grown using irradiated seeds along with the control seeds. Then different morphological features were studied. Statistical analysis with Graves software was done, and macro mutations also observed. So coming to the results, due to the increase in gamma ray doses, I have observed seedling growth reduction in Karuppu Kavani related to control. And I have got LD50 value of 300.561. And these are the different morphological features that I have studied. And I have got a gradual reduction with the increase in gamma ray doses. And this is the macro mutation that I have got from the nursery seedlings. And coming to the conclusion, gamma ray irradiation to paddy seeds has the potential to induce mutation, and LD50 value helps to identify the dose of gamma ray at which the seeds are mutated without much lethality, and different types of mutations can be visualized in M1 generation. So in future, we can use this mutated variety for the further plant breeding studies for getting beneficial varieties. Thank you.
Okay, thank you, Shaili. And how many generations you have tried for taking?
Actually, I have done 2 generations. 2 generations, but only I have presented one generation.
Okay. Is it sufficient for taking this, the mutated is?
No.
I think
no,
it's
it
very
is a
less,
primitive study.
2 years
It
means
is only a primitive study.
And how far it is sustained this, when mutated genes are there, is it a chance to recover the mutation changes on further?
Actually, I have done only 2 generations. I couldn't say I have to answer for this question.
Okay, thank you, Shaili.
Thank you.
Next oral presentation is T3OP8, Minu Susan John. T3OP8.
Good evening to one and all. I am Meenu Susan John from the Department of Entomology, College of Agriculture, Padnamad, here to present on the topic Host Range Expansion of Humerus vestitus, first host report on spider lily. Coming to the introduction, syrphids are also called hoverflies or flower flies, which account for about 6,200 species which has been reported worldwide, which will be famous for its role in pollination and biocontrol. An exception has been seen in the tribe Meradontini, where it is act as a phytophagous one, in that the genus Humerus will be included, which act as a bulb crop pest. It is known for its importance in phytosanitary as well as quarantine importance. The genus Humerus is less studied and underexplored in India for its host association as well as for its taxonomy. Thereby, we introduce our objective, survey and collection of the bulb flies, which is infesting the bulb crops in Delhi, and identification and redescription of the bulb flies, identification of the host plant, which is associated with the predominant species of the bulb fly species. Coming to the materials and methods, field surveys has been carried out in and around Delhi, in nurseries, gardens, resorts, and also in IRA campus for the collection of the species. From the Sundar nursery in Delhi, we have collected the Humerus larvae and we have reared it in a transparent jars, and after a period of about 15 to 20 days, the adult has been collected. And after that, it has been preserved via both dry and wet preservation. And further processing of the specimen is also being carried out for the identification of the species. For that, Thomson 1999 key and description has been used. For species level confirmation, we have used a Smith et al. 2017 key. And photos has been taken using Nikon Z6 full frame mirrorless camera body, which has been attached to the microscope, which is given in the figure. And redescription of the collected species is also being done. For confirmation of the species level, we have also carried out the male genitalia dissection tube. Coming to the comparative morphology, in the oscillate triangle in figure 1, it will be having a tuft of hairs is also been present, an important character for the genus Humerus. And in the case of the figure 4, an scutellum structure is also an important character for finding the species. Coming to the antenna, which is aristate in figure 5, the fossetae or sac-like structure is also an important character for differentiating the species. In the case of figure 8, the lunules will be present on second and fourth tergite, which is an important character, which is having a pale golden polynosity, which is also being present. In the figure 9, the shape of the male sterniphore is also an important character for differentiating the species. In the case of the genitalia, the epandrium and the hypandrium structures will also determine the species level. Come to the results and discussion, the syrphid fly, which has been emerged from the spider lily, has been identified as Humerus vestitus, and this is the first host record of this Humerus vestitus from Delhi in spider lily. In the figure 1a, this is a spider lily plant in a non-flowering season, and in the flowering season, it has been depicted in B. Come to the symptoms, we can see that in the C, the infected bulbs will be having a short boreholes will be there, which has been created by the larva, and after that decaying of the specific root portion is also been observed. Finally, which will be rotting of the bulb will happen, and finally the wilting of the plant is seen. This is the illustrate morphologic description of the species. In the dorsal habitat, we can see that in the abdomen, 3 lunules will be present. Pale, yellow to white polynosity like lunules or maculae is present, which is an important character for identification of the species. And 2 longitudinal stripe is also been present on the thoracic part too. While coming to the eye part, it is a holoptic for male and dichoptic for female. In the case of antenna in D, it is almost reddish brown and it droops down and then it will become parallel on lower part. Coming to the figure for H, we can see that the male basitarsus is having a triangular depression with a hairy part is a very important character for differentiation of the humerus. And in the figure E, the male sternite-4, which is having a V-shaped depression, is also an important one. Coming to the Humerus vestitus, it is the first orental record in tomato and banana, which has been reported by Anuj and co-workers in 2020. Other pre-record include potato tubers, watermelon, grapes, and other rotten stems of pawpaw. As it is an afrotropical species, it has been introduced into India via the commercial trade, which risk the pest spread and warrant the need for biosecurity and phytosanitary interventions. Come to the salient findings, the first record of the Humerus vestitus in spider lily and a host range expansion has also been studied. And coming to the identification character, the male hind basitarsus and sterna is also an important one, which having a wide host adaptability based upon the diverse cropping systems in India, which raises a biosecurity concerns for pest dissemination. Come to the future line of work, larval taxonomy, host preferences and the new host associations need to be found out, and distribution and population dynamics. Also, a detailed survey of the export at the trade centers and seaport need to be carried out for further study. I acknowledge all the mentors and other institutions for guidance and support, and thank you.
Meenu, it is a nice presentation. Then what about the economic loss caused by this particular pest?
Ma'am, it was my MSc, when I was working in MSc, it was this genus only I have worked out. In that, it is having a wide range of host ranges there. About 80 to 85% of loss is also being caused by this pest.
In which crop?
In ginger, I have been observed.
The same
from South India.
Same species.
Not same species, ma'am. It is Humerus figurans. The mostly it will be causing up to a loss of about 80% has been caused in Kasaragod, ma'am. During my research work, I have been worked out.
What
The
about
species can also cause the loss up to 70 to 80% as if no proper management is carried out, ma'am.
in ginger, other maggots also there. How will you differentiate?
Based upon the siphon tube which is present in the larvae, ma'am. And while during the rearing time also in ginger, first the other larvae or first rotting will be happening for the ginger rhizomes, and after a period of 7 to 8 days this humerus larvae will be seen in that, and then it will be causing the complete rotting of the larvae rhizome is being observed.
But in the
this is the another this species has not been reported from Kerala, ma'am. It has been reported in Delhi.
Okay.
Yeah, ma'am. The chances of spread to like Kerala, Karnataka is also more due to its wide host range adaptation, ma'am. The chances are more.
The next oral presentation is T3 OP10 Madhuji. Madhu, are you there? Online?
Yes, sir.
You can start your presentation now.
Sir, am I audible, sir?
Yeah, you are audible. You can start now.
Good afternoon, distinguished scientists and fellow researchers. I am Madhuje from Division of Anthropology, ICR IRI, New Delhi. Today I am here to present my part of PhD research for genomic characterization and population structure of Xanthomonas species causing bacterial leaf spot in tomato. Next slide, please. Tomato is one of the most important cultivated times. Its production is severely affected by disease caused by several species of Xanthomonas, which include Xanthomonas juscatoria, Xanthomonas vescatoria, Xanthomonas perforans, and Xanthomonas gordoneri. The production of small water-soaked lesions on leaves, stems, and fruit, which totally resulting in severe yield loss and quality losses. Especially this disease is known to be highly severe in warm and humid conditions. Earlier, this was as a minor disease, but recently, due to its and aggressiveness, now it has become a major production constraint in tomato production. So, therefore, my research mainly focuses on understanding the genetic diversity and population of Xanthomonas species associated with this disease. The major objectives include to survey, isolate, characterize, and document Xanthomonas species associated with leaf spot tomato. The population structure of Xanthomonas species. So, next slide, please.
Madhu, there is
Slide, please.
You are not clear.
Sir, my voice is not audible. Hello, sir. Hello.
Madhu, is it audible?
Hello, sir. Hello.
Your network is.
Yes, sir.
Network issue. You are not clear here.
Sir, now I am audible, sir.
Yeah, now it's clear.
Sir, can I continue, sir?
Yeah, please continue.
Sir, next slide, please, sir. Next slide, please. Coming to material methods, for this study, the extensive field surveys were conducted across major tomato growing regions, which includes Karnataka, Andhra Pradesh, Himachal Pradesh, Assam, Meghalaya, and Uttar Pradesh. The diseased tomato samples, which are showing the typical symptoms of bacterial leaf spot, were collected from different climatic zones. For pathogen isolation, we have standardized two methods. One is lesion cut method, another one is lesion crush method, where isolation and purification was carried out on each glucose calcium carbonate agar medium. For pathogenicity testing, we have used Pusa ruby with the standard bacterial concentration of 10 power 8 cells per ml. And for molecular characterization, the genomic DNA was isolated and subjected for sequencing of 16S rRNA gene. Next slide, please. Coming to results and discussion, where these are the collection sites which were used to capture diversity of Xanthomonas species from different agroclimatic zones and different locations. In right side, it indicates that these are some of the representative field photographs I have taken during my course of survey. Sir, next slide, please. These are the set of data I have collected during my survey, which include disease incidence, variety grown, previous crop, and severity of the symptoms observed. Next slide, please. All over survey, among surveyed regions, the severe disease was particularly observed in Karnataka and Andhra Pradesh region, majorly Royalpad region of Andhra Pradesh, which is accounting around 95%, and lowest was recorded in DOT of Himachal Pradesh. Sir, next slide, please. For isolation and characterization, where we have followed two methods, that is lesion crush method and lesion cut method. Among both, lesion cut method is known to be predominant and superior compared to lesion crush method, where the chances of getting single cell colonies and chances of getting contamination is very low in lesion cut method. So these are some of the representative isolates which are showing the typical symptoms of Xanthomonas, which include yellow, slimy, shining, elevated, dome-shaped colonies with the even margins. Next slide, please. For pathogen testing, it was known that most of the isolates produced visible symptoms within 5 days of inoculation, initially characterized by that symptoms were produced within 5 days and started with production of small water-soaked leaf. Next slide, please. So to assess the population structure of Xanthomonas species, that is to analyze the genetic diversity and fingerprinting, we have employed box PCR fingerprinting. So these fingerprinting patterns revealed that variability among the isolates collected from different regions. So after removal of all the duplicates, I have got 20 isolates which were later subjected for 16S rRNA sequencing. So however, the species level differentiation cannot be done by using 16S. So next slide, please. Sir, next. Therefore, MLST analysis by using 10 housekeeping genes, these are the set of genes we'll be using for further analysis for identification of species level and population structure analysis. Sir, next slide, please. So this is the overall workflow of the current study. Initially started with survey and collection, followed by isolation and purification, and fingerprinting and 16S RNA. For further species level differentiation, we will be going for MLST by using the 10 set of housekeeping genes. Next slide, please. Coming to salient findings include, through genetic fingerprinting, it was observed that 20 unique Xanthomonas isolates was obtained. From pathogenicity testing, it was confirmed that most of the isolates produce symptoms within 5 days of inoculation. For 16S rRNA gene sequencing, it was confirmed that all isolates collected were known to be Xanthomonas. And for species level differentiation, we will be going for using MLST. Next slide, please. Coming to overall take-home message from my presentation is that this pathogen in the recent times become a major concern in the tomato growing regions because of its spread and rapidly has shown high diversity with the more aggressive strains. Therefore, studying its genomic characterization and population structure is important to identify the predominant strains or understand how the pathogen evolves under the changing climatic conditions or support the development of climate resilient disease management strategies. I sincerely acknowledge ICR IRI, my advisor Dr. Andy Kumar sir, and Lab 23 colleagues and all collaborators for their valuable support and guidance throughout this work. I also acknowledge Malabar Plant Protection Society for providing platform to present my work. Thank you.
Okay, thank you. And I have done a past survey in the initial period from the very cooler area to the Andhra and other area. And you have isolated about 20 related species.
Ma'am, actually I isolated around 56, ma'am. but after doing box fisher analysis, I have got around 20, ma'am. Like the banding patterns of isolates collected from one location, they are matching with banding patterns of other locations, ma'am.
So you have not taken
So-
from the other area, you have in the initial stage you have said
Yes,
that.
ma'am. Yes, ma'am. So we will be removing all the duplicates. After that, we will be going for population structure analysis now by using MLST.
Okay. I think, is there any relevance in this climatic occurrence or the related species occurrence, or its intensity in the disease?
Yes, ma'am. Yes, ma'am. Ma'am, in the current 10 years, like initially it was a minor disease, ma'am. Minor disease, and it was like a cosmetic only, ma'am. Like in recent 5 years, recently 1 month ago only, some current study has been done from IRA itself, where they have reported one species of Xanthomonas, that is Xanthomonas equatoria, which is known to be predominantly present in South India, ma'am, around like Andhra Pradesh region, ma'am.
Okay, okay, thank you.
Thank you, ma'am.
Moving on to the next presentation, T3OP11, Shiva, is joining online.
Yes, sir. Am I audible, sir?
Yes, yes, you can present.
Good evening to one and all present here. Myself, Shiva M. from Department of Plant Pathology, TNI Coimbatore. I am here to make a oral presentation on epidemiology of cocoa boing disease and its association with crown mealybug in sugarcane. Next slide, sir. With that introduction, the cocoa boing disease, also known as twisted top disease, which is caused by Fusarium species complex, is a re-emerging disease in both tropical and subtropical regions of India, especially in the state of Tamil Nadu. Previously, the disease was considered as a minor concern, but due to the rapid epidemics, this disease was considered as a major concern, even worse than red rot disease. It causes nearly 40 to 60% of yield loss in sugarcane cultivation. And the term cocoa boing is a Javanese term, which indicating the malformed and disrupted top. With that, recently, the invasive pest crown mealybug, which is called as Phenacoccus saccharifoli green, which get associated for the first time in India, in Tamil Nadu in 2022, and it leads to the aggravation of the disease severity. With that, we have framed the two objectives, and the first objective to study the role of crown mealybug association in the development of cocoa boing disease, and the second objective was to study the epidemiology, especially the disease predicting models for the cocoa boing disease complex with the crown mealybug in sugarcane. So coming to the materials and method, we have collected the samples all over the Tamil Nadu in 16 locations, and with the collection, we have started with the morphological and molecular characterization of crown mealybug. In the morphological characterization, we did the slide mounting and FESM analysis. In case of molecular characterization, we use the two housekeeping genes such as cytochrome oxidase 1 and D2, D3 regions of 28S rRNA. Followed by, we have confirmed the association of PP pathogen, that is Fusarium pathogen, which got associated with the crown mealybug at the in vitro level. Followed by, we have did the pathogenicity, morphological and molecular characterization of Fusarium species, which got associated with the P. saccharifoli. And then followed by, we did a molecular confirmation also, where we could see the fungal spore got adhered with the powdery wax of P. saccharifoli. And further, we confirmed the same analysis and the GCMS profiling of powdery wax to confirm their association. And finally, we did the glasses condition also to show the association between the crown mealybug and the cocoa boing pathogen. And with the second objective, we did the correlation and prediction for the cocoa boing disease incidence using statistical and machine learning based models. Next slide, please, sir. Coming to the results and discussion, and here you can see the exact infected field where you can see the combined infection of cocoa boing and crown mealybug in the field condition. And you can see some association of ant also, which is showing a tropobiotic association. And even a single ovary sac will lead to 700 eggs, which leads to the rapid multiplication. Next slide, please. And coming to the morphological and molecular characterization, in case of morphological characterization, you can see the exact typical characters, 8-segmented antenna, which is a key character of Phenacoccus saccharifoli. It was confirmed in both the morphological characterization using slide mounting and FESM. Further molecular characterization were done with the two set of gene, that is cytochrome oxidase 1 and 28S RNA, where you can see 680 base pair have been amplified in case of COI and 760 base pair in case of 28S RNA. Further, the phylogenetic analysis is clearly indicating all the 16 isolates have been closely associated and found to be associated with the Phenacoccus saccharifoli isolate, which is in the China. And this is the first molecular characterization which was done in India. Previously, it was done in 2020 in China. So among India, this was the first molecular characterization. Next, please. And coming to the association study, so we have a doubt whether the fungal spore was added here. So we started with the internal and external association, and it is clearly indicating in internal association no fungus growth was seen even in the gut also. Whereas the external association, you can clearly see the growth of fungals around the body of the crown mealybug. So we have a doubt whether, so we preliminarily confirmed it was present outside, but we don't know where exactly it was present. So we isolated mouth parts, legs, and the external powdery wax, and we have isolated the DNA separately. And interestingly, we could find that DNA which is isolated from the powdery wax got amplified for the fungal gene, TEF gene. So it was clearly molecularly confirmed. Then from that, whatever the isolate we got from the mealybug, we did a pathogenicity assay also, where we exactly produced the porcupine symptom. And further in SEM analysis also, you can clearly see the microconidia was get attached to the powdery wax. Further in GCMS analysis also, the fungal derivatives such as furan and furanons was present here. And finally, we get into the Glasser's condition. You can see in the treatment 2 exactly where we inoculated both the crown mealybug and the pathogen. It have produced the symptom, and the aggression was higher when compared to the single application of fungus and the mealybug. Next slide, please. This is the overall representation we have where we confirmed the association. You can see both the molecular level and the cultural level. We have isolated the Fusarium saccharine from the powdery max and confirmed the pathogenicity. Next slide, please. Moving to the second objective, we deal with the epidemiology. We have conducted the field trial, 3-season field trial at the hotspot region of sugarcane resuscitation in Kadalur for the season 2022 to 24, and where we noted the 4 abatic factors such as maximum temperature, minimum temperature, relative humidity, and rainfall. And here you can clearly we can see the maximum temperature and the minimum temperature have been positively correlated with the crown mealybug incidence and porcupine incidence, whereas rainfall and relative humidity have been found negatively correlated. And interestingly, you can see in ADPC curve for the 3 year, it is getting year by year, it is getting increased. And exactly the week 21, which is having the higher incidence of porcupine incidence, and it was correlating with the tillering phase. So the entire plant population will be killed in the stage of 21. Previously, disease was found to be minor only after the northwest monsoon, but due to the presence of crown mealybug, the disease porcupine was found to be found throughout the year. Next, please. There you can see in the tillering phase coinciding with the higher incidence. And next, please. Coming to the model, we did a statistical model and also machine learning model. In case of statistical modeling for predicting the disease, we used the multiple linear regression, step-based multiple linear regression. In case of machine learning models, we did a decision tree, support vector machine, random forest, and artificial neural network. Among the different models that we have used, we predicted, among that artificial neural network was found to be best fitted for the actual and the predicted values. So it is clearly indicating the better performance was seen in the artificial neural network in terms of R-square value. You can see 0.97 with the minimum RMSE and MAE value. So it clearly indicating the artificial neural network will be a better model for the prediction of crown mealybug and porcupine disease. Next, please. Coming to the salient findings and conclusion, it is very clearly indicating that the invasive crown mealybug is acting as a disease aggravator by creating the physical wound for the pathogen entry, as well as it is harboring the fungal propagules on its wax coating, thereby significantly contributing the epidemic severity throughout the nation, especially in the state of Tamil Nadu. And in case of machine learning models, the ANN neural model, which is better in terms of predicting the disease, and it is clearly elucidating the elevated temperature was the main reason for the intensified disease outbreaks. Based on the result, it is important to have a management strategy to tackle this multi-phased pathogen, as also the rapid spreading sucking pest during the combined infection in both the plant and root crop. Next, please. So I'm here take this opportunity to acknowledge ICR for providing the PhD program and Tamil Nadu Agricultural University for providing the seats, and ICR Sugarcane Breeding for conducting the experiment in ICR SBA. And I'm here to thank Sugarcane Resuscitation Kadalur also. Then I here to thank cooperative private sugar mills and sugarcane farmers for Tamil Nadu. At last, once again I thank MPPS for providing this platform to deliver a part of my research work. Thank you.
Shiva, it is a very nice presentation. So here, this crown mealybug, you told it is an invasive pest. And how you identified, confirmed the identity of the mealybug?
Ma'am, I have did both the morphological, next please. Next slide. I have did both the morphological characterization and molecular characterization for confirmation.
Okay.
In terms of morphology, we used the slide mounting and SEM analysis for the it is clearly indicating the 8-segmented antenna, whereas
Okay,
in case
okay.
of molecular, use the gene.
Then the spore is, you told that the spore is attached to the wax secreted by the mealybug.
Yes,
Okay. So it acts as the transmitting this particular pathogen?
Ma'am, actually in this study, I'm clearly indicating that it is acting as a carrier only, ma'am. So whenever the disease, first of all, I just want to tell you, ma'am, whenever the porcupine disease was in the field, it will come in two ways, ma'am. One is with crown mealybug and without crown mealybug. Whenever there was association with crown mealybug only, we could see the fungal spore on the powdery bags, and where in the other case, we couldn't find any association, ma'am. So,
Okay, okay, thank you. Nice.
So moving on to the next presentation, T3OP12 by G. Durga.
A very good evening to one and all present here. I am Dr. Durga. I am here to present on a doctoral topic where I have studied on the host expansion and genetic diversity of one of the invasive thrips parvispinus. So generally these thrips are very small and they are very Can I resume?
Yeah, you can resume your presentation.
Generally, thrips are very minute insects and they are very clever and stealthiest of all the insect invaders. Because of this smallest insect, they are very easy to travel from one place to another place. And most of these thrips are very difficult to identify to the species level, because when we see under the naked eye, we cannot identify just by seeing how it looks. Okay. So there are 8 steps where we need to do morphologically in order to mount into a slide, and then we can go through the literature and then we can identify these thrips. So during 2021, all the plant protectionists might remember that it was a great pathetic situation where this actually thrips parvispinus was invaded in the Guntur district of Andhra Pradesh. So with that background, initially I worked on Cytothrips dorsalis. Later all of my objectives got changed to thrips parvispinus. So where I worked and I did intensive surveys to know the diversity of this thrips parvispinus on different vegetables, ornamentals, and also on weeds. And I have also studied the genetic diversity, if there is any genetic diversity in the different locations. These are the different steps that I was talking about, and this is a very tedious job for any of the thrips taxonomists. So if we wanted to identify any of the thrips species, we need to wait for 3 to 4 days for processing the slide. So initially we collected the thrips in glacial acetic acid media, followed by transferring to 10% NaOH to remove the traces of NaOH. Then we have to follow with ethyl alcohol 50%, 70%, and 100%, and then we need to transfer to 1:1 mixture of ethyl alcohol and then terpenol for 2 to 5 minutes. So for identifying the thrips parvispinus, these are the taxonomic characters, and this is the slide mounted for this thrips parvispinus that I have recorded. So the image A represents how a female is looking. The female is usually generally dark brown in color. It is confused with the other thrips when it is looked under the naked eye. So we have used different taxonomic characters where we can use 7 antennal segment is a typical thing where by this we can also identify, and then there will be a continuous rows of spines through which we can identify and confirm the species. So when I visited different parts of Andhra Pradesh, Karnataka, where where I have done exclusive surveys, I got to know that this Thrips parvispinus are aggregating with 3 or 4 different types of other species. Especially, this was the image taken exclusively in the greenhouse and polyhouse condition, where I could record Thrips association of Thrips florum, Thrips hawaiiensis, Thrips orientalis, and Thrips parvispinus. All of these species will look exactly similar. So, my job was to do the morphological thing. Because of that, I could identify. So, these are the symptoms, how it looks. So these are the symptoms, how it looks when there is a huge infestation and coexistence of 3 to 4 different types of Thrips species in polyhouse condition in capsicum ecosystem. So there is a colonization of male and both female Thrips parvispinus on the chili flower. So when we look close, here we can see a light yellow one, which is the male Thrips parvispinus, and the dark brown or blackish in color, by which the name came black Thrips. So there is existence of Thrips parvispinus, both male and female, and in few cases there was existence of Cytothrips dorsalis as well. So from my studies, initially it started with just chili, and then throughout the intensive surveys, I got to know that the host range got expanded like anything. It could feed on anything and anything, and then I could see there was a great adaptability of this particular pest. So these are the different hosts that I have recorded. So I have also done the molecular characterization to check if there is any genes change in different locations. So, and definitely there was a gene change. So the population that I have collected from different locations was categorized into 2 clades. In one clade, all of these GKBK population, Kolar population, all of them clustered at one place, and the remaining population, which was collected in North Karnataka, clustered differently. So earlier, such type of studies was done for the Thrips tabaci and Cytothrips cardamomni. And it was during my time where I have done particularly on this Thrips parvispinus for the first time in Karnataka. So from my findings, I can clearly say that this Thrips parvispinus is a very dominant species, and all the organizations from all throughout our country are finding ways to manage this particular pest. Last month when I have attended the brainstorming session on this Thrips parvispinus management, we found that it was very difficult for all of us to manage this particular pest because of this high adaptability. Initially it came with the male was yellow color, and now it is also said that even the females are in yellow color. So it is a very big problem, and scientists are thinking to honor a Nobel Prize award if somebody is coming up with a perfect management strategy for this Thrips parvispinus. I would like to extend my thanks to my guide, Dr. Kaleshwar Swami CM, and to my university who has provided the funds during my study, and also Venkat Ramanappa, who has provided space for me to work for my molecular studies. And I also extend my thanks to my guide members and lab mates. And I personally thank Malabar Plant Protection Society for giving me the opportunity this evening to present on my very beautiful work on Thrips parvispinus. Thank you.
Okay. Durga, it's a informative presentation. So you reported, it is the first record?
Yes, ma'am.
Then what about in fruit crops like mango? Also it is reported?
Yes, ma'am, it was recorded, ma'am. During my time it was not recorded, ma'am.
But is there, if you told that there will be other complexes along with other species?
Yes, ma'am.
It is there. So if this parvispinus there, what is the population of other thrips?
It is definitely less, ma'am. This is perfectly replacing the other species. other species
Oh,
of
it is-
thrips.
it
It
is
is showing
dominating.
its compatibility and its adaptability. That's what everybody is wondering and they are finding difficulty. That
So
is
in
the main
Chile,
problem.
nowadays in Chile, it is the dominating species?
Yes, ma'am, it is not in nowadays. Over the past 5 years, this is the only pest which is ruling this chili ecosystem as well as other vegetables also, ma'am.
Okay.
Okay, thank you. Sir, they are showing sexual dimorphism and they are showing different morphs, sir. That's what. That is not happened over the night, sir. We feel that it happened over, we will blindly believe that male is black and female is, male is yellow color, female is brown color, when we see through naked eyes. So when we get into this actual taxonomic thing only, we are getting to know. Not everybody knows that there is a dimorphism. Only the taxonomists who are doing intensive work on this slide preparation are getting to know, sir. That's what, it needs too much of work and maybe it will take some years for any of the organization to come up with what exactly is happening with this pest. Earlier, for any of the invasive pest it was establishing, there used to be 1 or 2 management practices which was good enough for in this 5 years. But this is the one pest, till now there is no perfect management aspects. But what I personally felt is, when I visited to the Rave village as a part of agro clinic, me, Saji sir and Siddharth, we went to Madikei Taluk, sir. There we visited a farmer who is practicing organic management. There we saw a chili crop which was having a minimal population of Thrips parvis spinus, and I was very happy to see that there was good enough number of predators as well. So far there are no predators exclusively feeding on Thrips parvis spinus. What I feel is, if we calm down and nature will take its own way to tackle the pest, I feel, sir. This is the only solution as of now. Sir, we are using systemic insecticides. Sir, sticky traps are also working well, sir. But problem is the population here is really high. We cannot just rely on the sticky traps. Actually, blue sticky trap is working. Yes, sir. No, sir, it is showing resistance to that. That's what even company people are trying some way to tackle with this management, especially on a chemical basis. Yes, sir. I have met several of my friends and they say that what they say exponents, the others say they say some other chemicals. They are not static with the chemical also, sir. Yes, sir. Yes, sir. That's what's. Sir, Indonesia, it got invaded to our country, sir. Indonesia. Yes, sir. So this Guntur people used to import the chili seedlings from Indonesia, and it is said that Indonesia is the main cause where majority of our farmers are also being troubled by this particular pest. Yes, sir. Anthocorid works are doing good job, sir, but we have to give some time for them to develop, sir. Instead, we are spraying chemicals. That is the problem. Yes, sir. Chili is the one crop where majority of the Indian economy is based, and they are depending on this chili for their export as well as income, sir. If we convince farmers saying that natural enemies would emerge and they will control, they may not listen, sir.
But Durga, there in Velaini, the student of Tania, he had done work in this.
Yes, ma'am.
And there is insecticides with which we can manage it.
Ma'am, not fully, ma'am. As per the literature or discussions made with the companies.
It's already published work from Velaini.
No, sir. This doesn't transmit any of the virus. Thankfully, that is the only plus point with regard to this pest.
Okay, thank you. Next.
With this, we complete the oral presentations of this technical session. And now we invite Dr. K.M. Srikumar sir to present a memento to Dr. Susha S. Tara, the chairperson, on behalf of Malabar Plant Protection Society. Thank you, sir. Now we will continue with the poster presentations. And the chairperson for the session is Dr. Sainamal Kurian, Professor, Plant Pathology.
So we are moving on to the poster presentation. We are a little bit short of time. The first presentation is T3PP6 by Reshma SR. T3PP6.
Time allotted is only 5 minutes. Okay. 3 minutes, 3.
Good evening all, respected teachers and friends. I am Reshma SR from College of Agriculture, Vellanikara, Thrissur. Bemisia tabaci is one of the most important globally distributed polyphagous insect pests that cause severe damage, and since it is a cryptic species, the present study was mainly done to investigate the morphological diversity of Bemisia tabaci across various host plants and all over Kerala. So, in order to do this, first purposely sampling surveys were conducted in the selected agroecological units covering all the 14 districts of Kerala and approximately 60 locations. And the main crops selected were cassava, okra, chilli, brinjal and tomato, and the puparium, that is the 4th nymphal stage of Bemisia tabaci, along with adults were collected from each crop from each 60 locations, and the processing was done for mounting, and staining procedure was also conducted by acid fuchsin, and then clove oil was used to mount the slide. And then 15 pupal characters, which include pupal length, pupal width, vesiform orifice dimension, lingular dimension, the wax margin dimension, and 9 adult characters like body dimensions, antennal dimensions, forewing dimension, and the main male important taxonomic characters like Aedegus length and clasper length was also studied under this research. And since there are a lot of characters, principal component analysis was done, and it was seen that the populations from the cassava showed a larger size in pupa compared with that of chilli, and significant differences was seen in pupal length, pupal width, lingular length and wax margin length. And in case of adult, brinjal showed a highest dimension for body length and body width, and in case of Aedegus length, okra showed a higher dimension, and in case of clasper length, the populations from cassava showed a higher dimension. Regional wise significant differences were also noted. The the populations collected from Wayanad and Idukki showed a higher values in characters like pupal length, pupal width antenna length, forewing dimension, and lower values were seen in districts like Kollam, Ernakulam, and Kasaragod districts. So we can conclude that Bemisia tabaci population in Kerala show notable significant difference for various characters in various host plants. Thank you.
Okay. It is a nice presentation. So, is there any difference in the infestation? You told that there are difference in populations.
Yes,
So,
ma'am.
is there a infestation level?
Yes,
Is
ma'am.
there-
In Wayanad and Idukki districts, the cassava plant was mainly infested. The infestation rate was highest for cassava, and the dimensions was also highest for cassava, the pupil width.
So what is the relevance of the study? You are collecting many populations from different places.
Yes, ma'am. There
So-
is significant morphometric differences, and this reveals that there may be further studies have to be done because there may be high, since the Aedes length was observed higher, there will be reproductive capacity higher, virus transmission like that. So this morphotype along with genetic diversity studies were also conducted, and it was seen that the cassava was infested with heavily with this Bemisia tabaci population.
So what about in okra?
Okra, ma'am, the rate of infestation mainly seen was highest for cassava, then tomato, then brinjal, then okra, then chili. That was the order of infestation.
Okay. Thank you. Next.
So moving on to the next presentation, T3PP7 by E.S. Lakshmi Priya. T3PP7 Lakshmi Priya.
Good evening all, respected teachers, judges and everyone present here. I am Lakshmi Priya ES, Development Associate from MS Swaminathan Research Foundation, and I am here to present on the topic effectiveness of participatory agri-clinic interventions in crop health management and farmer awareness, evidence from Manjapra village, Kerala. As you all know, crop health management is an essential strategy for improving the agriculture productivity and for sustainable farming. But farmers often find it very difficult in order to identify their pest and disease incidents in their own agricultural field and to adopt some scientific crop management practices. So, this participatory agri-clinic intervention could be an essential tool in order to bridge the gap between the farmers and the researchers and to adopt the practical solutions in the agricultural field. So, in regard of this, the study was conducted in order to evaluate the effectiveness of this agri-clinic to the farmers and to adopt some scientific management practices to the agricultural field. So, the methodology which followed include data collection through regular field visit and farmer interviews, group discussions were conducted and expert diagnosis were taken. Then assessment of awareness and adoption behavior was done. It was recorded before and after the intervention through direct questioning and by response scoring. Then participant satisfaction analysis was also done through the farmer feedback, which recorded high, moderate and useful through scoring manner. The results include, out of the total crop health problems reported, the banana and coconut reported the highest crop health problems, in which the banana constituted 28% and coconut about 24%. And out of the major problems discovered, the pseudostem weevil attack was found to be higher in banana, and the rhinoceros beetle were found to be higher for coconut. And out of the total crop health management problems, the total disease was about 44% and the insect attack occurred about 56%. The awareness level of the farmers were also calculated. So before the intervention, the awareness was only about 38%, and after the intervention, it was found to increase to 82%. That is, the improvement occurred about 44%. And the participant satisfaction level was also calculated, and the 88% of the farmers reported it as highly useful, while 12% of the farmers reported it as moderately useful. The farmer adoption behavior was also calculated, as the 76% of them were found to be willing to adopt this, while 24% were not willing or not sure to adopt these problems. So in conclusion, the participatory agri-clinics empowered farmers with scientific knowledge and improved crop health management and promoted sustainable farming practices. Thank you.
So, in Wayanad only, in coconut only, rhinoceros beetle problem only?
No, ma'am. Means majority of them reported this rhinoceros beetle.
No Rugose spiraling whitefly they reported?
They reported many issues for coconut, including the nutritional disorders also. But the major one was rhinoceros beetle attack.
In young palms or in?
Ma'am, elder palms, 10 years and above, they reported.
Okay. Okay, thank you. Next.
Moving on to the next presentation, T3PP1. It's being presented online. Annie Matthews. T3PP1. Are you ready?
Yes, sir.
Okay, Anu, you can present.
Okay, thank you. Good evening to all. I am Annie Matthews, PhD scholar from College of Horticulture, Vellanikara. I am here to present the role of defense-related enzymes in malformed tissues associated with jackfruit bronzing. As you may be aware, bronzing is an emerging disease of jackfruit in Kerala, especially in Vietnam Super Early variety, which is a popular variety for orchard planting in Kerala. And the symptoms include yellowish to orange to reddish brown discoloration and rusty specks on the bulbs and the rags. And there are no external symptoms on the fruits. And the pathogen associated is Pandia stivati, subspecies stivati. And in Kerala, in addition to this, we got Pandia stivati, subspecies indogenous is also found to be associated with jackfruit bronzing. And the histopathological studies using microtome sections of diseased and healthy rags have shown that what we see as a speck and streak on the rags and bulbs are actually host tissue proliferations like hypertrophy, hyperplasia, and wound periodem formation. So the role of defense-related enzymes in these malformed tissues associated with jackfruit bronzing was studied. And the methodology included estimation of total phenol content of the diseased and healthy rags, as well as the enzyme activity was analyzed for peroxidase, polyphenol oxidase, phenylalanine ammonia lyase using spectrophotometric methods, and calcium and boron of both the healthy and diseased tissues were analyzed. When coming to the results, the total phenol content was significantly higher in diseased rags compared to the healthy rags. And in case of peroxidase and polyphenol oxidase, though their activity was higher in case of diseased rags, they were insignificant compared to the controlled or healthy rags. Whereas the PAL activity was markedly elevated in the diseased jackfruits, and also they were significantly statistically significant compared to the healthy rags. And in addition, the calcium levels of the diseased tissues were found to be almost 10-fold higher compared to the healthy tissues, whereas the boron, which is was almost the same in case of in case of healthy and diseased tissues. So from this, it is inferred that the increased PAL activity is involved in the structural and biochemical changes associated with jackfruit bronzing disease, which is triggered by Pandia stivati. Thank you.
Anu, is this the first report of this disease, or where it is reported for the first time?
Teacher, it was reported in 2014 from Philippines. Thereafter, from 5 different countries, and from India, it was reported in 2023 from BRS.
Okay, when do you think it was introduced to India?
Teacher, that is, I'm not sure, teacher, because this variety Vietnam Super Early is particularly given by one particular private nursery. It is so we are not sure the origin of this Vietnam Super Early, teacher.
Okay, thank you, Anu. Thank you.
Okay.
So we will move on to the next presentation. T3PP3 Arsha Kamal. Arsha Kamal, are you ready?
Yes, sir.
Okay, you can present.
Good evening to all. I am Arsha Kamal from Department of Plant Pathology, College of Agriculture, Velaini. I am here to present the poster titled Prevalence and Serological Detection of Virus Diseases in Culinary Melons of South Kerala. Culinary melon, the Cucumis mellow variety acidulus and conomon are economically and culturally very important to South India and in Kerala, and they are valued for their shelf life also. However, their productivity is severely affected by a high incidence of virus diseases. Despite increasing this threat, the scientific data on the viruses that affecting these varieties in our region actually remains very limited. So to address this issue, a systematic survey was conducted across 50 locations in Trivandrum, Kollam and Alappuzha on AUs 1, 3, 4, 8 and 9. Using a 0 to 5 disease scale shown here, the plants were assessed for disease incidence and vulnerability index. Later, it was identified, these viruses were identified using serological detection methods, specifically indexing for cucumber mosaic virus, papaya ring spot virus, cucumber green mottle mosaic virus, and cucurbit aphid-borne yellows virus, using DAS and TAS ELISA. The surveys revealed that there was a high incidence in areas like Kaliyur in Trivandrum and Mannancheri in Alappuzha, and reaching 100% of disease incidence. The field also observed with the complex range of symptoms like mosaic, mottling, leaf deformation, puckering, and vein banding and stunting. The serological data from 15 locations were recorded that, and the cucumber mosaic virus was considered as the predominant virus, and it is detected across 10 locations. Papaya ring spot virus and the cucumber green mottle mosaic virus were detected in 5 locations. Cucurbit aphid-borne yellows virus were detected in 3 locations. The survey and the detection found that rather than a single infection, the mixed infections were more common in the field. While single infection caused a notable damage, the data reveals that the increase in vulnerability index were more in multiple infection, and there were a significantly jump in vulnerability index up to a 72.96 in Kaliyur, where all 4 viruses were detected. This indicating that when these multiple viruses were interact, it can be more destructive to the host plant. In conclusion, the study established the prevalence of cucumber mosaic virus in culinary melon in South Kerala, and the mixed infections are a major reason for crop damage. And this is the study. The study recorded the first ever documentation of cucumber green mottle mosaic virus and the cucurbit aphid-borne yellows virus infecting culinary melon in Kerala. Thank you.
Asha, can you hear me?
Yes, ma'am.
What do you think the reason for increasingly reporting this multiple infection of viruses in vegetables, especially in cucurbitaceous vegetables? What may be the reason for that?
Mainly, cucurbits have a wide host range, like even in wheat, especially in the case of cucumber mosaic virus, it is having a wide host range. So the increase in the prevalence of CMV can be justified by that. Also, the new viruses like CGMV and CABV were also emerging in India, and especially even present in Kerala because of the climate change and the host change, and especially the associated virus vectors like the microspecific and the FSOCP.
Okay, Arsha, thank you.
Thank you.
Thank you, ma'am. We'll call upon the next presentation, that is T3PP4 by Sherin Mariam Reji. T3PP4.
Good evening.
Good evening all. I am Sherin Mariam Raji, Department of Plant Pathology from College of Agriculture, Velaini. Today I am here to present the poster titled Cultural Characterization of Thread Blight Pathogen Infecting Cocoa in Kerala. As you all know that
cocoa is an economically important plantation crop where thread blight is one of the emerging serious disease which is nowadays seen on the different cocoa growing regions, and this pathogen, this disease is caused by the Marasmius species, and this is characterized by the thread-like symptom which is seen on the cocoa stems and leaf, and also this disease leads to further defoliation. As you see in the picture, the first two figures shows the horsehair blight typical symptom, whereas the third picture onwards it shows the white thread blight pathogen, and these pathogen show different patterns on the stem. Because of this serious disease, it is necessary to understand the cultural variability for further studies and disease management strategies. Coming to the methodology part, purposive sampling survey was conducted in cocoa growing regions across the Kerala, include Idukki, Kottayam, Ernakulam, Thrissur and Kozhikode, and isolation and purification was done after the sample collection from the infected field, and then pathogenicity is proved with the help of Koch postulate, and different observations was recorded. Coming to the result part, it shows that the cultural variability among the collected isolate in the different parts of Kerala, and as you see in the table, in colony color, colony texture and growth rate, it shows the variation. In case of colony color, it shows the color ranges from creamy white to pure white color, and in case of colony textures, it changes from cottony, dense, fluffy, compact, etc. And in case of the average growth rate, as you see in the table, WE2, which shows the higher average growth rate per day. This isolate was collected from the district of Ernakulam, and it is molecularly identified as Marasmius palmivorus. And compared to the other HT and HI, which shows the comparatively lower growth rate, and it is horsehair blight. And in case of media standardization, we have used different media, and in that V8 shows more faster growth, and has to conclude that in pathogen collected from the isolate collected from the different district of Kerala shows culture variability, and this study help us. This study is help us for the further molecular as well as for effective disease management strategies. Thank you.
Your name? Okay, can you tell me what is the extent of spread of this particular pathogen or disease in Kerala?
It is mainly happen because of higher humidity.
Extent of spread. Out of the number of gardens you have visited and samples were collected, how many were infected? How many did you get the samples, infected samples?
Almost all places I went for collecting, 5 district I went, and all the district I got sample. One district, Wayanad, I went, but there was no thread blight infection on the field I visited.
So except for Wayanad, all the other districts where cocoa is cultivated, this disease is present?
All the other districts means, ma'am, Idukki, Kottayam, Ernakulam, 5 AU, it was there.
How many gardens in each AU?
How many?
How many gardens were visited in each AU?
For the purpose of studying, I am taking samples from 5 from each AU.
Okay, thank you.
Thank you.
Nice work.
Yes, sir, it infects other crops like nutmeg, tea, coffee, rubber. It is seen in these different species, sir. In cocoa itself, 4 species are there. Thank you, sir.
Moving on to the next presentation, T3PP5, Deepana M.
Good evening everyone. I am Deepana M, PhD scholar from Department of Plant Pathology, College of Agriculture, Vellanikara. Today I am presenting my work on pathogenic and phenotypic variability among isolates of Lacediplodia species infecting cocoa. As we all know that cocoa is an economically important plantation crop. It is as a raw material for cocoa chocolate industry, and its productivity is significantly affected by many fungal diseases. Among these, the fungus Lacediplodia causes dieback, leaf blight, and pod rot diseases in cocoa. Earlier, it is considered as a minor pathogen, but they are increasingly associated with severe disease outbreaks due to the varying environmental conditions. And therefore, this study was undertaken to characterize the different isolates of Lacediplodia and to study their pathogenic variability. And for the study, the infected samples were collected from different parts of the Kerala, including Thrissur, Wayanad, Idukki, Ernakulam, and Kozhikode. And then the pathogen was isolated and purified under laboratory condition. And after that purification, the isolates were subjected to morphological and cultural characterization, and then pathogenicity tests were done, and after that statistical analysis were done. Then coming to the results, most of the isolates showed average growth rate of 3 cm per day. It is similar for all, but it differs in morphological characters. And some isolates showed black woolly to grey woolly colonies, and the conidial morphology showed different differences in their characters. And in the conidial color ranged from dark brown to light brown, and the conidial shape ranged from ellipsoid to ovoid and sub-ovoid. And this picture shows the morphological characters of Lacediplodia. And coming to the pathogenic variability, the pathogenicity test can reveal the significant difference among the isolates in virulence. And based on lesion diameter, they were classified into highly virulent, moderately virulent, and less virulent. And isolate KL1 is identified as highly virulent, and significant differences were observed in statistical analysis. And I conclude that the isolate had similar growth rate, but they differ in morphology and in virulence. So this is considered while developing effective management strategies in cocoa cultivation. Thank you.
What are the other symptoms? What are the symptoms? You have not explained any symptoms here. What are the symptoms caused by this particular pathogen on cocoa?
Mainly 3 diseases are there, ma'am. In dieback, that twig blight and leaf blight, everything will be done.
Dieback, then?
Dieback, ma'am. From tip to downwards, it is the plant is dying.
Okay. Other than dieback?
Other than that, twig is also having that dieback symptom, ma'am, or in branches like that. Then leaf blight symptoms, and in pod rot, that pods are having black color spore moss on that surface, ma'am.
Okay, is this pathogen associated with any disease of unknown etiology? Have you come across? Is this pathogen has been reported with a disease of unknown etiology in cocoa? Have you come across any reference? There is a disease of unknown etiology in cocoa. Is this pathogen reported with that? Associated with that?
That I didn't know.
Okay, you may refer.
The poster presentations are completed. We will proceed with the flash talk now. Calling T3FT3 Ajmala Jeven for the presentation. You can start now. The time allotted is 3 minutes.
Good evening, respected judges, distinguished scientists, esteemed faculty members, and my dear friends. I am Ajmala Jabin, final year BSc Agriculture student from College of Agriculture, Padnakad. And it is a great privilege to present my flash talk titled The Hidden Cost of Green Revolution, a Soil Health Perspective. Green Revolution is widely celebrated as one of the greatest achievements in the agricultural history. It enabled different countries like India to move from food scarcity to food security. Yet, beneath this extraordinary success, there lies a critical question that, what did this progress cost our soil? In the pursuit of higher yields, we relied heavily on high-yielding varieties, intensive cropping system, monocropping system, and synthetic fertilizers. These practices boosted the production, but they also initiated a gradual but profound decline in soil health. Soil organic carbon has diminished, excessive use of nitrogenous fertilizers has disrupted nutrient balance and induced deficiencies of essential micronutrients like zinc, iron, and boron. The living components, that is the beneficial microorganisms which drives the nutrient cycling and organic matter decomposition has declined in both its activity and diversity. And also at the same time, the physical degradation, including reduced porosity, lower water holding capacity, and compaction has restricted root growth and increased vulnerability to drought. The consequences are increasingly evident. In many regions, crop yields are stagnating despite ever-growing dependence on external inputs. This reminds us of a fundamental truth that the soil is merely not a medium for crop growth, but it is a living system that sustains life itself. To restore this vital resource, we must adopt sustainable practices such as integrated nutrient management, conservation agriculture, crop diversification and rotation, organic amendments and mulching, biofertilizers and biological inputs. So, these approaches rebuild soil organic matter, revive microbial life, and strengthen long-term resilience. So in conclusion, the Green Revolution taught us how to feed the crops, but the future of the agriculture depends upon learning how to feed the soil. Because when we protect our soil, we are protecting the very foundation of sustainable agriculture, environmental stability, and global food security. Thank you for your kind attention.
Excellent topic selection and excellent presentation, Ajmala. Give her a very big round of applause. Thank you.
Calling the final presenter, T3FT4, Fatima Niba Rihan. Sorry, Dian Asrin. T3FT4.
Good evening, respected dignitaries on and off the dais. I am Diya Nasrin, a fourth year UG student from College of Agriculture, Padnakad. Today, I will be talking on the topic, impact of climate change on plant disease dynamics. So, while we often focus on how climate change causes droughts, heat waves, floods, etc., there is a quieter but more dangerous shift that is happening in our fields. Climate change is fundamentally reshaping the plant disease dynamics. It isn't just about the weather. It is about the change in interaction between the plant host, the pathogen, and the environment. And now, it even threatens our global food security. So, how are the pathogens winning? First, the rising temperature that acts as an accelerator, shortening the infection cycles, and also it helps the pathogens to grow faster, which leads to more virulent and adaptive strains. And in second, the changes in the rainfall and humidity creates a perfect nursery condition for the pathogens. The excessive moisture, it enhances the spore germination and infection. While the rain splashes that act as a physical transport system for both the soil-borne and airborne pathogens all across the field. At the same time, the environment is moving, which means that the climate change is driving the pathogens into newer geographical areas, exposing the crops to diseases that they have never seen before. In addition to this, our crops are becoming more vulnerable. The extreme events like droughts and floods are actually weakening the plant's natural defense mechanism and leaving them open to many other infections. Even the elevated carbon dioxide levels in our atmosphere, which we might think that it would be beneficial for the plants, are actually changing the plant's internal chemistry and physiology that further disrupt its defense mechanism. So, how can we fight back? For that, we must move beyond the traditional methods, and we'll need to integrate more strategies like climate-based disease forecasting, early warning systems, and our goals should be more aligned towards integrated pest and disease management, climate-resilient crop protection practices. So, I would like to conclude by saying that even though the climate change is reshaping the plant disease epidemiology and also posing a threat for our global food security, by closing the gaps in our understanding how the pathogens adapt to the field and on working to it, we can secure a more sustainable agricultural future. Thank you.
Excellent presentation, very nicely presented. Thank you, Diya.
With this, we conclude the technical sessions. We request Dr. K.M. Srikumar, President, Malabar Plant Protection System. We request our Honorable Dean, Dr. T. Sajida Rani Madam, to present the memento to the chairperson and co-chairperson of the previous technical session. We'll be starting the valedictory program shortly. The students are requested to come to the front rows. Please occupy the seats in the front rows. I repeat, the students are requested to come and occupy the seats in the front rows. Inviting Dr. Sumia for the welcome address. Inviting all the dignitaries to the dais. Welcoming all the dignitaries to the dais. We'll begin the program with the welcome address by Dr. K.V. Sumia.
Respected dignitaries on and off the dais, a warm good evening to all. It gives me immense pleasure to extend a warm and heartfelt welcome to all those who have gathered here for the valedictory function of our two-day national seminar on resilient agriculture, innovations in plant health management and biosecurity. For the past 2 days, this seminar served as a vibrant platform for scientific deliberations. We had, over the 2 days, we had seen insightful deliberations over different topics such as invasive pest and pathogen, which are causing challenges to agriculture, importance of following strict regulations for biosecurity, as well as we have seen several innovative approaches aimed at a sustainable agriculture system. Being the first national seminar of the society, we are very happy that we could receive 108 registrations. We had received around 74 abstracts, of which we had selected 36 for oral presentation, 10 for poster presentation, and we had 4 flash talks. So that's it. I welcome our chief guest, Dr. K.N. Anith sir, the respected Director of Research, Kerala Agriculture University. We are honored to have your gracious presence for the occasion, sir. Next, I extend my warm and sincere welcome to Dr. Sajida Rani, respected Dean of College of Agriculture, Padnakar, Madam's constant guidance was there throughout the organization of the seminar. I extend a warm welcome to Dr. Sri Kumar, President of Malabar Plant Protection Society. Sir has been taken keen interest in the seminar from the very inception of the idea. He was behind all the organization. I extend a warm welcome to you, sir. Dr. Sajish PK, he is, as Dr. Sanamul Madam had mentioned, he is the very person behind the idea, and it is mainly his hard work that has transformed into the success of the seminar. It's a pleasure to welcome you, Dr. Sajish. I also extend a warm welcome to Dr. Sainamol Kurian, the person who had taken leadership for the meticulous organization of this seminar. I also extend my heartfelt welcome on behalf of Malabar Plant Protection Society and College of Agriculture, Padnakad, to Dr. Nisha MS, Head of Department of Entomology, for the support. And her support was also very instrumental in the seminar. I extend a warm welcome to all the delegates present here, online and offline, for the valedictory function. And I take this opportunity to place on record my appreciation for your efforts, for the participation in seminar, and as well as for sharing your ideas, your research findings, and that has made this seminar a success. I extend a warm welcome to all members of organizing committee, faculty and students of this college, without whose support we could not have conducted this seminar. I extend a warm welcome to one and all on behalf of Malabar Plant Protection Society and College of Agriculture, Padnakad. Thank you.
Thank you, ma'am. Now we invite Dr. T. Sajida Rani, ma'am, Dean, College of Agriculture, Padnakad, and General Council member of Kerala Agricultural University, to deliver the presidential address.
Respected Dr. Anith, the Director of Research of Kerala Agricultural University, Dr. Sri Kumar, President of MPPS, Dr. Saidmol Kurian, the Secretary of MPPS, Dr. Nisha, Dr. Sajish, and my dear colleagues and students and the participants on and offline. Today, this is the closing ceremony of the 2 days hectic schedule on different aspects of this plant health management. I think we had a good session. For the 2 days, there was elaborate presentation of different topics, and I think that, and there was a talk about this Pesticide Act. So I can, because I think that it's the best day in our college, because this is the first time a society making this type of symposium for 2 days. And that is two fruitful discussions and deliberations, as well as too much knowledge came through this. So I'm sure that this seminar will definitely help the farmers, so that new techniques have been discussed here, so that will reach in the hands of farmers. And with this few words, I would like to conclude my presentation. And I congratulate Dr. Sainamul, Dr. Sajish, as well as Dr. Sri Kumar for making this event a grand success in this college. Thank you all.
Thank you, ma'am. Next, we invite Dr. K.M. Sri Kumar, President, Malabar Plant Protection Society and retired professor, Department of Entomology, to present the report on draft pesticide management bill.
So, we had a very good discussion, very elaborate discussion on the draft pest management bill at the AV lab, which was attended by scientists, farmers, agricultural officers, as well as some experts from the Department of Agriculture headquarters at Trivandrum. It was chaired by Dr. Bindu Podikunju, Professor, Head of the Department of Agriculture Extension, and Dr. Binita N.K., Associate Professor and Head, Department of Agriculture Science and Agriculture Chemistry. And we had a detailed discussion. I am not going into the details of it because of the shortage of time at the VR of this time. So, one of the major recommendations we have finalized was that giving a number on the pesticide container. If it is an insecticide, we have to follow the IRAC number classification, and that number can be given on the container, so that when a farmer buys it, he will be remembering a number, maybe 1A or 1B. And if it is a fungicide, it will be FCRF, fungicide classification. There will be some numbers. That number can be given on the container, so that when the next time he purchases another pesticide, it shouldn't be of the same number, so that the chance of resistance buildup will be very, very low. So that can be incorporated in the pesticide literacy that going to be imparted to the farmers by the Agriculture Department as well as Agriculture University. So that is one of the major recommendations, and there are many other recommendations. And also one of the concerns raised by the farming community was that price control. See, at present there is no price control. There is price control for chemical fertilizers because it is an essential commodity and price regulation is there. But for the pesticides, there are no price control. So one of the suggestions came from a farmer was that, like generic drugs, we are giving through the Pradhana Mantri Jan Aushadhi Yojana. Likewise, there should be a scheme for generic pesticides which are out of patent regime. Not the new ones, but the old ones which are out of the patent regime, which is produced in the country, can be given at a controlled price, so that it will be reaching the farmers at a fair price. So that was a recommendation by the farmer, a suggestion by the farmer, and our Pidambaran sir supported it. He said it was a very good suggestion. Likewise, there are few more suggestions. We'll be writing it, and that will be submitted to the Ministry of Agriculture, Government of India soon. The time is already over. The last date for giving the suggestions was February 5th, but we will be anyway submitting because it is not closed. It is not taken for discussion in the Parliament. So time is available. We will be submitting at the earliest. So that was a very fruitful discussion. Thank you very much.
Thank you, sir. A significant highlight of the final session is the distribution of awards. We invite Dr. Anith KN, Director of Research, Kerala Agricultural University, to distribute the awards and certificates to the distinguished winners.
Technical session 1, oral presentation. First place, Ashwati J. Ashwati J. Second place, Ratnakala B. Ratnakala B. And third place, Ashita Joy. Ashita Joy. First place, Ashwati J. Second place, Ratnakala B. And third place, Ashita Joy. Technical session 1, poster results, Naga Jyoti N. Only one prize is given, Naga Jyoti N. Technical session 2, oral presentation. First place, B.R. Ajesh. First place, B.R. Ajesh. And second place goes to G. Durga. Second place, G. Durga. And third place goes to the team Rashmi Vijay Raghavan, Aisat Nabina and Swati GS. Team Rashmi Vijay Raghavan, Aisat Nabina and Swati GS. It is shared by 3 persons. It is shared by 3 persons? 3 persons. Rashmi Vijayaraghavan, Aisat Nabina, and also to Swati GS. Technical session 2, flash talk. First place, Fida Naureen Iwe. Fida Naureen Iwe. And second place, Ashna P. Aziz. Technical session 3, oral presentation. First place, Shiva M. First place, Shiva M. Second place, Nandita K. Second place, Nandita K. And third place, Ann Tom. Third place, Ann Tom. Technical session 3, poster presentation. First place, Anu Annie Matthews. Anu Annie Matthews. Second place, Sherin Mariam Reji. Sherin Mariam Reji. Sherin Mariam Reji. And third place goes to Reshma SR. Third place, Reshma SR. Technical session 3, flash talk. First place, Ajmala Jabin. Dia Nasrin also bags the first place. Both are having the first place. Congratulations to all the winners. As the end note of today's proceedings, we invite Dr. Sajeesh P.K., Assistant Professor, Department of Plant Pathology, and Treasurer, Malabar Plant Protection Society, to deliver the vote of thanks.
Thank you. A big list is there in front of me in mobile as well as in this one. So, honorable dignitaries on the dais, respected teachers, dear participants, good evening. As we reach the conclusion of this insightful two-day national seminar, it's my distinct honor and privilege to propose the vote of thanks to, on behalf of Malabar Plant Protection Society and the College of Agriculture, Parnakad, as well as KU. To our guest of honor, and the first and foremost, I extend our heartfelt gratitude to Dr. T. Sajida Rani, Dean of COA Parnakad and General Council member of KU, for her presidential address and her unwavering support in making this seminar a reality. Your leadership continues to inspire us, madam. Wholeheartedly, she supported for this event at its beginning till today. Thank you so much, madam, on behalf of MPPS, College of Agriculture, KAU, and my personal behalf to our distinguished awarder and the guest. We are deeply grateful to Dr. Anit Kayan, Director of Research, Kerala Agriculture University, for joining us today from the morning onwards till now as chairperson of the session, as well as a lead speaker in the session, and also distribute the awards. Your presence has added great prestige to this ceremony and encouraged our budding researchers. Thank you so much, sir, on behalf of MP College, KAU, and my personal behalf. And special thanks to our own President, Dr. K.M. Srikumar, for this comprehensive presentation of the report. This is, and also have involved in the draft pesticide bill, management bill 2025. And this was a critical topic of, for these farmers and industry, as well as our research community. And more than that, he is one who led us a successful completion of this grand event. Thank you so much, sir, for this one. And Dr. K.V. Sumaya, for the warm welcome address that set the perfect tone for this valedictory session. And she is very instrumental to build this seminar in this level. Her leadership within has been vital to this event success. And I would like to extend my special gratitude to Dr. Sainamul Kurian, Secretary of MPPS. Her meticulous planning and coordination, dedication have been the backbone of this entire seminar, who guided us from the beginning till now and very much inspirational for the conduct of this national seminar. Thank you, madam. Thank you so much. And Nisha, madam, Head of Department of Entomology. We extend our special thanks to Dr. Nisha M.S. for her support, for the collaborative spirit of the entomology department in this making this event so success. To the participants who joined us from various institutions like ICR Institute, State Agriculture University, Kerala Agriculture Universities, University Centers, Central Universities, deemed universities, thank you so much. Thank you for your active engagement and for contributing to this vibrant discussion over the course of this seminar. We hope the knowledge shared here will be instrumental in your future endeavors. And finally, I need to thank, special thanks to certain members of certain people. A special thanks to Dr. C.K. Pidambaran and Dr. Joseph Rajkumar, patron of our MPPS, who are here, who are in online always and guiding us for this smooth conduct of this program. And special thanks to all the lead speakers, chair, co-chair, rapporteurs of all the technical session. Special thanks to all the farmers who attended this program, that is technical session 4. All the department officials, industry, and deputy directors, ADAs, and AOs. Especially, thank you so much on behalf of MPPS. A special thanks to my member of MPPS and scientist from different institutions of KAU, like CEO Velaini-Kara, RARS Pilikot, KVK Kannur, PRS Pannur, etc. Special thanks to my fellow faculties and conveners and co-conveners and the members of various committees of this event. And special thanks to the media people who are attended and who covered the entire event. And special thanks to the Sigma Live team for the smooth conduct of this event and the smooth conduct of this event in online too. And Mr. Umesh for this, who wholeheartedly support, especially the technical things. It is wonderful actually. And some breaks were there in between, but he managed a lot. And last but not least, there is a list of wholehearted thanks to those people. I can't mention those. In my heart, I'm telling this to those people that, and who are sitting here in front of me. Dr. Dhanyalakshmi is here, who is one among us. Dr. Sinthamol Madam is sitting here, and along with Remya and Jubi. This was nicely managed and announced nicely, and sitting a lot, more than I think 12 hours and all. They are sitting together and they are managing this program. And Krishnasri, who did this, our mess or our menu or our diet more excellently, and her team. I can specifically mention specifically mention her team, Darin, Manish, and Nishant and team. We were sitting here, and we are doing a lot of things here, but they were running for the food and arranging those things. And our students also, Ajin and Abhinanda, they were also behind that one. And I appreciate that their efforts. And thank you so much. And also our team include Faiz for documenting and all. And Unni and Karthik were sitting together and managing this session and arranging the session and casting and planning everything for the successful conduct of this each online and offline seminars. Thank you, Unni and Karthik, for your support. And I need to mention one more person who is there for reporting this one, and we are having the report, and she is none other than Rose. She is also behind that one because we are not noticing those people here, but she is also there. Thank you, Rose, for your support. And more than that, I need to tell another category of people that is none other than ELP students. They are there because of this decorations. They are there for the participation. They are there for their excellent talk in the various technical session as flash talks. Thank you so much, ELP students. And yesterday night was wonderful because of the cultural programs by various students from the department, not only in the departments, as well as from the UG students. And more than that, the event was so beautiful because of a Mohiniyattam performance by Annie Sajayan, daughter of our Sainamal madam. Thanks to madam, and thanks to the whole team for their this contribution to the cultural program. And last but not least, my department as well as my neighboring department, none other than pathology and entomology. We are family, and the COA Parnakad, how the family is, again inside one family is there, that the plant protection. And all the faculties that's sitting here, all the faculties are here. Leena madam is here, Anjali madam is here, Nisha madam is here, and the V team, Durga, Siddharth, Anusri, Ajesh, and team of this one. And it includes PG students, seniors and juniors. Especially I need to mention some part, I think entomology students were missing in between, but still some people contributed. Last time they came and they contributed, but I need to mention some people who are instrumental for preparation of this abstract, coordinating this abstract, that is the PG students like Pooja. This design, it's by Pooja. I need to appreciate that one. This design is by Pooja. And the abstract, what we are just have seen, that is because of the efforts of students and all. Pooja, Ashwati, Adira, and Aksa Bilyada Sacharde, and even Aditya and Ajin, Abhinand and all, and my entomology students, they are also contributed. And thank you so much for your sincere effort, because you are the backbone of this seminar. You are the backbone. Continue this effort further in your life too. So thank you so much for your all the participants. And finally, I'm thank the technical staff. Anybody I'm missing, technical staff, students, volunteers from other department and all, and everyone. I forget to tell about one more person, that is Hebin. He was not in any committee, but he came to me and asked works. Just give some works, I am ready to do that one. Hebin, he was very instrumental in different committees, and thank you so much for Hebin. And I apologize in advance that if anyone I missed to mention. Okay, so with these words, I officially conclude the valedictory program, and I'm sincerely and wholeheartedly thankful to everyone who are here. I need to I'm so happy that it was so successful. It's like a national level, it's so successful. Thank you all. Thank you so much. Thank you so much. Thank you everyone. Thank you so much.
Thank you, sir. And finally, we request all the dignitaries, delegates, and all the persons assembled here to kindly stand up and join the national anthem.
Janagana mana adhinayaka jaya he Bharata bhagya vidhata Punjab Sindh Gujarat Maratha Dravida Uttara Vanga Vindhya हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य